Yeah, and I'll make sure I put your own, okay. I'll make sure I put this one tabletop.
All right. We're almost there.
And you want to come and listen to Daddy?
Yes.
He's going to introduce Danny. But can you hear?
Yes, we can hear you. Okay. So good morning, everybody.
You can hear now.
Have you muted everybody? Okay. I hope everyone had a restful night. There was no lodger yesterday, officially. So I feel that everybody slept, had a good night, and we can finish this out strong. Yesterday was quite a packed day, as we like to do it. I think it was quite heavy, but we got a good exposure to the other side of the research, which is trying to address impact, which is the point of why we do everything that we do. And we closed that out with 3 wonderful presentations, all trying to solve the same problem, but from slightly different angles. And I hope that during the course of this program, there's been lots of ideas, lots of potential for collaboration. And I think today will not be any different. We're going to kick off with a keynote from Yabediako. Then we'll follow up with 2 rounds of host-pathogen interactions. There'll be a coffee break, and then close it out with vaccines, bio-therapeutics, and then drug discovery mechanisms and resistance. Today we'll finish much earlier because, we try to party hard, work hard and party hard. And the party hard comes after 5. I'll make an appeal now that the room is half full, that again, we're trying to democratize the scoring, and those who got the emails for the oral scoring, please try and score the orals that you listened to, that you felt resonated with you, or all of them. Now, the change we've made is that you don't have to score everything, so you can skip. And then the posters also, that was sent to everyone who registered on the original registration list. And you didn't receive it? Okay, we'll verify that. And if you received the link, please try and score it, because all the student orals and then the posters, student posters, are being scored, and they will have a recognition during the gala. All right, so without much ado, I'm going to introduce Yao Bediako. So Yao has his PhD from Calvin University, postdoc. Oh, PhD from Northwestern. His first degree from Calvin University, and right after running around in Akosombo. And then he did a postdoc at Kemri in Kenya, followed that up with a postdoc at the Francis Crick Institute, and then he transitioned here as a research fellow, started his career here in Africa, right at WACCBIP in the University of Ghana. And along the way, he decided that, well, as head of advancement at WACCBIP, his job was basically to try and galvanize interactions between us as an academic research center and the pharma or biotech ecosystem in Ghana. And he realized that it doesn't exist. And somewhere along the line, he decided to go into private enterprise to set up a biotech company. I'm not going to steal his thunder. So I'd like to call up Dr. Yao Bediako, research fellow at WACCBIP, and also the CEO of Yemaachi Biotech Incorporated.
Thank you very much, Peter. Peter, well, I think I got here a year before you, or you got here before me. I got here, but people tend to think of us as interchangeable. We are the WACCBIP Obronis. Obroni means, white person, or Muzungu for the Kenyans here. So yeah, many times people say, "Oh, I saw you here," and I was like, "No, that wasn't me. That was the other one." So, but no, Peter is a great colleague. We've done a lot of great work together, and yeah, even throughout the journey of Yemaachi, Peter is somebody I can count on to always, bounce ideas off of or complain to, and then vice versa. So obviously we are competing with graduation, but it's nice to see everybody here. In thinking about the talk, I decided I would try to illustrate some of the things that I talked about on the panel about the need for a biotech ecosystem. Anybody who's heard me talk over the last 2 years probably is like flogging a dead horse. Like, we need biotech in Africa, we need biotech in Africa, we need to be ambitious. And so what I'm going to try to do today is present this journey of Yemaachi, but I'll do it slightly differently from what I've done. I'm actually going to try to now show you actual work. Like, it's not just talk, but there's actual science that we can do. And then also discuss the importance of partnership between academic institutions like WACCBIP and industry, and what we hope to achieve. And I'm going to put this in the context of what I think is a new opportunity for Africa, which is precision medicine. Medicine is becoming more precise. It's extremely hungry for new data. And as Dr. Pemka introduced yesterday, Africa has the most genetically diverse population. And so I think it's a huge opportunity. You could almost think of it as the next revolution in genomics should be here. We talk about fourth industrial revolution. The second genomic revolution is going to happen here. And it's going to be institutions like us at the University of Ghana and biotechs across Africa that, if we take advantage of this, they can really revolutionize our systems and generate incredible revenue, and really move Africa to where we all believe it should be in the global discourse when it comes to research and development. Oops. So first of all, I'm going to frame the problem. So Africa is in the midst of an epidemiological transition. Can somebody switch this on? I can. The screen. An epidemiological transition where we have a dual burden of disease. We still have high rates of infectious disease. Many of you study infectious diseases, malaria, HIV, tuberculosis. But then we have a dual burden now of NCDs that are rapidly rising, and in many ways outpacing infection. So cancer, for instance, kills 700,000 people in Africa now, relative to about 500,000 from malaria. So the NCDs are rapidly rising, and by 2030, it is widely accepted that we will have higher rates of death in NCDs than we have in infection. And as you can see from this graph, if you look at HIV and malaria, 2008 versus 2030. 2030 is a dark bar. You can see the trend is that we had more than we have now, or we will have in 2030. But if you look at the four non-communicable diseases, cardiovascular disease, see this pointer works. This one here, cardiovascular disease, cancer, respiratory disease, and diabetes, you can see the difference. So NCDs are trending up, infectious diseases are trending down, but slowly. And so right now we have both, and so we need to tackle both. We cannot ignore one or the other. And I think it also presents interesting research opportunities to look at the interaction between infection and NCDs. Oops, sorry. And then it's not just about burden, but it's also about mortality. And Africa currently bears a disproportionate burden of NCD mortality. So even though cancer rates in Africa, recorded cancer rates in Africa, are still lower than other parts of the world, the mortality to incidence ratio is considerably higher. And so if you get an NCD in this part of the world, you're more likely to die than anywhere else. So it's a rapidly evolving problem, and one that we need to pay close attention to. And even in developed countries like the United States, people of African descent bear a disproportionate burden of mortality. So black women are 40% more likely to die of breast cancer in America than white women. Now, some of that is social determinants, but some of it is also science. Because, as Lily talked about yesterday, we don't really fully understand breast cancer, for instance, in black women. Why is it so aggressive? Why does it present so much earlier? Because we haven't done that much research. So even in the West, and this is where the intersection begins to come in, there are problems in developed countries that are problems here as well. And I think our challenge, being pragmatic, is to look for that intersection to show how the science we do here is also relevant there. Not to devalue the lives here, but the truth is the money is there. And so we need to be pragmatic about showing value for African science outside of just Africa. Then you begin to move beyond charity towards commercial interests. And if you're not in commercial interest, believe me, you'll never attract the amount of money you need. So it's not fair, but that's the way the world works. And so we need to be able to align ourselves quite well. And I think we have a huge opportunity to do so. It doesn't take much of a stretch. If you want to study breast cancer in black women, do it in Ghana and Nigeria. You don't need, you do it in Chicago as well or in London, but there are more black women in Ghana. So why are you not doing breast cancer research here? So just to define, I talk a lot about precision medicine. It's something that many in this room I'm sure are familiar with. But I think it's really important that we understand the way medicine is going. We've evolved from a time where we had drugs or treatments that were just broadly applicable. Now we are focusing our treatments and trying to make them much more targeted. And there have been some achievements. BRCA1 and 2 have revolutionized how people think about breast cancer prevention or screening. Can you hear me? Oh, okay. You are doing this, so I was wondering, Lydia. It's not for me. Okay. I figured you were waving at me. So, Angelina Jolie publicized this when she chose to get a double mastectomy because she tested positive for the BRCA variants that had high risk, right? Pharmacogenomics, we've heard about that a little bit, about how we metabolize drugs differently. So you need to keep that in mind when you're providing drugs or providing specific doses. Genomic risk scores. These days we talk about polygenic or genomic risk scores. We're now able to predict your risk of a particular disease, cardiovascular, cancer. But the catch there is typically if you're European. There are actual tests, they have to basically disclose and say, we don't quite have that much data if you're black or brown, and so it may not work as well. And that is largely because the data that has been used to develop these scores is really from people of European ancestry. And then there's gene therapies, whether it's sickle. We heard Prof. Che gave a really great talk about the HIV cure strategy, some of them involving gene therapies. So, really, medicine has made huge leaps forward over the last 2 decades. It's a really exciting time, but the scary thing is it's not actually necessarily benefiting everyone. And I think that's where we as African scientists need to take a stand and be included in the discussion. Because these are great advances, but they will not help the rest of the world if they are not applicable across people of diverse genetic ancestries. So this is just a figure that describes, the idea of stratified medicine versus precision medicine, trying to personalize our solutions. And on that side, what I'm trying to depict is the fact that precision medicine depends on data. Everybody talks about AI, machine learning, natural language models. It's all data. Precision medicine is the same. You need large amounts of data around the disease, genomic data, clinical data, really multi-omic data, right, to begin to identify drug targets, to begin to identify biomarkers. So precision medicine is extremely data hungry and is dependent on data, and its utility is skewed by what data is available. Dr. Pemka talked about this data gap. This is another way to look at it. So if you look at GWAS studies conducted over the last several years, from 2006, you can see we didn't have very much, and then almost exponential growth. The red, though, is European. So it's 800 million total people in GWAS, and you can see what proportion of them are people of European ancestry. Let me see. I'm colorblind, so I won't try to say what color this is, but this is African, and I can't quite see it down here, right? This is East Asian, so the second bar. Africa is barely visible. Okay, so this is the data gap. Now, one of the best characterized genomic databases we have is UK Biobank. It's a fantastic initiative in the UK, recruited 500,000 people. I think nearly all of them have been genotyped or sequenced now, thanks to Genome England and some of these initiatives. 94% European. Now, the UK is more diverse than this. It's not 94% European ancestry, but still, it's a UK database, right? So it only has 2% black representation. So it's because of databases like this that we have what this looks like. But then again, these are databases in North America, in Western Europe, a few now coming up in Asia. They are skewed towards, the dominant demographics in those countries. Not always fully representative, but If this was a Ghana biobank, it would obviously look very different. But we don't have a Ghana biobank, right? And so that is why we have these deficiencies in data. Now, it's not just a representation thing. It's not just equity. You know, these days, diversity and equity are dirty words depending on where you are in the world. But it's not just a matter of justice or equity or representation. We're not saying we should have diverse data just so that everybody feels kumbaya and included. This has real-world effects. All right. So this was a study done looking at prediction accuracy of risk scores. I talked about risk scores, the way we, biomarkers and risk scores to predict a disease or to predict your risk of a disease. In this study, by Martin et al. In 2019, they looked at 17 quantitative traits in 5 continental populations in the UK Biobank. So once again, we had dependent on the UK Biobank, but at least there was some black data in there, so they managed to look at it. And what they found was that the predictive accuracy of these 17 traits was lowest in African people and highest in people of European. I don't quite know what European versus American is. I don't really know what American ancestry is, so that's a weird thing in the paper. Like, it's not definitely not Native American. They would be right here with us. But you can see that a steady decline with African people or people of African descent being the least predictive. So it means if you are an African patient and you use this test, your predictive accuracy is somewhere around 25%, right? Compared to somebody who is of European ancestry where it's almost 100%. Now that is a huge problem. So that is where precision medicine actually exacerbates disparity. So as we're getting more precise with medicine, we have to keep this in mind, because if our data is not big enough, then we actually may as well go back 30 years and just use one-size-fits-all medicine. At least then maybe everybody would be equally poorly served. But if you're doing precision medicine and if you're in this group, like many of us in this room are, this is not precision, right? This is the opposite of precision. In fact, in actual fact, it's actually potentially harmful because you'll be a false negative or a false positive, right? So another example is they studied polygenic risk scores for type 2 diabetes, which is a very big problem in people of African descent, 5 times less predictive. So the group of people who are known to get a lot of type 2 diabetes are least served by the predictive test that detects type 2 diabetes. And there are many, many more examples of this. So this is why data gaps are deadly. It's not about equity. It's not about everybody feeling like they're part of it. I mean, that is part of, that is useful. But at a scientific level, there's clear problems with a lack of data. Oops. So I decided to try to create a chart. What does precision medicine look like? And it was kind of nice to try to put this together just to kind of see where we have come from. And you can see 1998, the first precision medicine is arguable, but if you ask ChatGPT to tell you that it's Herceptin, so I figured, okay, well, ChatGPT knows best. So, but Herceptin is a drug that is used to treat women with HER2-positive breast cancer. And it's one of the first, it actually made Genentech, if you're talking about biotech companies. Genentech eventually got acquired by Roche for, I think, ultimately like $50 billion, but it was partly due to drugs like Herceptin. Herceptin is thankfully available in Ghana, provided for free by Roche or subsidized by Roche through the Ghana government. But it's a really great treatment for women who have HER2-positive breast cancer. Of course, if you don't have HER2-positive, then it doesn't help you as well. But it's a great example of one of the first targeted therapies, because it was treating breast cancer by focusing on a marker on the tumor. All right, so it could only work for women with a particular marker, but for those women, their responses were very strong and very positive. We had the first human genome published or finished in 2003. H3 Africa, which is the big hereditary sort of genomics initiative, $180 million initiative by the NIH, was in 2012. So it took that long from the human genome for anybody to really say, okay, let's do some genomics in Africa. I think H3 Africa is arguably the first real continental big push to do genomics in Africa. Keytruda, we talked about that in the panel yesterday. That's the $30 billion a year drug, right? That launched LifeArc, was discovered in 2014. And then 2020 is when a Nature paper featuring some people, not in this room, but colleagues of ours, and basically the H3 Africa investigators, demonstrated clear evidence for the value of African data. So this paper made the cover of Nature. If you make the cover of Nature, you've done something great. And in 2020, they basically demonstrated how diverse African data is. And I'll talk a little bit about what they found. But I think, as far as I can tell, this is the seminal moment where there was real data saying that African genomes are actually as diverse as we say they are. And it was because it was the first time anybody had put together over 400 whole genomes from the continent. Then in 2022, we have the T2T genome, which is the first fully complete telomere to telomere. So that's only 3 years ago. So you can see genomics is really young, and we're still really, even the technology is still quite young. But in 2025, we still have less than 3% of genomes being African. So from 1998, when we began precision medicine, 2003 having the first genome, 2025, 22 years later, we still have only 3% African genomes. And you can see all the advancements that have happened, all the drugs that have been developed, and yet only 3% African genome. So it's really showing how in 20, 30 years, how precision medicine is already skewing. And what do we need to do to self-correct that? Because if we don't, the disparities will just be exacerbated. So Lily showed us a graph like this yesterday. I won't go into too much detail. Evolutionarily, we're all African, so welcome home, everybody. And our species evolved here, right? We spent most of our lives, or our evolutionary life as a species, on this continent, and then began to migrate. And so this is not me saying it, this is widely accepted. And so Africa is home to pretty much all the genetic variation. You can think of it as a big bowl of M&Ms. We have every color, and then a few colors are elsewhere, but those colors are also here, right? So in my mind, in 2003, they should have set up a sequencing, Sangha, nothing against the Sangha or Broad, but they should have come to Lagos or Kinshasa or Accra. We are small, but we are nice people. They should have built a sequencing center here, because this is where our species comes from. This is where the lineages are. Linkage disequilibrium blocks are so small, you can find traits. I mean, all the signs point to the fact that we should have been sequencing the heck out of Africa, but we didn't. But anyway, at least we have the ability, we can show that yes, there is huge genetic diversity. And so this is an opportunity for us. And I talked about this paper, the Nature paper that came out. They sequenced 426, or they collected data from 426 whole genome sequences across diverse African populations. They discovered 3 million new variants that had never been described before, right? Just from 426 Africans. This is juxtaposed to probably hundreds of millions of European ancestry individuals. So you take 400 Africans and you find something that you've never found in millions of European ancestry people. That is the power of the African genome. And what they tried to do was show whether it would, in this paper, they tried to show the cumulative effect. So they took people from Botswana and then as the baseline, and then gradually looked to see if they would find additional variants as they added different populations. And what they're basically showing is it doesn't plateau. So even in this study, by going, they keep finding like at least 6,000 or 7,000 new variants each time they added a new geographic region on the continent. And as you can see, the curves don't really plateau. So they've not exhausted the diversity, is the point that this figure makes. So even those 3 million variants are probably scratching the surface, right? And this has bearing on disease. So by studying African populations, one of the more famous ones is APOL1. It's a variant associated with with risk of kidney disease. And the interesting thing is it appears to give you some resistance to trypanosomiasis. So by co-evolving with TRIPS in Africa, we selected for this variant that protects you from TRIPS, but then makes you more likely to develop kidney disease. This variant is also found in Europeans, it's also found in Asians. It's just easier to find in Africans, right? So it now provides a method to now identify people at risk of kidney disease so they can be surveilled. So it's not just an African trait. There are very few, you really can't call it an African trait. It is a variant that is probably easier to find here, but is applicable across humanity. And there are people all over the world who will benefit from such research. We've talked about pharmacogenomics and drug safety and efficacy, and we obviously, the idea is that there's a huge global benefit to genomics research in Africa, and I think we need to be trumpeting that. H3 Africa should not be the end. It should have happened even earlier. And the question is, where are the African-driven initiatives to really push this? Because this is something we should be marketing. People should be trooping here asking us to do studies like this. All right, so I'm going to switch gears and talk about Yemaachi. I talk a lot about Yemaachi. Many of you are probably tired of hearing about Yemaachi. I should say, this is my first keynote at WACCBIP Research Conference, so I've arrived. I think my first conference was 2018. It's taking me this long to earn a keynote. I had to set up a company to earn a keynote. But that also shows how competitive this conference is. You don't just take anybody. So, I mean, I'm going after Emma Wall and Prof. Che. I already feel a little bit, insecure. But yes, at least if you want to get a WACCBIP research conference, either become a professor or start a company. So anyway. But I'm going to tell you about Yemaachi, but I'm going to try to do it in the context of work we've achieved. So this is a scientific talk, so I'm going to actually talk science as opposed to my big vision for Africa, which you're probably all tired of hearing. But for those of you who are not as familiar, Yemaachi is a company we started began operations in 2021. We're based here in Ghana, but we have partners across the continent. We actually now have a subsidiary in Kenya and one in the US. So I'm actually now a group CEO. It's kind of exciting. Anyway, but what we've tried to do is build a team with considerable skills, very diverse skills across life sciences, across technology, across medicine, and then partnerships. That is a huge thing in working in Africa. Yemaachi is a small company, but we have partners in 9 countries. Many of these are clinicians and hospitals, people who are treating people with cancer. We've built quite considerable in-house capacity and then also tap into the capacity at WACCBIP. So, I'm a research fellow at WACCBIP. Yemaachi was born out of WACCBIP. Prof. Awandari gave me the freedom to try to do this even as I was still a fellow actively teaching and involved in research here. And so we work very, very closely with WACCBIP. And we're now at the phase where we're now trying to transition all these relationships and this capacity into products and services. At the end of the day, we are a business. Like Lord said yesterday, if you don't make money, you die. And so our vision is, how do we take our science and make it commercially viable? We don't do diagnostics, so we're not in that same space of making diagnostics. In our space, we are a data company. We are generating the data that I said the world needs, and we're trying to do that from Africa in a commercially sustainable way. And I'll show you a little bit of what we're doing. I talked about our network. I'm extremely proud of this. The light green is where we currently have relationships. 9 countries. Many of these, if you look, are partners that are linked to WACCBIP because that was my personal network and I took advantage of that. But they're all in oncology, which has pushed us beyond the infectious disease area. So Senegal, Cote d'Ivoire, Ghana, Nigeria, Tunisia, Uganda, Kenya, Zimbabwe, and South Africa. And we're currently in discussions in Rwanda, Ethiopia, Mali, and Morocco to continue to expand the network. My hope is if I came to give a talk in 2 or 3 years, the whole map would be bright green. That we will literally have a presence, or at least would have partners across the whole continent. In fact, right now we're about to sign an MOU with the Seychelles, which I'm excited about because I get to go to the Seychelles. So it's, But what is really cool is these are access to cancer patients. So we are building an oncology network. In fact, I believe that this is arguably the most diverse oncology network in the world because it's across the most diverse continent. So we are linking hospitals in Meru in Kenya in Kano in Nigeria, all right, with Kolebu in Accra, with Saashu Trècheville in Abidjan, all right. And we are trying to link up these clinicians so they talk to each other, but then also collecting data from these populations and seeing how varied they are, what drives cancer, different types of cancer in these areas. So with this network, we have access currently to over 100,000 new cases across our current network. So we have over 25 institutions, mostly hospitals, linked in this network. And we now have over 100,000 new cases over the next 5 years. So that is the foundation for that data, right? We're trying to build big data. You need access to big numbers of people, and the network has provided that. So I told you I was going to talk about science, talk about productivity. This is our first paper we published in 2024. So I told you the company started in 2021. We had a few commentaries and so on. This is our first peer-reviewed publication. It came out in 2024. And was a small study we did in Cape Coast, which is on the west coast of Ghana, about 3.5 hours drive from here. And we did this as a liquid biopsy study. So we partnered with a San Francisco-based liquid biopsy company. Liquid biopsy is a blood test for cancer. And we wanted to see whether we could find actionable mutations in a Ghanaian population. Most liquid biopsies, you've heard the big stories about Grail and all these companies. Very few of them have been trialed on predominantly black populations. So we recruited a small number of women with breast cancer from Cape Coast Teaching Hospital, collected a biopsy and collected a matched blood sample. So we wanted to compare tissue with blood to see how representative blood would be of the tissue. And so then subjected them to, in this case, actually, at this point, we were not yet doing our own sequencing. And the group, the panel is a proprietary panel from Lucence, which is a Singaporean company, San Francisco Singaporean company we worked with. So we collected all the samples here, did all the QC here, but then Lucence did the sequencing in this case. Funny story, the CEO of Lucence he was a postdoc when I was an undergrad in the US. And he started a company, and then when I started Yemaachi, I reached out to him and I was like, hey, Minhan, I've started a company too. And that's how we had our first project. So he was teaching me how to do immunohistochemistry when I was basically a graduate intern a few years ago. And that's how we had our first project. So you never know who you meet, is what I'm trying to say. This was a very important first for us because it was a legitimate project with an up-and-coming Singaporean and San Francisco-based company. But anyway, what did we find? We found that 30% of the women had actionable mutations. Those are the ones with the asterisks. We also learned that how you handle your FFPE samples is really important. Many of our samples failed QC because they were overfixed. And so the DNA was just destroyed. So we were working with we were working initially with samples that had already been fixed in the hospital. We had to completely change the way the formalin was prepared. So we provided buffered formalin for them to use for the blocks, and after that QC was much better. But we learned an important lesson, that if you don't worry about how your samples are handled, you end up really struggling with genomic analysis. But from a research perspective or from a results perspective, yes, 30% of the women had actionable mutations, but that means 70% of them didn't. And the question is, okay, so what are the mutations causing these cancers in these women, and how can we develop drugs to address this? But you can see some of the well-known TP53, BRCA2. So it was neat that this was working, and the idea of the conclusion of the paper was this would be useful in our context, but there were also a number of women who would not be helped by this because the mutations they had were not currently considered druggable or actionable. And then it also raised the question of how many of the drugs are actually available locally. And unfortunately, many of these drugs are not. So they would be actionable if they were in the US. But in Ghana, those drugs are not here. But at least we showed that we could find the woman, and so you can make a case for making those drugs available. So yeah, this is just summarizing, so we could detect the paired liquid biopsy and tissue were comparable, so the test worked. So the blood was reflective of what was in the tissue, provided the tissue. In fact, we concluded that because of the challenges with fixation, blood was actually a safer method. Because if you collected blood into a Streck tube, there was no fixation. And so you were more likely to actually, so it made the argument that liquid biopsy in resource-limited settings, liquid biopsy as a way for cancer genomics was actually more preferable than FFPE-based genomics because of the problems with fixation. So this was our first study, and we're excited. 2024, young company, at least we have a peer-reviewed publication out, and we show that we can begin to do work locally. Then we said, okay, we want to understand all these mutations, so we launched Brexome. Brexome is basically breast cancer exome. So at the time, we wanted to do whole exome sequencing across multiple sites. We partnered with 5 different countries and facilities there, so Tunisia. So we've actually sequenced breast cancer from Tunisia and Accra, which is really neat. So Tunisia, Cote d'Ivoire, Ghana, Nigeria, and Kenya. So far, we've actually recruited well over 1,000 people into this study. We've not done all the sequencing yet, but samples are coming in. In this case, we went back to FFPE because we had a retrospective component. So we knew there'd be some attrition. Some samples will not make QC, but if we recruited enough people, we still have enough, we thought. And the idea was to do somatic whole exome sequencing of tumor samples to really begin to try to identify novel mutations. Because we already knew that the panels would miss a large number of people. So the question was, can we find mutations in known genes? Can we find new mutations in new genes? So this study is actually ongoing, but there is some data. This manuscript is in preparation. This is from northern Nigeria. So we work in Kano and Katsina, the hotbed of Boko Haram, but we are not scared. Well, actually, I am scared, but my team is not scared. But we found the doctors there to be incredibly knowledgeable, incredibly interested in research, and they have great facilities. I mean, they have -80 freezers, they have a whole molecular lab, they can do their own DNA extraction. I mean, this is in a hospital. In fact, those hospitals will put some of our hospitals in Ghana to shame. But anyway, this is data analyzed. I should mention, everybody knows that part of why I stay here is to recruit. So this data is analyzed by Charles Owal, who is a PhD student, my PhD student actually, who's graduating today. So he's in Ghana. Our PhD, yeah, whatever, yes, fine. Our PhD with my work husband. So one thing is, as I started Yemaachi, Peter graciously accepted to co-supervise with me because, as my students know, I'm hard to find. So Peter has really supported me very much. I should give him credit for that. So yes, our student Charles, yes. We don't have a custody battle. Anyway, so this is data showing the mutations we are finding. So it's difficult to see. This will hopefully be out soon. We're writing it up. But we found mutations in TP53, PIK3CA, so some of the known canonical genes. But actually what we're finding was the mutations we're finding were more prevalent in genes. The genes that were mutated were different from the ones that you'd find in the COSMIC database. So they are known cancer genes, but the ratio of genes the ratio of different genes would differ from ancestry to ancestry. And so here we found that there was actually higher mutational burden in the African samples. So for TP3, PIK3CA, called 4A5, and so on, we found 41% versus 27%, 21% versus 7%, comparing the ratios in our sample to what was in COSMIC. Now, obviously, this is 40 people, so it's relatively small, so we have to be careful how much we extrapolate this. But certainly, it appears that there may be differences in the mutations that we find in breast cancer. The truth is, nobody has done this. So 40 whole exomes from Nigeria is probably the first time that we've done 40 exomes from Nigeria, at least certainly sequence them on the continent. So we need to build up much bigger data sets, but this is still showing that there is something to look for. And then we were also finding novel variants. So the top mutated genes, even if they were known genes like BRCA, we were finding novel variants. And this is important for diagnostics because PCR diagnostics typically look for specific variants. So they won't sequence the whole of BRCA. They'll look for a particular mutation in BRCA. But it's possible that in our populations, BRCA may be mutated and it may be a missense mutation, but it might not be the one that is in the panel. So this work is really important when we think of diversifying our precision panels as we build better diagnostics. So stay tuned. We have a bunch more data coming, so we're probably going to preprint this and then add a whole bunch more whole exomes before we publish, but it'll be out for you to read soon, authored by Charles and a few other people. This is another study. We call this precision medicine on a budget. So we've talked about pharmacogenomics. We were interested in seeing whether, even though there's limited African data, can we go into databases and pull out genes known to be involved in drug toxicity or drug efficacy and compare their relative allele frequencies in different genetic ancestries. Basically, can we find evidence of the fact that if you don't do precision medicine accurately in African people, it could be dangerous? So we picked two cheap and commonly available cancer drugs, capecitabine and cyclophosphamide. And we basically just did a screen. This was led by Lindsay and Charles. They were actually doing their PhD here, but then we had an internship program. So this was their internship project. It was accepted 2 days ago. So really exciting. We finally got over the line. It's just been accepted for publication. And Dr. Pemka was instrumental in guiding this process as well. She is the corresponding author on this paper. And so this study was basically, can we identify drugs, genes known to mediate toxicity, and do their allele frequencies differ? So it's a little bit difficult to see, but what I'm showing here is, I think this is Black American and African, and then Caucasian. I can make this available to you so you can read it later. But the idea was, for certain genes, we found that, yes, indeed, the allele frequency was different. So these are genes known to be involved in mediating toxicity to capecitabine and cyclophosphamide. And we find that, indeed, they do differ. So it means if you're a cancer patient and you're African, you probably want to do this test before you administer these drugs. And yet we're administering these drugs without any. From Europeans or to Asians, we do see a difference. Interestingly, in some cases, we found differences between continental Africans and African Americans. Of course, African Americans are an admixed population. So it shows once again that you cannot just necessarily extrapolate African American data to African. Right? So this was an in silico. We didn't do any of our own sequencing. It's just based on existing data. But I think it provides good evidence for the kind of work we are doing. All right, so then I'm going to talk a little bit about PROGRESS. So PROGRESS is a pediatric cancer study. Now, I don't know how many of you are aware that 80% of childhood cancers occur in low and middle income countries globally. So 80% of the children with cancer are in low and middle income countries, many of whom are in Africa. right? Additionally, whereas for a disease like leukemia, in the US, survival is over 90% for children. In Ghana, it's below 30%, right? So children are dying in Ghana every year. In fact, it's estimated about 1,200 children get cancer in Ghana every year. Many of them are dying because of where they are, right? Not because of their disease. If they were in Europe or North America, they'd probably survive. Children are incredibly resilient when it comes to cancer. And so we launched this project in partnership with the Pediatric Cancer Fraternity, or actually sorority. It's almost entirely female driven, in Ghana. A project led by Prof. Kathy Segbefia at University of Ghana Medical School in Kolebu, with advisory help from Professor Lorna Rena, who is basically the mother of pediatric oncology in Ghana. And we partnered with the 3 hospitals in Ghana that treat children with cancer: Kolebu, Komfo Anokye, and Ridge Hospital. Basically, every child in Ghana who gets a cancer diagnosis is going to be at one of those 3 facilities. So we had a great opportunity to have a funnel. And the idea for Progress is to sequence every child with cancer in Ghana over the next 5 years. We want to build Ghana Childhood Cancer Biobank, or Genome Bank. And the reason we wanted to do this was obviously the burden of childhood cancer is in this part of the world. And it's also children with cancer. And if you're losing your children to cancer, what hope do you have as a nation? So we got some support from LifeArc for this, because this is not a commercial project. This is entirely research-based. And we also got support from Nanopore. And since November 2023, we've successfully recruited 380 children. We've collected detailed clinical data, collected multiple biological samples. One of the things we've achieved in the first year is a pilot. We're trying to get more money from LifeArc now to continue this. But for the first year, we introduced flow cytometry. So we have flow cytometry capacity at Yemaachi. As an immunologist, I was horrified to find out that they are treating leukemias based on morphology. Under a microscope, you cannot distinguish a T cell from a B cell, right? But that is what we're doing. So we're treating children, and the treatment regimens may differ, depending on if it's a B cell or a T cell leukemia. Hematologists in the UK and the US would be horrified if they had to work under those conditions, but that's what our doctors were facing. Flow cytometry has not worked at Kolebu for years. So we introduced flow cytometry for our research participants at Yemaachi. And so whenever a child was suspected to have leukemia, a sample comes. Within 48 hours, Eliza, who is in the audience, would run the flow cytometry, and we'd revert the results back. We shortened diagnosis time from 12 days to 2 days. So now children with leukemia were being diagnosed in 48 hours, compared to almost 2 weeks. And we did this simply by introducing flow cytometry. So I think that shows the power of small interventions and how a company like ours could have an effect. We're currently working with Nanopore to develop an NGS assay, working with colleagues at the University of Newcastle to develop an NGS assay to measure measurable residual disease. After treatment, you want to know whether or not the children have blasts, right? Leukemic blasts remaining after chemo. In the US, they measure MRD, and that determines whether or not you get additional chemo or whether you can tail back. Many of the children who die not because of the cancer, but they die because of the immunosuppression and the chemo, right? Many of them get infections. Many of them are immunocompromised because you're basically wiping out all their white blood cells because they are cancerous, right? But if you knew whether or not you got rid of the cancer, you could decide whether to give them more chemo or less. That happens in Europe, happens in the US. It's currently not happening here. We are trying to develop an assay that would allow us for the first time offer MRD testing in Ghana. So that's one of the outputs of Progress, to show it can be done here and to show that it can be scaled. So this is ongoing, but even with just flow cytometry, we've already shortened the diagnostic time for leukemia in Ghana from 12 days to 2 days. This is Collins's data. So as you know, Collins also works at Yemaachi. This is sequencing. So for the first time, we've sequenced whole genome sequences, long read whole genome sequencing of leukemia patients. So this is bone marrow from a child with leukemia, and we actually were able to determine the gene fusion that is responsible for their leukemia, mapping the breakpoint. So the beauty of long read is you have longer reads, and so mapping structural variants is much easier. And so this is why Oxford Nanopore is so excited about this. Even in South Africa, I don't think they're doing this routinely. And so this is one of the first times in Africa that a child with leukemia was sequenced, and that child was sequenced in Accra, right? And data analyzed by WACCBIP scientists, right? I think that deserves a clap. Yeah. So then the neat thing is we're able to show, it's a fusion between chromosome 1 and chromosome 19. This child's parents had actually been able to afford cytology. So the sample had been sent to India for a molecular test, not a whole genome, just a molecular test. And this is a well-known fusion in B-cell leukemia. That confirmed it. So at least we had a diagnostic confirmation of our NGS sequencing. Which I think is important because obviously also nanopore NGS is research use only, which is what they will always tell you at this point. But by doing this in the research context, we're able to begin to map these, and we're going to find new variants, right? We're going to find new mutations, new structural variants that are important for this. So it's really exciting. This is ongoing. We have tons of data that we are trying to get through, and we hope to have a publication coming out soon. And we're hoping that this will provide LifeArc with enough incentive to fund us for 4 more years so we can really do this properly and follow these children. We're actually looking now to include an AMR assay to try to track AMR, because a huge user of antibiotics in our hospitals are children with leukemia. And many of them have drug resistance. So those of you who are infectious disease, people interested in bacterial infections, a huge population you should be looking at is children with leukemia, because they are on all kinds of antibiotics and many of them are drug resistant. So there's a huge opportunity there to understand AMR in the context of pediatric cancer. And that's something we're excited about working on. I'm almost done. Emma did a really good job of heritage, but I felt I had to show the paper, especially because my boss is there and my friend is there. But it also gives you an idea of the people. This is a study funded by the Gates Foundation in a fellowship to me, but to Yemaachi as a company. So one of our first big grants was this Kalestos Juma Fellowship that I applied for. All these people here are staff at Yemaachi, some of whom have gone on to, like Barakisu has gone on to do a PhD. Harry is also doing his PhD. He was here this week. But he's just Eliza, who was first author, who has done a lot of the immunology work. Emma talked about the results that we found. I'm just showing one figure, comparing legacy in London with heritage in Accra. You can see the maintenance. The maintenance of the response over 12 months. So we basically recruited 300 adults, healthy people in Accra. We sampled them 7 times in 12 months. We had almost 0% dropout. And that is a testament to the Yemaachi. WACCBIP has a WACCBIP machine. Me too, I'm building a Yemaachi machine. And that is a testament to, we followed them up bi-weekly. We kept them engaged. These people were healthy. They had no reason to show up and get a blood draw, but they came. And we got really extra exciting data. And you can see basically in individuals who, as Emma pointed out, in people who are nucleocapsid negative, meaning they'd never been exposed, they looked a lot like the legacy cohort, versus individuals who were nucleocapsid positive, even though never reporting a symptomatic episode, but they were nucleocapsid positive, meaning that they had antibodies, meaning they had been infected at some point. Their antibody response, this is baseline before the vaccine, and then 3 months after the vaccine, you can see an increase, and it maintains over 12 months. Whereas the legacy group and the nucleocapsid negative group follow a much sort of a lower. They all, in the end, catch up. And legacy obviously with boosting. You can see actually here with Omicron BA2, there is a difference between the nucleocapsid negatives get higher than the legacy, probably due to environmental exposure. We had lots of COVID here. But this showed that in our case, previous exposure and then getting the vaccine was a very advantageous thing. In the end, actually getting the vaccines late may actually have helped us, because there was a bit of premonition followed by a vaccine that really acted as a boost to our immune system and has led to very, very well-maintained antibody responses. All right, so final piece, where are we going? I said we need big data. So yeah, Yemaachi has recruited about 2,000 cancer patients over the last 4 years. We have 40,000 samples in our biobank, obviously multiple samples from the same individual. We've done some sequencing. But the truth is, you saw that GWAS curve. I mean, we're talking about millions of data points. Now, if you do disease-specific work, you can maybe have slightly smaller populations, but we still need tens of thousands of data points in specific diseases to really make a dent in the issue of the lack of African data. This is the American, probably gold standard cancer genomic database, TCGA. It's smart because they use the nucleotide letters. The Cancer Genome Atlas. NIH initiative. Took them 10 years. $370 million. They sequenced 11,000 cancer patients twice for 22,000 genomes at the basic level. Now they've added a bunch of other multimodal transcriptomes and so on top of it. But this is a publicly available database. You can look for it. Biggest problem, almost 80% Caucasian, about 73% Caucasian. It's 8.9% African American, but it's spread across 33 types of cancer. So for 30 types of cancer, you literally have 5 people who are black. So that is its huge Achilles heel. So it's very useful if you're European. It's much less useful if you're not, right? But this is what all the drugs that I talked about, many of them have come from this, right? Many of them have come from analysis of TCGA data. In fact, you can check online. You see many drugs that are currently available have come from TCGA. So how do we generate big data? Well, let's look at how the Europe and North America does it. Turns out the government spends money, but they don't spend all the money. UK Biobank is a partnership with the UK government, Wellcome Trust, GSK, Regeneron, Amgen, AstraZeneca, Biogen. These are all huge pharma companies. FinGen, AstraZeneca, Genentech, Merck, Pfizer, Sanofi. All right? It goes on. So even H3 Africa partnered with Illumina to create the H3 Africa chip. So there's a clear evidence for pharma, industry, and government, or industry and academic partnership, even in some of these big biobanks that currently exist, right? So the question is, can Africa do something similar? Can we find ways to establish these databases by partnering between public sector and the private sector? And so at Yemaachi, we decided to try. So in my philosophy, if you build it, they will come. So we're going to build it. So we're building TACA, which is the African Cancer Atlas. I couldn't figure out a way to put a G in it. I was like, maybe I'll try and put Ghana or something like that. But in the end, I just gave up. TACA it also sounds catchy. We just don't have any G's. So our nucleotides are a little bit off. But anyway, we're building TACA, which is the African Cancer Atlas. The goal is 15,000 genomes in 3.5 years. I told you, NIH did 22,000 in 10 years. We think we are better, so we'll do 15,000 in 3.5. Maybe better or crazier. But anyway, the ambition is 3.5 years, 15,000 genomes across a variety of cancer types, but importantly, across the African continent. No genomic database I can find is across national boundaries. UK Biobank is UK. TCGA is US. FinGen is Finland. No one has built one across national boundaries. This, in my opinion, arguably will be the first. It also means it's very difficult, right? But this would be huge if we can go across Africa, 54 countries, and at least have representation across different regions. The question was, how are we going to fund it? I don't have, it will not cost us 300 million, but it will still cost us several million, which I don't have, or we don't have. So we went to pharma. We went to Roche, and I pitched to Roche that I know you need this data. Oncology is a big part of your pipeline. You don't have diverse data. In 5 years, I've proved that I've created a pipeline and the relationships that can generate this data. Give me money to generate the data that you want. It sounds crazy, but they bought it. So at the end of last year, I didn't have much of a Christmas break. We're negotiating throughout, literally, the 24th and the 26th of December. But we signed a contract with Roche for the first 33,000 of the 15,000, right? Because Roche has an affinity for breast and lung cancer, that's where we are prioritizing to begin with. But this is the first step. The goal is to hopefully use Roche as a carrot to attract other pharma into a consortium. So we're trying to form a pharma consortium to build TACA. It has started because we have money from Roche, and so we're funded to do the first 3,000 genomes. Even if we stop at 3,000, it will be the largest cancer genomic database in Africa, but we're not going to stop there. And so this has started. We're recruiting in Ghana. As I speak, samples are coming from Cape Coast to Yemaachi. I think samples came yesterday or the day before. Some of my team are in the audience, so if they look tired, it's because they are working till midnight processing samples. We're getting samples from Cote d'Ivoire. We're getting samples from northern Nigeria. We're getting samples from Kenya. We're about to get samples from Zimbabwe. So TACA has started. We have to deliver the first genomes by the end of this year. That's the part about being an industry. Pharma have really strong deadlines. It's not academic. Oh, my reagents didn't arrive. They don't care, right? So that's the pressure. That's why I'm rapidly aging. But we are going to deliver the first genomes, or should I say, Joyce is going to deliver the first genomes by the end of the year, and then we'll continue from there. Updates. This is our new lab. It's in Abelemkpe. We moved, but just down the road. So some of you have come. So I just threw in a couple of pictures to show you what our lab. That's our biobank. We'll continue to grow it. And our big achievement this month is we're installing a NovaSeq X Plus to allow us to do this. So the point is, by signing this contract with Roche, it allowed us to now scale up. Now, it still means I have to find additional money because Roche alone is not going to pay for all the capex. But by signing this project, by having the ambition of trying to do TACA, it has forced us, by hook or crook, to expand. So we've doubled the size of our lab, and we've installed the 7th NovaSeq X Plus in Africa. Nigeria has 2, South Africa has 2, Ethiopia has 1, Morocco has 1, now Ghana has 1, right? And that is huge capacity for us as a country and also for the region. And so if any of you are doing human genomic studies, we are open for business. You can come and we can talk. And the idea was to complement WACCBIP's capacity. WACCBIP has great sequencing capacity, especially for pathogens and for targeted sequencing in humans, all the way to the NextSeq 2000. Now with the NovaSeq, projects that require whole genomes can now be run at Yemaachi. And so I think that collaboration continues. There's no point us getting a NextSeq 2000 when WACCBIP has one. But instead, if we can get a NovaSeq, now we can do certain things and support projects here as well. So this is currently being installed, very, very exciting. And the first data will be coming out very soon. So in conclusion, I think genomics is a huge opportunity. The next revolution is here. It's already started. And those of us, all of us can be part of it. Whether you are a geneticist, an immunologist, a parasitologist, there's an aspect of African genomics that I think you could plug into. But we need to do it ourselves. We can't wait for it to happen. The advantage will only be ours if we initiate. You will find partners. If you build it, they will come. We went to Roche. This is not a Roche project. The press release that went out said Yemaachi and Roche announce. But it's a Yemaachi project. I lead that project. Roche meets with me. They've given me money, so they have me by the neck a little bit, but it's still my project. All right? It is our project. What I should add is TACA is going to be open access for African universities. That is something we fought for. Roche would not have given it if we didn't ask for it. But because we built the leverage, and they know they can't get it anywhere else, we said, yes, we can be exclusive with you and a few pharma companies, but we want to maintain that database so that students at the University of Ghana, students at University of Nairobi, students wherever in Africa can log in and use that data for their research. And so that's something that we've achieved. Right? So I think that is possible when you step out and you say, it is our data. Yes, we can be commercial about certain aspects, but because it's ours, we also want the right to use it. And Roche have agreed, and they're very happy, and they've announced it. It's like good PR for them, right? So that's the leverage. But if we had not initiated, and they had said, oh, we'd like some African genomes, could you help us? They'd have taken the genomes, done what they would like with it, and we would not necessarily have been able to extract the same value. So ownership is very important. The data is owned here. They get a license to it, but it's ours. Right? Inclusion of Africa is not an ethical favor. It's a scientific and commercial imperative. And I think that's the argument we need to make. I'm honestly quite tired of the equity argument. Equity is important. I believe in it. But it doesn't make the world go round. Money makes the world go round. We have to be pragmatic. All right? And so while we champion justice, I'm all about that. As scientists, we also have to be strategic. So when you make an argument for your project, make the argument about the value it brings, whether it's scientific value or commercial value. Don't forget that aspect. It's always good, yes, to support equity, but if the only arguments Africa makes for science are equity-based arguments, then we're always going to be looking for charity. But if we can make value-based arguments, then we begin to get commercial and scientific input and scientific interest. And I think that would be much more sustainable, because we are more than just a charity case. We are not, in fact, we are not a charity case. We have a lot to offer, and so let's put that out there. All right, so I'll end there. Thank you so much. If you wonder why we do Yemaachi, I want to be Novo Nordisk one day. Novo Nordisk is in 2024 was worth $626 billion. The Danish is a Danish company. Denmark's GDP is 410 billion. Without Novo Nordisk, that year Denmark would have posted negative GDP growth. Thanks to Novo Nordisk, Denmark posted 2% growth. You can imagine how the CEO of Novo Nordisk can basically walk into the Prime Minister of Belgium, Denmark's office anytime. Because basically, if he decides to move his company out of Denmark, Denmark's economy goes down, right? But that's just one company in a country of 5.9 million people. By comparison, Africa is 1.4 billion. Our GDP is only 3 trillion. The US's GDP is 25 trillion. Japan's GDP, Japan is a very small country, 4.2 trillion. If we have 5 or 6 or even 3 Novo Nordisks in Africa, that would be powerful. So I think that's the challenge. Not all of us can be entrepreneurs. Not all of us should go and start companies. But this is the power of biotech. So if you ever need to wonder why commercial in science is important, this is why. But it's not just about money. It's also about influence and also about leverage. If we don't want to be left behind, then we need to lead, right? And so this is where we can lead. The genome is ours. They can't take it from us. We were born with it. It's our birthright. But if we don't study it, we'll never extract the value. So thank you. I acknowledge my team, many of whom are in the audience, many of whom are at multiple levels. We have people who join us as national service persons all the way to postdocs. Not all scientists. So I have to acknowledge the admin, the finance teams at Yemaachi, as well as our engineering and computational teams, all together come together to work. And then, of course, our clinical partners across Africa, and indeed across the world. We have partnerships now with Dana-Farber in Boston. We have partnerships at the CRIC. We have 3 MIT students who have come to Ghana to do internships. Now, isn't that cool? MIT students leaving America to come and do an internship in a tech company in Accra. We've arrived. Well, not quite, but we're getting there. Anyway, so thank you so much for your attention. And this is our new office if you are looking for us.
Thank you, Yao. You threw our schedule for a loop, but I think it was well worth it. So we'll take some questions.
Thank you very much, Yao. That's a very exciting talk. Very much enjoy. I really like your boldness and the way you tackle precision medicine. I remember when I started pitching about precision medicine, I got bullied that you are in a continent where you have one doctor for 10,000 patients. How can you be talking about precision medicine? So I really want you to give me a perspective of how, after finding this diversity, we are still stuck with one doctor for 10,000 patients. How will you drill it down so that it really has impact? Because, I mean, are you saying we are finding ourselves in a context where each patient will have to be sequenced to tailor the treatment, which will not be possible because, again, we have one doctor for 10,000 patients. So the way I've really been thinking about it is stratified medicine. And, but, you know, it's a loaded word because, I mean, the parameter for stratification, you have to define it. So just give me a perspective about that, because I keep talking about precision medicine, but the applicability is something that we really have to, you know, digest.
No, thank you very much. I think it's a very good point. Yeah, it's always the challenge in Africa. Like, you don't have roads, so why are you sequencing genomes, right? But I think it's a false dichotomy. We shouldn't have to choose. So I was in Kigali and they were talking about, you know, Malawi spends, I think, $4 or $45 per patient per year, and America spends $4,500. And so they're talking about cost efficiency and saying that certain things don't fit in the 45. And I said, well, then grow the basket. Like, why are you forcing yourself to spend $45 on a patient? We shouldn't be spending $45. We should be spending $400, right? So I think we need to be the audacity to overcome what one of my great heroes, who unfortunately I never got to meet, is Paul Farmer. And he says, overcome failures of imagination. Africa has to overcome failures of imagination. Because if Prof. Awandari had looked around, he would not have started WACCBIP. Like, he came here, there was one PhD student over the last 5 years, right? Why would you bother? But now we are all sitting here because he had that dream and he found colleagues who worked with him, because he decided that he wasn't willing to accept that, right? So I think each of us has to overcome that. I fully agree with you. The challenge, precision medicine implementation in Africa is going to be the hardest in the world. Not because of economics, but because of our genomics, right? We are so diverse that, wow, it's going to be a challenge. But that also means we can teach the world how to do it. We're teaching the world how to do mobile money, and Pesa in Kenya is better than most credit cards. I mean, the mobile inclusion in Africa, we never had landlines. We went straight from no phone to cell phone. And we basically, you know, that's the classic leapfrog, the example everybody gives. I believe that I may not have the idea myself, but there are people in Africa who will figure out how to have a blended, stratified precision medicine model that will work. We just have to create the data and have the vision to do it, right? So I think that is, right now, that's where we all need to be. We each, in our corner, identify the problem. So for us, you can't do precision medicine without data, so I'm trying to solve that problem. Once I have that, perhaps I'll have leverage to go to pharma and say, make the drugs available. But of course, at the end of the day, how do you stratify? We will figure it out, right? Because we must. So I think we shouldn't let the scale of the problem hold us back. Instead, you pick an angle, you chip at it, you do it excellently though, because we cannot afford failure either. So we don't do it half measures. We go full bore, 100%, and you strike for Mars, and hopefully we end up somewhere. So I really think it's a matter of a lack of ambition. And if we can overcome the lack of, it's not really a lack of ambition, but it's a fear. So if we can overcome that, I think we will lead the world. I mean, I think Africa should be leading the world in human-based studies, right? We have all the disease cohorts here. You don't need a mouse model to study malaria. You have children with malaria, right? But how do you develop the technology, the AI models to do it properly? Because obviously what you can do in a mouse, you cannot do in a child. But we should be leading in those areas. So I think it's certainly possible. Yeah.
We have a question online. Is
Sure.
that okay for me to? So, how practical is genomic medicine in real world, given the enormous cost associated with it? For instance, not all breast cancers are the same, and cancer drugs cannot be used for all breast cancer patients. And in some cases, molecular profiling of the cancer is needed for proper diagnosis. How soon can this be implemented in Africa, or implanted in Africa?
So I think that follows on with the previous question. I think, yes, it is challenging, but if we don't start, we'll never get there. I think there are ways to begin. I mean, Herceptin is in use here. Probably at some point, people thought that we could never even use Herceptin because we can't do immunohistochemistry. But we're doing it. Herceptin is available for free for women with breast cancer in Ghana. There's no reason why the PARP inhibitors should not be. There's no reason why tyrosine kinase inhibitors should not be available. Yes, we need to build out the molecular diagnostics, but that's a whole industry also that could emerge, right? So I think we need to recognize that as we develop the science, it could also develop industry and develop enterprise, which ultimately the problem we have is poverty. If people are not poor, they can afford it, right? So how does our science begin to transform our economies? I think that's a challenge each of us has. But once again, you start somewhere. During COVID, they said we'll all die. Turns out our antibodies are better. So, I mean, as far as I'm concerned, we just get it done. Like on Prof. Andres, though, I said, no excuses, get it done. That is the mindset that every African has to have, right? And if we have that, yesterday I was at a meeting and they're talking about the resilience of Africans who were taken as slaves. They survived terrible conditions, went there and were still strong enough to work on farms, right? We have it. So we shouldn't allow ourselves to be held back. It may take time, we'll have to be deliberate about it, but I'm a firm believer that we will get there. So we just have to take one step. And there's evidence. We've achieved so much already. There's no reason why we can't. Any other questions? Yeah. Oh, please, Abdul.
Interesting talk, Yao. So thank you. The cancer change and cancer cells also evolve with time, which means some mutation you identify this year may not be the most prevalent next year. So how are you considering this longitudinal aspect of evolution? Yes.
Very good question, the longitudinal aspect. So TACA as currently designed is phase 1. I mean, if you ask me my vision, TACA is going to end up being a foundational model. If people are in foundation, like we're going to build the cancer foundational model from Africa. I mean, you have to dream big, right? So I think we are already building, because of the way we've set up our cohorts, we are building longitudinal cohorts. Uvosio is our cloud-based tool for collecting clinical data that we built ourselves. It's better than RedCap, so if anybody's interested. But the idea is we are creating longitudinal virtual cohorts of cancer patients across Africa. And on the back end, we are standardizing the clinical data so that it's compatible. So we're linking it to OMOP, which is an international standard. Because the idea is if I collect data in Cote d'Ivoire, collect it in Ghana, it's French and English to begin with, but then also the medical systems are a little bit different. So the same thing is called two different things. So if you just collect it based on the terminology the hospitals use, you can't mine that data. So what we've built into Uvosio is specific codes that will standardize it on the back end. But that means now we can create these huge cohorts across the continent and follow people. In some cases, we are collecting longitudinal samples. It's all obviously it's driven by resources that we have. But I think the platform is there. And that's what I mean by Africa is where we can do this kind of work really, really easy. Well, not easily, but really, really successfully if we apply ourselves. So I fully agree. And I think some of these new mutations, then, in 5 to 10 years, the new drugs for cancer will come from this continent. And we just have to start. So I think, I fully agree with you. I'm an immunologist. I can't wait till we are doing real immunotherapy work. I mean, the genomics is low-hanging fruit. To be honest, doesn't quite get me going. The immunology really does, but you have to start somewhere. And so we're looking to build multimodal data. And we're beginning to pitch that to our pharma partners. We're saying, hey, why are you stopping at the genome? We can do a transcriptome for you. We can do a proteome. Now they want us to do digital pathology. Before you know it, they'll buy me a mass spec and say, do a proteome, right? Pharma have deep pockets. They are extremely hungry for data. We need to just position ourselves as the place for them to work. Clinical trials. I know UGMC is building a clinical trial facility. We need to recognize our place, but we have to build it first. Right? We take one step, they'll help us build 10. So, yeah, I think we are on that path. We're trying. Yeah. Yes, please.
Thank you. Thank you. I see you're on fire, and
always.
I will start by saying that learning from the European genomes how to do translation, is for me is not a negative. I can imagine if we started from Africa, we probably would have been discouraged and probably lose steam on the way. But the opportunity is there in Africa, and the challenges are myriad. They are enormous. So I just wanted to pick your mind on where your worries are, because from genomics to translation is much longer in Africa versus Europe. And pharma companies are investing in Europe because they get returns in a shorter time. Yeah. How do you think we're going to integrate, in terms of what are your thoughts about how we get, investments in long term with the hope that we will hit whatever we're looking for in Africa? Yeah. And bear in mind, what you find in Nigeria might not work in Kenya, or what you find in Gabon might not work in Ghana. Yeah. So, do these things keep you awake in the night, or how do you manage?
Yeah, I don't sleep. I sleep maybe 4 hours a night. No, you're exactly right. You've hit on. You say what my worry is. You've described my worry. The worry is more. It's really difficult to attract investment to do this. I have experienced firsthand the skepticism. People raised $30 million with a back of the envelope idea in Silicon Valley. I have 5 years of traction, a proven pharma partnership, and they look at me like, they'll ask you questions like, what is your power situation? Right? As if I wouldn't have figured it out by now. Right? So the battle we have is a one of overcoming the world's failure of what Africa can do, because they underestimate us. Unfortunately, the money to do this, though there is some here, that money too is also suffering the same failure. When I started Yemaachi, I met nearly every high net worth individual in this country. I got 0 cedis. If I told them I wanted to build a block of flats, I'd have raised the money faster. So the African entrepreneurs are not yet supporting life science research. Aliko Dangote, all these people, we need them to begin to understand. But if they don't understand, how much more people in Silicon Valley? And believe me, I have had somebody send me an email saying, talk to Bill Gates, we don't do third world. Like, I'm not paraphrasing. That is what the email said to me. Who uses the word third world anymore? Right? But Silicon Valley does, because they have no visibility of Africa. So the challenge we have is, how do we know we are on the right path, how do we survive long enough to reap the benefits? And that is my challenge. How do I keep Yemaachi alive for us to reap the benefits of the path? I can see where we are. I can see Canaan. I can see the promised land. In fact, I can touch it. Right? But I don't want to be Moses. I'm a pastor's kid, so I don't want to be Moses. I want to be Joshua. I want to march in. Right? So I think that is the challenge we all have. But I think there we have to be pragmatic. The market here is small. So if you just say, I want to make a medicine for Ghana, then Ghana's population is too small. We need to think broader. We need to think Africa, right? And I was happy that the National Vaccine Institute Deputy CEO was talking about, trying to establish that African thing. But even Africa is too small. So I think the successes now are going to come where we tap into the self-interest of the West, right? Like I said, it's not fair, but it's the way the world works. There are diverse populations there. The US is 40% non-white and rapidly diversifying. They need more diverse data for them. And those people, many of them have health insurance, right? So the drugs will sell there. So the pitch to pharma for us right now is a bit of a Robin Hood style, right? That's what I meant by the intersection of what Africa needs and what pharma wants. Pharma does not care about Africa right now from a commercial perspective, right? Because they don't see money here. But they care about maintaining their positions in their big markets. US is the biggest, and then Europe. So how do we provide value there? And so if we can package our science, say, look, we provide value for you there. Indirectly, we are providing value here. And then gradually we look back and we say, look, people, come on, can we also begin to build our capacity? Can our governments begin to build infrastructure? Can investors locally begin to build in projects to focus here? But it's going to take some time. We are at the beginning. And so at this point, it's about finding that intersection. In time, we'll build for ourselves. But right now, we have to build for ourselves while building for those who really have the resources. Once the investment comes, I will then have more freedom to do. The pediatric project is not a commercial project, but I'm able to do it because I got grant money. The more I stay alive, the more I can do stuff like that, right? So I think we have to be pragmatic. If we go full, I want to solve trips, trypanosomes. Okay, do the trips, but where's your other side of your research? If you build a company to do trips, you will not raise money from pharma because they can't see where the value is. But can you do trip research while doing something else? Can you leverage your platforms across? So we're going to have to be innovative and ingenuitive, and I think we also have to collaborate. So one thing that we are thinking about now is how do we build an ecosystem? How does Yemaachi work with INCAS to support one another? Yemaachi and INCAS on our own, we struggle. But is there a way that we can see how we are in a value chain where we can stitch it together? Because like you said, genomics to translation is a very long road. If Yemaachi is to build the capacity, it will be 10 years before we have a drug. But there's a company called Bio-Usawa who are now starting monoclonal anti-production in Rwanda, and they're coming to Ghana. I began to talk to them because they have GMP monoclonal antibodies. If I find a target, maybe they can we can begin. So the pathway to clinical trial, if Yemaachi and Bio-Usawa partner, is shorter than either of us go on it our own. So I think we're going to have to be innovative. The opportunities are there, but it takes a lot of hard work, and you need guts. I mean, anybody who knows, they see me some days, they're like, hey, like my guys, they'll be like, hey, today, doc, this one day you look dark. It's like, yeah, I'm in the tunnel, right? I'm in the valley. So it's extremely difficult. I will not trivialize it. This is the hardest thing I have done, but I believe in it, and so I think we have to be smart, pragmatic, and strategic. And so, but then also those of you who have access to the Dangotes of this world, please point us out to them, because we will need Africans to support Africa. We have to do this for ourselves. So we have to survive till we can get them on board. But I think they will come. I think I wouldn't do it if I didn't believe it. But it doesn't mean, just because you believe it doesn't mean it's easy. So, but we'll get there. I mean, WACCBIP is a testament. I came to Ghana because of Prof. Wandari and what he had built at WACCBIP, period. I would probably not be in Ghana if it wasn't for that. The idea to build this in the biochemistry department, 10 years later, it's there, right? So if that can be done, then maybe we can do something else there. So I think we just have to keep pushing. Thank you very much.
All right. I think that's a rousing start to the day. It was always very aspirational and makes us to dream bigger. 2 announcements. I'm going to let our monitoring and evaluation officer come and make some appeals.
Good morning, everyone. My name is Felice Eugenia Mills, and I'm the Monitoring and Evaluation Officer for WACCBIP. First, car number GR435824. Your car window is down, so you can kindly go and turn it up. For after coffee break, we would like to reserve these seats for table talk. So if you are doing a table talk, kindly come to this side and you'll be directed accordingly as to where to sit. And also, if you've not submitted your table talk presentation, kindly do so to WACCBIP ICT. Lastly, kindly check your mails, and then you would see an email from WACCBIP Monitoring and Evaluation to score faculty to score orals, and then also all participants to score the poster presentations. Thank you.
Thank you, Felice. I just wanted her to make the appeal because she's been appealing to me and I'm appealing to you. So I think we will get along with the rest of the day with our first session. I'll invite the two chairs to take us forward, Dr. Henrietta Mensah-Brown and Dr. Dominic Amuzu, both of whom are alumni of WACCBIP and now part of our teaching and training faculty.
Hi, good. Is it working? Can you hear me? Okay, good morning, everybody. I'd like to welcome you to the morning session. We have two presentations. Just a minute. Which is the glycophorins color. Okay. So we'd like to invite the team vet for the fellow session, if they're ready. Come here. We'd like to invite Team Vet for the fellow session to present their broken crayon still color.
Hello, good morning. Good morning. My name is Sabina and I'm representing the vet team. We are here to share our journey. So, okay, so it all began with this call. I remember getting so excited when I saw this call. And that's because I love business. And for me, I thought marrying business with science was a great opportunity I didn't want to miss. So I immediately applied. And when I went to work, I shared it with a friend. Her name is Yvonne. And she also said she's seen it and applied for it. So together we began deliberating how the whole program was going to be like. Okay. Okay, so the program started, and I must say it was a great learning experience for us. We had a lot to learn from well-resourced people, and I must commend WACCBIP and the Business Hub for such a great initiative. So whilst on it, we were challenged to come up with an idea. And again, my friend and I, we decided to sit and brainstorm to see.
Next slide, previous slide. This is the next slide, and then point it to the crowd.
Sorry. So we brainstormed to see what we could bring onto the table. And we had two ideas. Initially, we had only wanted an idea so that we could be in the same team. But then we had two ideas, and we didn't know which of the ideas to drop. So we decided to split and champion both ideas to see how far we can come. She led the home lab team, and then I led the vet team. And we wanted to be in the competition and win. So right from day one, when I started my team, the vet team, we had winning in mind. We wanted to win. And so we worked for it. We made sure we've done everything possible to do. We dotted our I's, crossed our T's, and we wanted to win. And for me, I thought that day when we were going to pitch, we also brought on our A game. We tried our best. But unfortunately, not every day is for winning, some days are for learning. And so now reflecting on that day, we think that it was for a learning process. We didn't win the pitch, we lost. And we felt, my team felt disappointed and we were sad. And I also felt broken. I felt broken because I'm the team lead, and I felt that maybe my actions or inactions cost our team. So I remember when we left the stage last year and we went back into our seat, I quickly apologized to my team that I was sorry I let them down. And they were like, oh, but you tried your best. So why? But I felt they were trying to massage my pain or mitigate the whole issue. So until we closed, and then I realized that everybody came to me, like a lot of people came to me and said, oh, they were rooting for our team to win, but it's okay, you did your best. And it wasn't just a few people, a lot, students, faculty, and even the visiting participants. So at that point, I felt encouraged, and I forgave myself. So I must say thank you to everybody who came to me last year to encourage me on. Actually, you were the fuel I needed to propel me back into action. So after that, I decided to look for another opportunity. I told myself, if people think we have something good to offer, then let me look for another opportunity. And I think in about 4 days' time, after our broken heart, I saw this one. It was the NIP presidential pitch call. So I sent it to my team and told them, I've seen this, are they ready for it? And they said, yes, let's try it. So we applied and we were shortlisted. The WACCBIP's pitch competition was just between 6 teams, and we virtually knew everybody on board. But we went to the national stage and we had 49 other contenders. It was, it was really a fierce competition. And we had 3 different pitching rounds. The first round shortlisted us to the group of 30. The second round shortlisted us to the group of 20. And the final round, which would have shortlisted us to the group of 10, we couldn't qualify. And that's because at that level, we needed something extra more than a pitch. We needed a prototype. And unfortunately, it was just an idea at that time. So we couldn't make it to the top 10. However, we were number 11. And for that position, we received we received 50,000 Ghana cedis to develop our prototype. So we started working on our prototype, and somewhere this year I saw another call too. This time it was the Hollard Group, and this time it was the Hollard Group. It was the Hollard business streetwise business pitch competition, and this pitch is basically between the University of Ghana and then the University of Cape Coast. So each university brings on board 5 teams, and we compete to find a winner. And I must say, at the end of this year's competition, which ended just 4 weeks ago, we were adjudged the winners of the competition. And we walked home with 40,000 Ghana cedis. So in a space of, okay, no, let me just go to my next slide. So now we've registered our company, we've designed our 3D model, and we thought that we would need a lot of capital to start building the products from scratch here. So we thought it wise to partner with an existing company outside the country so that they will do the manufacturing for us until we find our feet and we go. So we've already signed an MOU. We are waiting for the products to come in about 2 months' time for us to do our validation studies, do our pilots, and then product registration, and also raise money for our initial first batch. So to summarize it all, last year, we lost $5,000. It was really heartbreaking for us. And within a space of 330 days, we've been able to raise $8,600. And that for us has taught us that broken crayons indeed color. It all depends on how you decide to handle it. Life is full of challenges, and science is difficult, I must say. Experiments fail, grant applications get rejected, and even publications sometimes become a daunting task. But in all this, I have come to the conclusion that we only fail if we refuse to rise when we fall. To our partners, we are grateful. Thank you, WACCBIP.
Right. We'll take any questions for Sabina.
Thank you very much for that resilience. I mean, very inspiring. So you really mentioned your journey with a lot of learning on the way, but I'm still left asking myself, what is it all about? You didn't mention the prototype, the product, so at least let us know what it was all about. Thank you very much.
Okay, so we've come up with an RD kit that's able to test for that's able to test for 5 pathogens at a time for vaginal infections. The last slide, please. Yeah, so that's what we are developing. We thought that the regular process of detecting pathogens is very intrusive. You have to go for someone else to be taking things from your personal properties, and we don't like that. So that's what we are working on.
Yeah, just a comment for you. When you started the presentation, I saw from pitch to pit.
From pitch to...
But I think you should add to the peak, because currently I think you are doing very well.
Thank you.
Do we have more questions?
Okay, thank you very much. I think that was inspiring. I would like to ask, so at the first time when you were not successful with the pitch, you had a team. How did you hold your team together after that? And then how did they choose to follow you on with the vision? Because usually you who have the idea is the one who has the vision ahead. How do you grab the others with you? Thank you.
Oh, thank you for your question. That's deep. Like I said, I was more affected by losing than my team members, because I felt that it was I caused it, and I was the one who pitched too. So for me, I felt I didn't do much. Actually, they encouraged me. And then, like I said, like after everything, the people who came around encouraging me on also helped me know that, well, I really did well. So we talked about it, laughed about it. We are friends. So it was a bit easy. And then when I saw the NEIP call, I really thought they wouldn't even buy into it, because it was just like 3 or 4 days after Broken Heart. But I'm really thankful to them. And I must say, they make leading easy. I just bring something on board and they are ready to rally around me. Thank you, team.
I think I also have a question for you. You've had a remarkable journey. Are you having any form of mentorship program or something that is guiding you? Because clearly Yao is doing a good job, and maybe you might learn some lessons so that you don't make certain mistakes here.
Yeah, we have quite a number of mentors. We are studying them from afar for now. And like I said, we were incubated at the hub. So anytime we have challenge, we just go there and they are ready to welcome us. They link us up with people who will help us. And they are always on us, trying to know our progress. So,
okay. It looks like Yao's talk did a good job. The next speaker is not online, so we are really back on time. The ancestors are actually happy with us. So they managed to probably switch the guy's internet off just so that we can be back on time. I don't know if my colleague has something else to add to it.
I don't have anything else to add, but it's coffee break now. So thank you all for joining this section.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentation, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meeting, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Now, going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway, I'm at the School of Hygiene and Tropical Medicine, and it's been my pleasure to be working with Gordon and with his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to enjoy the last 10 years having a good relationship between the London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that it is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I've not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
Grant from Carrowmore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. Yeah. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation the snake venom talk that has just happened was interesting because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, you know, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's Kaiser, South Africa. And our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center, and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa. And it's made me want to fight harder with the South African government to allow us to get visas for students, so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentation, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meal, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Going to MIT or to Rockefeller or to Stanford or whatever, you any of these elite institutions, you could find trainees of similar level that you find here?
I'm David Conway at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to be working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that it is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I've not been aware of before that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Caramore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's Kaiser, South Africa. And our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa. And it's made me want to fight harder with the South African government to allow us to get visas for students so that we can have joint supervision of students, so that we can have more cross-collaboration. And I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meeting, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Now, going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway, at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to be working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
Grant from Caramore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception. and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's GISA, South Africa. And our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center, and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa. And it's made me want to fight harder with the South African government to allow us to get visas for students, so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
All right. Somehow we're falling behind again. But you know the deal. If you know somebody outside, please text them, message them, ask them to come in. We're starting now. So I'd like to invite our chairs to take us through another session of host-pathogen interactions. Dr. Christel Chokba and Dr. Biola Isawumi will take us through that. And this will be 3 plenaries. James was affiliated with us, and followed by 2 postdoc presentations. So Dr. Chokba and Dr. Isawumi.
Hello everyone, welcome back for this next session after the very nice talks we have this morning.
All right, our first speaker for this section is going to be Dr. James Aboagye. Dr. James will be talking to us on the impact of latent tuberculosis on the expression of immune markers and inflammatory cytokines among people with HIV. So James, you are welcome. Please, a round of applause for him.
Thank you. Good morning. I'm James Aboagye, and I'm part of the HCRIS team, which Prof. Che talked about yesterday. And I'm presenting on one of the works that we are doing currently to understand, in terms of towards cure, persistence of HIV in individuals who are latently infected with TB. So a lot of data has been given in terms of the burden for HIV, which we know Africa actually holds the majority of burden, which is over 70%, and that is critical to us. However, in Ghana, we know that the prevalence is quite low, but still significant, and there's a lot to be done. In terms of HIV infection, within the acute phase where you have the infection, there is a high viral load, and then this persists for a while. Then as it goes on, the CD4 T cells are depleted. If there's no intervention, then the disease will definitely progress to AIDS and possibly to death. However, ART comes in and helps to reduce the burden, and so it lowers the viremia of the patient and improves upon the CD4 T cell counts of the patient. So these patients are able to now do well and probably even have a very high expectant life within this. And this is, however, in case there's any interruption, so when these viremia go below detection and there is any interruption, then the viral load will rebound. And there is a rebound because HIV actually establishes latency. And so that is the sleeper cells. I must say these are not terrorist sleeper cells, but it behaves in that way. And this is because the virus is able to integrate itself into these immune cells and then silence the cells from being active so as to express them for these cells to actually be killed. And they do. So these are various ways by which the virus does that. So it can do so through the resting T cells, replicative active cells, which will go on definitely, as they do, yes, definitely some of these CD4 T cells will die, but some also still go into the resting. And then you have like resident memory T cells, which can still also be infected and then quietly be in the latent mode. And all of these harbor the viruses. They do so through several mechanisms, of which notably is the epigenetic changes. So some of the things that are known is, for instance, when the virus integrates into the host, it changes the way that its DNA or the folding around, let's say, the nucleosomes, and how these are presented. The transcription factors are actually represented, and these affect the way that these viruses are actually transcribed. And for HIV, most of this transcription is actually initiated by the TAT protein. And so responses of binding factors that actually activate these transcription factors for TAT to actually go on are usually silenced. And also, upon entry as well, HIV is known to affect the metabolism of these cells, especially with metabolites that are produced by the cell, and hence also tends to put these cells into resting mode. So there is one of the hurdles is that because it's able to establish this resting T cells, it does so through almost majority of the organs or tissues within the cells. And so you have this repertoire of immune cells hiding or existing within all these tissues. So you can see almost that's from the central nervous system all the way to the genital sites in terms of reservoir. And for instance, with somewhere like the genital site, and somewhere within the genital tract, or they say the genital site, for instance, which acts as a primary, it is believed like this seeding of cells or seeding of the HIV in terms of reservoir is actually high, because this is like the primary site for infection before it even gets to the bloodstream. And as you can see, the diversity of immune cells varies, and even for those that in terms of, like, let's say memory, resident memory T cells, which are one of the most cells of which HIV actually utilize in terms of reservoir. There has been studies that have shown that the phenotype, for instance, that you find within like the genitals will be different from that of the blood, and so it makes targeting, like, it has to be like more specific in terms of when you are targeting. So there are several cure strategies that have been established to handle especially these quiet cells or sleeper cells. So, and one of it is the shock and kill, which is to activate these sleeping cells and then have them be inactive, replicate, and then you actually initiate a killing. So first you can do so, and then those that are being replicated and circulating, ART for instance, can clear them. But then the issue again will be with the cell itself. There are some that will, as I've shown in terms of active, they will still go and then still go back again into resting T cells. So then how are you able to address this? And you might, it means that you need mechanisms to actually initiate the killing of these cells so that you can actually eliminate them. So, and then we have the block and lock, which is inherently forbid the cell from being active. So you lock it into the cell and then you prevent it from being active. And then you have the immune therapies like CAR T cells, which are supposed to present this immune antigens of the virus, especially within cells that are latent, and then activate them for killing. You have the gene therapy like CRISPR-Cas9, which goes in and then cuts the integrated genome of the HIV and then activate it so that it can replicate and kill. So these are various strategies that have been deployed. And for our lab, we mainly use the shock and kill and then the block and lock approaches for now. So even though HIV persistence exists in terms of during the ART, in between or during periodics, there are what we call the viral blips. And these viral blips, so the virus is undetectable, but there's certain times where, or for once in a while, you see that the viral load comes up. It goes up and then it goes down. So these are situations where we call the viral blips, and these occur due to several reasons, of which some would be to ART adherence, where you are taking your ART, you stop it for a while, then definitely these virus will be reactivated and then come back. And then you have drug resistance, which could actually also contribute to it. And then you have stress of the immune cells, especially during inflammation states. And then infections, acute, which usually escalates immune stress and killing and all that, which could also introduce that and activate. And then you have the chronic. But the chronic ones are the ones that it's like they are also sleeping with the HIV in terms of co-infection. But as to what now they do and what their role is in terms of activating, let's say, HIV to come up, those are the questions or some of the things that we want to understand. So as I mentioned, we have a whole lot of organs in this, and both of them have specific, like you have, so this is not exhaustive, but I mean in terms of pathogen infection, you have several pathogens that will infect these tissues. And one of them, for instance, is the gut tissues, and which harbor the majority of HIV, what is a reservoir. So those are some of them. So all of these, it means like in terms of cure, for instance, if you are going to target, that is a whole hurdle. You have to understand what exactly in terms of the patients, in terms of infection, what is happening, what is there, and then which pathogen is there. And then even within these tissues, some studies have shown that, for instance, if the virus is in there and is being harbored in terms of reservoir, around the surrounding, it creates something like a barrier. So it actually activates like immune checkpoint markers, which prevents the surrounding, like the cell that the HIV itself is inhibited, is harboring in, to be activated and stuff. So those are some, so it's a hurdle to actually target all of it in terms of HIV persistence. So in order to understand that within our study, we have, HCRACE has set up a cohort, which is 390 patients, which have been followed over the past 5 years. And as part, we try to first go through their data record and see whether we can see these co-infections and stuff, but you don't get the data. So we then have to initiate and then screen these patients ourselves. So this is something that is ongoing, to screen several pathogens within this HIV patient. But at the moment, we've started with TB, hepatitis, and then syphilis. And as you can see, so from those, it's from the 390, we've had a high prevalence just based on these pathogens, over 30% of them having these infections, so which is quite high. And then we have several of them also having co-infections. And then for TB, especially, we realized like the terms of TB infection, this was associated with individuals who are age 60 and above, probably because they are living long, and then their immune system will also definitely go down and be compromised. And then also those who have actually been diagnosed with the virus for more than 10 years were also quite associated with TB infection. So then, as they say, so then the question was like, what was the role of these co-infections, especially in HIV persistence? So we know that definitely these opportunistic infections coming in, the main thing is like they help progress the disease. So for instance, TB, if TB should come and it's active, there is a full-blown, like it's so fast. And then for other infections and stuff, these, when they come, because if like the immune system is really compromised, there is. And there are several questions as to whether it is doing these through the transcriptional factors, those that I've mentioned, epigenetic, and then metabolism. It also induces apoptosis, which we know, and then modulates these immune responses, especially for chronic as well, where there is persistent activation, which is inflammation. This inflammation calls for the need like to understand what is actually happening in terms of these patients. And then also they have the ability to induce like immune checkpoint, that is to prevent the signaling of other immune points to actually target these. And then one other thing is we ask ourselves is whether they are able to activate in terms of clonal expansion, which is an inherently resting T cell, which is harboring the T cells, for instance. Can it and then when you have this infection, does it proliferate and then expand in terms of the size? So these are questions that, in terms of this study, we want to be able to answer. Does the antigens of like these co-infections actually stimulate the expansion of these seeding cells, these cells that are seeded in terms of the HIV pathogen? So those are things that we want to know. So for now, in terms of the studies, we've started with TB and HBV, but at the moment I'm presenting on the TB part. So for TB, which we do. So for instance, when one is infected with TB, the alveolar macrophage phagocytoses it, and then it stimulates the activation of several immune cells which come in to kind of help to contain and then prevent the pathogen from actually being full-blown. So if there is, for which include, and these several T cells, these several immune cells include CD4 T cells, and then they form this granular molecule, which, so you have like the TB molecule, TB antigens, pathogens within, which is surrounded by all of these, and then it goes into latency. So that is like if your immune system is good and is able to prevent, then there is the prevention of it going into active TB. But where like HIV comes in, where there is the killing of CD4 T cells, these do not contribute to the formation of the granuloma in terms of how it's supposed to protect you. And then you have active infection, of which usually will be the full-blown. But then because ART is there, it has helped these CD4 T cells to be restored. And hence, the question was like, yeah, when we looked into the data, we are not seeing so many of the patient in terms of active TB. So then the question was that because of this granuloma, has, even though it is preventing the antigen from actually being active and then replicating to cause the active infection, they still, even though to inactivate, they still elicit some small amount of inflammation. And this constant inflammation, in terms of what they elicit, is the question that we're asking. How does it impact HIV, especially on immune responses that could affect its progression and then its persistence? And again, for us, in terms of cure, we also want to know in terms of reactivation, how does it impact it? And so based on this, our study was to determine the effect of MTB on the infection and expression of immune markers and HIV persistence in people co-infected with latent TB. So when we did, we screened our patients for latent TB, which we identified about 37 of them, and then from there we matched those patients in terms of age and gender. By the matching, we did it like plus or minus 2, and then we tried. So the main matching was in terms of age and gender, but we tried as much as possible to also match very well in terms of like disease, like when they were diagnosed, years that they've been on ART, so that we get at least most of them being close. We didn't get it like 100%, but at least we tried as much as possible to get them there. So when we harvested their PBMCs, we did separate the CD4 T cells, and then we looked at the expression of cellular markers, immune cellular markers on them, so both activation and exhaustion markers. And we realized like there was no difference, so similar expression of both exhaustion and immune markers between them. But this is just looking at them. And then we went ahead and then we stimulated the CD4 T cells. And then when we stimulated, we wanted to see if, for instance, in terms of which of these, in terms of these groups, which of these CD4 T cells is more exhausted or is highly activated, then what is the expression of these markers in terms of them. So we did that, and we realized like even though after stimulation, still the patterns were similar. So those like CD69, for instance, was the highest that was stimulated, which is an activation marker. But we didn't see any difference in terms of the expression patterns between co-infected, HIV co-infected with TB and HIV mono-infected. So we then went on to do the reactivation to see if, like based on these the data that we have, and whether which of these groups would be difficult to actually reactivate. And we found out that from the study so these are like two individuals, two different paired individuals, And we found out that from them, for the HIV co-infected, it was difficult to actually reactivate the resting T cells from them. So then the question was like, what is happening? Could it be that really, even though we are not seeing that much difference in terms of the exhaustion patterns, in terms of exhaustion, for instance, within TB co-infected patients, could it be that something rather is also happening which we are not seeing? So we went on further to screen for various cytokines. We looked at a few, not a lot, but out of those that we also screened, so this was also by activation, like after we had actually activated the cell, stimulated the cells. So as we stimulate, because for healthy donors, we expect them to be high because then they are not primed to these infections and they are not exhausted. So we see very high elicit of these, or expression, secretion of these cytokines, inflammatory cytokines. However, between the two, which is HIV only and then HIV TB, we also saw something similar, no difference. So then the question was like, okay, so if this is what is happening, but this is like a cluster, so we are doing it in terms of we pick HIV TB patients and then we are comparing them with, and then it's just their sports, like right, what value we are getting, and that is what we're doing. So we wanted to rather look at, okay, if we do correlate the secretion, is there like, because we are seeing that there is reduction in terms of these inflammatory cytokines, when we do correlation in terms of the patterns, is there something different about these groups? So we just did correlate, with, especially for this one, we just did for the cytokines. And for instance, you can see, yes, though there was like you can see like slight correlation for the HIV TB co-infected, but I realized like this was higher within the HIV mono infected patients, suggesting like in terms of their secretion of such cytokines like, let's say, interferon alpha and interferon gamma, which are required for high inflammation. Actually, interferon alpha can play both roles in terms of either activating or inhibiting or resisting HIV from being reactivated. And so we saw that something like this is happening within these patients. So it means that HIV, even though like the expression seems similar, they are actually more correlated within the HIV mono infected than the HIV TB co-infected patients. So we went on and then we also wanted to see, how these correlate in terms of the cellular markers and then the interferon markers. So for instance, if you look at CD25 for HIV TB, you could see that it's either like almost no correlation, but and then also goes into the negative. But then if you pick the mono, you could see like most of them were actually heading towards the positive end of in terms of correlating. So this showed that the two definitely have divergent in terms of immune marker correlation. So even though we're seeing that in terms of pattern because we're clustering them, when you do check these correlations in terms of the immune markers, they look different. So we went on to look at in terms of seeding, which is the persistence of HIV in it, and likewise, we also didn't see any difference between the two groups, though we see something slightly for the HIV TB group for both the unspliced RNA and the proviral DNA. I mean, there's no difference, but we haven't finished with the work, so we hope that by the time that we are done in terms of estimating these reservoir sizes within both groups, we might actually see something to report on. But at least if even like there's one-fold change between the groups, it is quite significant. So as mentioned, we are saying that there is similar pattern in terms of cellular secretion and cytokines between these two groups. However, in terms of correlation, there is divergent correlation in terms of the immune markers between the group, which definitely is contributing to the low latency reversal potential in HIV co-infected patients. And then, therefore, so as we've already been reiterating, like, there is the need that when you have co-infected people in terms of cure, they suggest that there should be targeted strategies for those that are actually co-infected. And so these are various things that we hope to do. Look at patterns, especially for HIV, especially because some study or one work like we are doing with a collaborator who says, like, for instance, who made us aware, for instance, when you look at infection prior or before in terms of whether HIV or before another, like TB, the immune patterns are different, and this could contribute to some. And so we may have to pick, like, patients, identify in terms of infection which one is before and which one is after, and all that. Do like antigen-specific T cells and see what the effect of is in terms of proliferation and all that. Look at what we did was we just measured the reservoir. We didn't look at like the defective against intact, and then in terms of which ones is replicative competent, we are also going to do that in terms of it. And then we also have to look at other pathogens. I would like to acknowledge the HCRIS team and all the study sites and collaborating sites as well. And then for the good work done, and then what is going on. I mean, it hasn't been easy for the past 5 years, but they've been actually helping us and assisting us and going. So we acknowledge every person, especially the hospitals. We appreciate them and the patients. So thank you.
Thank you so much, Dr. Abraji, for the beautiful presentation. So can I have about 2 questions because of our time? Okay, so let's start with Dr. Kwashi, then we'll come.
Thank you, James. I'm just wondering, do you think it's the not seeing such a strong difference between them is because you're looking at latent instead of active TB? Could that be the...
Yes, actually, that is true. So studies, though they've not done it like with it, but they've shown that if you do like just HIV and TB in terms of like there is high, this in terms of these cytokines, like way above. And so that's why if I mentioned that for active TB, definitely when it's identified, you'll be treated and all that. But what we wanted to know was like if there's the latency, and especially where there is still persistent inflammation consistently happening with the patient, what happens? So for active, if this is definitely seen, but during that there's a really high boost which could actually, and then for cytokines like IL-6, IL-1B, those are known to actually impact on the reservoir. So those are kind of, but we wanted the latency portion, so that could actually contribute to it. But we may also have to find what exactly, but at least the reactivation portion says a lot. So we need to still find out what is really happening, and then, yeah.
All right, thank you, yeah.
Thank you very much for that excellent presentation. That also stemmed from what your colleague presented yesterday.
Yeah.
So, a couple of questions. I mean, you define latency without acknowledging a paradigm shift in this group of people. I mean, what we knew previously as latent TB is really a very divergent group because you now have subclinical TB,
yes,
incipient TB. So latency is quite a very, very divergent group, and I think some of your data really can be pointing out to that. So it's important to first clarify how do you define that latency, and what are the different assays that you use. The other question is, for those that have HIV, normally when you diagnose in quote latent TB, they need to take prophylaxis. Were a patient taking prophylaxis? You didn't mention anything about that. And because they were taking prophylaxis, that will impact the way that reservoir will be in their patient. So I mean, I think there's a lot of layers that you are not unpacking, because I mean, e.g. this last graph that you show, you see that there's a huge heterogeneity. I think there's 3 groups there. The top one, the middle one, and the bottom one. And I think if you just unpack these diagnostics, you might actually be talking about different people and then see the differences that you are looking for. I will just stop there. We can follow up.
No, I agree with you. I mean, in terms of looking at the different layers, I mean, for TB infections and how they could actually impact on this reservoir. So those are things that we actually want to look at. So, yeah.
All right, thank you. The last question here. Yeah.
Unless I didn't.
So the second one is... okay, so the PBMCs that you take from the patients, you said you seed them, and what do you stimulate them with?
Also, we stimulated them with PMAI.
Oh, the PMA, just as yesterday's talk, where it's overstimulated or something like that.
So it can be overstimulated, too strong. And then, so that is why also one of the things we want to do is, apart from like the PMA, we also want to do like the specific TB antigens.
And also take into consideration cytokine profiles change with time.
Yeah.
I don't know if you have taken that into consideration.
So we'll be doing those like the progressive this, and then as I mentioned, we want to see like the change in decay, like how in terms of seeding. So if those are going to change over time, we need to actually identify them within periods. So it is part of the work to do. Yeah. Thank you.
All right. Once again, thank you, Dr. Abaji, for that excellent presentation.
Yes, thank you, Dr. Abaji. Now we are inviting Dr. Bolahan. Dr. Bolahan, one WACCBIP postdoc based in Nigeria, to share his findings.
Good afternoon, everyone. Okay. So my name is Bolan Odushelu from Nigeria. I'm a postdoctoral fellow at WACCBIP, currently working on a project along antimalarial drug design. And this part of our project that's ongoing, titled De Novo Molecular Design and Bioactivity Prediction of Novel Hexahydroquinolines as Plasmodium falciparum calcium dependent protein kinase 4 inhibitors. So, as background, Medicines for Malaria Ventures, they've provided guidelines in the form of target product profiles and also target candidate profiles, and which can be divided into the TCP1, TCP2, TCP3, the TCP4, 5, and 6, targeting different stages of the Plasmodium life cycle. And the one of interest to us in this particular study is the TCP5, which targets the gametocyte stage. And existing antimalarial drugs are highly stage specific, and with majority actually targeting the blood stage parasites. So there are needs for compounds that also block disease transmission. Okay, so for disease transmission, the target TCP5 targets, we're looking at Plasmodium falciparum calcium dependent proteins kinase 4, and they are key mediators of calcium ion signaling in the malaria parasites, and they're also absent from the human kinome, and their divergence from the human homologous actually suggest high selectivity and also lower toxicity risk. And that's why inhibiting this particular enzyme helps in disrupting exflagellation of male gametocytes in terms of the microgametes, and also impairs sexual stage development in the mosquito. Okay, so the question is, are there known PFCBK4 inhibitors? Yes, there are known inhibitors. However, most of the known inhibitors have not been able to transit into clinical trials, majorly because of poor oral bioavailability and also because of toxicity issues. The major class that was first identified was the bound kinase inhibitors reported early as like 2012, and which has the pyrrolopyrimidine core as the major heterocyclic core in the structure. And yes, it was able to inhibit PFCBK4. However, based on toxicity concerns, they modified this particular structure by adding a methyl group at the piperidine ring in order to improve this particular compound. And by reason of that, the compound is PKI 1294, and it's highly selective. Just that is also associated with the egg liability, which is a major issue. So, other efforts have also been done in this area, but the challenge still remains the same, how that remains poor oral bioavailability and also toxicity. Okay, the advent of the 3D crystal structure of this particular enzyme has enabled inhibitor design using structure-based drug design. And what does that mean? It means that we're able to use the 3D structure in order to be able to design and also to optimize potential drug candidates. And that's the 3D structure as gotten from Protein Data Bank. Some researchers in 2016 explored structural requirements for the inhibition of this particular enzyme using multiple in silico approaches. And their key findings was that there are certain pharmacophoric features, we call them functional groups or moieties, that are present that are needed for inhibition of this particular enzyme. And that includes 2 hydrogen bond acceptors, 1 hydrogen bond donor, and 1 hydrophobic group. Also, a report in 2017 where they screened Novartis GNF malaria box of like 3,825 essential blood stage active compounds on PF gametocyte stages, and they described the identification and the antimalaria properties of this particular group of compounds. And these are the group of compounds we'll be looking at in this particular study. And they're quite different from the the bomb kinase inhibitors in terms of the heterocyclic core that is present in the structures. Also, the advent of AI and machine learning in drug discovery has also helped in order to improve how we're able to design compounds for drug discovery. And this particular one is Alva Builder in 2024, just last year. And they were able to develop a software for de novo molecular design. What is this about? Meaning you're computationally generating entirely new chemical structures from scratch rather than selecting from existing compound libraries. Why is this important? It's important because the possible chemical space we can have is like 10 raised to power 60. So we need to explore more chemical spaces that for us to be able to have more options in terms of selection of compounds that can act as antimalaria drug candidates. So with that, the aim of the work is to design novel antimalaria hydroquinolines and derivatives for this particular presentation, which is a part of the ongoing work, with improved predicted bioavailability and also reduced toxicity compared to the BKI1 and also the BKI1294, which I mentioned, are the known inhibitors of this particular enzyme. So in doing that, we started using the Alva Builder, and with this, what we did was to generate like 20,000 potential antimalaria hexahydroquinolines using this particular AI software, using the training set of MMV data sets. So MMV has like 18,925 hits that have been reported as good antimalaria compounds. So we use that as the training set, And also, we set different rules in terms of for drug likeness, majorly. And also, we use the training scaffold as the hexahydroquinoline that I mentioned earlier in order to generate these compounds. So after generating these compounds, we did the machine learning model. We built a machine learning model using the ChemProp. So ChemProp, it creates a classification model for predicting molecular activity. And that was developed also in 2024. And we used 2,000 compounds that were derived from malaria flow, 1,000 active, 1,000 inactive, in order to train the model. And we divided it into the training set, the test set, and the validation set in ratio 7 to 1.5 to 1.5. And after doing that, we were able to, so we validated the model, and basically the receiver's operating characteristic area under curve showed that for the training set, even the test set and the validation set were greater than 0.84. And what does that mean? It means that we have a strong and a generalizable model that we can use to apply. We can apply that on generated analogs from the alpha builder. And that's what we did. We applied that on the 20,000. I mentioned 20,000, but in the place of preparation, it reduced to 19,955. And we're able to get out of the 20,000, or out of the 19,995, 17,808 that were active, and 2,187 that were inactive based on this model. And we went ahead to filter using what we call the pan-assay interference compounds, and we call them PAINs, and they are majorly small molecules or substructures that have tendency of generating false positives. So we don't want that. And we're able to identify certain PAINs or small structures like the thiol, the nitro group, that were present on some of those structures. And we're able to classify using that as the ones that are PAIN structures and the ones that are non-PAIN structures. Because what we are interested in are the ones that are non-PAIN compounds. So we went ahead with that. Okay. Let me go back. Yes. We did oral bioavailability. I mentioned that during the problem statement that one of the issues is the issue of oral bioavailability. So we wanted to check can we get a model that can help us to predict the oral bioavailability? And we're able to get that from a Celia model. And we're able to apply that on our compounds that will be filtered from the PAINs. And that reduced the compounds from 10,525 to 9,160. Using a cutoff, a threshold of 50% for the bioavailability. Now, from here, we were able to do the virtual screening using pharmacophore model. What is this about? We used this particular model, which was developed by Ayer and Roy in 2016. And I think I mentioned that during the introduction. And they were able to identify pharmacophoric features like the HBD is the hydrogen bond donor, and the hydrogen bond acceptor. So they were able to identify 2 hydrogen bond acceptors that are very important for inhibition, 1 hydrogen bond donor and 1 hydrophobic. And we applied that on the 9,160 compounds that we have. Interestingly, so this is what is happening here. These are the pharmacophoric points for the model, and we ran the 9,160 through this particular model, and only 67 out of the 9,160 compounds were able to pass this particular model or go through this particular model, showing and meeting criteria for the potential PFCPK for inhibition. Okay. Next, we took the 67 compounds forward for what we call the structure-based virtual screening. In this case, we docked the 67 compounds against the binding site of PFCPK4. And doing that, we were able to generate what we call the docking scores. And we also correlated the docking scores with the fitness scores that we obtained from the pharmacophore model in order to be able to select our compounds or best hits to take forward. And we combined that also with 15 key parameters that are established as cutoff for initial drug metabolism and pharmacokinetics, which is the DMPK prediction. And at the end of the day, we're able to come up with 3 compounds which were able to pass this, and also comparing it with the BKI in 1, we're able to see that 2 of the compounds, HHQ01, HHQ02, had better docking scores. However, HHQ03 still lower docking scores, but was able to pass for the 15 key parameters. So we went ahead to do what we call the biassistar replacements, just to see, is it possible checking, let's change some chemical groups. We did that using Mestro, which is Codinja. Can we change certain molecular, small structures on these without significantly changing the docking score? And that's what we did. And interestingly, we're able to see that on HHQ01, that had a benzene ring as one of the structures, just changing the benzene ring to a pyrazine, we're able to get a docking score, not slight, but I think our major focus is the HHQ03, which was lower than the one for the standard. And interestingly, we're able to get one by changing just the pyridazinone to a pyridinone. And interestingly, we're able to get from initial minus 7.117 to minus 8.792. I think finally on this, so we're curious about the ADMET properties, which is the last phase of this particular one that I'll be presenting. And we have some interesting results here. For the HHQ, the ones that we did by assistar replacement, we're able to see that comparing that with the standard BKI1 and also the BKI1294. I earlier mentioned that major issue we had with this particular enzyme inhibition is poor oral bioavailability and also toxicity in terms of the egg. And so we tried to do this. These are the 15 parameters that I mentioned earlier that we used. And for the oral bioavailability, the F 30%, which represents the predicted probability of oral bioavailability ranging from 0 to 1, where the lower values indicate a higher likelihood of it being orally bioavailable. And when we compared that, we're able to see that what we're looking for is closer to 0. And we're able to see that we had some relatively new compounds, because these are novel compounds, not in any database. We're able to see relatively new compounds which had fairly good oral bioavailability compared to the standards. And more interestingly, is for the egg liability. And in this case, for the standard which I mentioned from literature that had egg liability, we're able to confirm that with this particular process, that actually those compounds actually do have egg liability based on prediction. And that also gives us some confidence with respect to this result, because we're able to compare that with what we generated through the AI machine learning process and the structure-based drug design. And we're able to see compounds, new novel compounds that are closer to 0, because egg represents the predicted probability of egg inhibition ranging from 0 to 1, and what we're expecting is something closer to 0. And we're able to see some of the new compounds doing fairly better, not fairly better, doing better than the inhibitors that we have initially. And so this is the first phase of the work, and in short, we've been able to get novel hexahydroquinoline derivatives with improved predicted bioavailability, and toxicity profiles have been designed. Also, the importance of and the role of AI and machine learning integrated with structure-based drug discovery has been established and confirmed also by reason of this study. And what are we looking at next? We're trying to optimize, and that's the stage we are at the moment, working closely with an organic chemistry lab in Nigeria, where we are trying to optimize the hits to check them for synthetic accessibility. And also further, after doing that, we'll do the synthesis and characterization. Then we also partner with some of our collaborators to do the experimental biological screening of these compounds and also the pharmacokinetic profiling. At this point, I would like to appreciate my advisors, Professor Wellington Oyinbo at the University of Lagos, also Dr. Lucas in Ghana, then Dr. David Conway, and also Professor Gordon, and also the founders, Host Lab, Covenant University, and some of our collaborators that have been working together, Professor Sampat in India, for helping us also along the line of the synthesis. Thank you so much.
Yeah. Thank you for the nice talk. Now we are taking a few questions from the audience. Where's the mic, please?
Please, can we have the mic? Nice.
Yeah, I'm curious to know. I want to know the biosteer replacement you did.
Yes.
Was it classical or non-classical biosteer replacement?
It's classical.
Okay.
Yes.
Thank you.
Thank you.
Thank you.
Yeah, nice talk.
Thank you.
I was just wondering, when you were looking at the oral availability.
Yes, please.
I wasn't too sure whether you looked at other ways of administration, whether you examined that also.
Yeah.
I only saw the oral bioavailability.
Yes.
So, were there any comparisons with, say, intravenous, if you were intradermal, and all that? And also, added to that, what about the kinetic as they move through the system? To what extent they are metabolized?
Okay.
Did you look at all these?
Yes.
Yeah.
Can I answer?
So, yes.
Okay. So the first one, thank you so much. I appreciate that. With respect to checking for other means aside from oral, now at this stage, the level we are at is still at the level of computational work, and that's based on the data we have available. So we may not be able to predict for intravenous because we don't have the data yet. So that's that for that. So we can only predict, but it's one of the things we're considering at the level of the experimental biological screening. So for the second one that you mentioned, with respect to, sorry again, I missed that.
Kinetics.
Okay, the kinetics, yes, pharmacokinetics. Pharmacokinetics majorly is the ADMET, the absorption, distribution, metabolism, excretion, and toxicity. Those have been considered in terms of the key parameters, which are the 15 key parameters that I mentioned. So that's at the level of prediction. So that's it. We've put all those in consideration at the moment. Yeah. Thank you.
All right. Thank you, Dr. Gulayan, for the excellent talk. Because of our time. Our final speaker for this section is our brother and friend.
Please, there's a question online.
Yes. Okay, please.
I have two questions. The first one is, can the software Alpha Builder allow live design options where scientists can work on the same compound simultaneously, just like Schrödinger? Second one, okay, you answer the first one then.
Let me answer that. Okay, so Alpha Builder, just like I said, it works more like generative AI. So you can subscribe to it. Yes, we have a subscription for that, and so with that, that's what we're able to use. So if possible, but it doesn't work like Schrödinger. It works more of generative. So for Schrödinger is not generative. So there are two different things.
Okay. The second one is, how can you only consider, it's not, the English is not Englishing, but how can you only consider, is it pains only?
Okay.
What about Lipinski's and
Lipinski's
plenty
rule.
other things?
Yeah.
Finally, what was your enrichment factor for your model based on ability to pick activities against decoys?
I missed the last question.
What was your enrichment factor for your model based on ability to pick actives against decoys?
Okay. All right. So for the first one, in terms of paint, did we consider Lipinski's rule of 5? Yes. And that was mentioned at when I was talking about the rules that were set for Alva Builder. I mentioned certain rules that were set. So Lipinski's rule of 5 was taken into consideration at the point of setting the rules for the generation of the compounds. While for the second question, in terms of enrichment, that's why we employed this particular model that I mentioned, which is the ChemProp. ChemProp is specifically for chemical compounds, so it's not just like any other machine learning model or package. That has been put in factor into consideration based on using that particular package specifically for chemical structure generation. Thank you.
All right. Thank you. Please let's welcome Dr. Amusu for his talk. Doc, you have 12 minutes. Thank you.
Is it afternoon or still morning? Good day, everyone. It's always a great joy to be at WACCBIP Research Conference where we dissect science and not football. Anyway, my talk today is about genetic diversity in the human glycophorins and their relationship with SARS-CoV-2 infections and their burden. And for those who are here for the first time, I just want to remind you that WACCBIP is 10 years plus 1. Thank you. Now, the main talk. Glycophorins are a group of sialic acid residues, proteins that are found on membranes of cells. And they work with other proteins in the form of a complex where their role is to actually keep the membrane integrity accurate or in shape, and also maintain their flexibility as they move through cells or as they interact with each other, especially erythrocytes that have to transport oxygen to various parts of the body. They are heavily glycosylated, and when we say they are glycosylated, it means they have a lot of sugars on them, and this contributes to a negative charge on the surface of these cells, and that enables them to interact with other cells and also different pathogens. Now, there are 2 different gene locus that you can find these glycophorins on. The first one is on chromosome 4 in that region, and over there we have 3 genes: glycophorin E, glycophorin B, and glycophorin A. As at now, glycophorin E is not really expressed. What we have is just the B and then the A. And then we also have glycophorin C, which is very similar to the glycophorin D on chromosome 2. There are a lot of diversity in this gene locus or gene cluster, and this is how they look like when you put them on the surface of erythrocytes with their different amino acids and binding site for both extracellular and intracellular domains. Now, the residues that they have enables them to interact with different pathogens. Protozoans, we have bacteria, as well as certain viruses. And Leffler et al. Were able to show that structural variant in this gene cluster reduces severe malaria by about 40%, and they used 1,000 genome data set for this analysis where they classified these structural variants. Now, on top over here, what we see as yellow is a complete deletion of this. So this is the gene cluster, and I must say that the sequence homology is about 97%, and it's been hypothesized that glycophorin B evolved from A and also E evolved from B in a series of events. So over here we have what we call the deletions. They've been arranged in terms of their frequency, whereby deletion 1 is the most frequent in the population, and deletion 8 is the least, and the list goes on. There are other deletions that have not been shown over here. And then we also have duplication. Duplications mean the DNA sequences over here have been doubled. So this is DUP1, which is the most frequent, and the color coding is just a way of explaining how many sequences have been duplicated in that region. And as I said earlier on, this was done using the 1,000 genome data set. After Leffler did this work, Sylvia Kariuki took it up to find out the mechanism under which this protection is happening, because it's really high, about 40%. And this was found in individuals that are in East Africa, and this variant was known as the DAN2. After showing that the surface tension or the tension of the cell membranes is what was causing these malaria parasites not to have the ability to grow properly or invade these RBCs, and therefore individuals can actually have malaria infection, but they never get severe malaria. That means they could just be harboring the parasite. She also went further to show how this DAN2 actually affects parasite growth. But more interestingly, a recent paper from her also showed that these glycophorins, the DAN2 variant, actually is able to reduce bacteremia, or a series of different bacteria in individuals who have this variant. And as you can see over here, there are several of these microbes that were used in this research. So now, coming to viruses, there have been a lot of papers that have shown that these glycophorins also interact with viruses. And as someone who got introduced to glycophorins for my PhD and also got introduced to sequencing, I tried to find out if there could be any correlation going on. So in the literature, there's a lot of work that has been done. And some of them, or the most interesting ones, are showing that even the SARS-CoV-2 actually affect erythrocytes at the precursor erythrocyte level, and also in the bone marrow. And these other papers have shown that this infection actually leads to the damage of erythrocytes, and this could actually be the reason why some of the severe COVID cases that were observed were in terms of respiratory distress, because you need oxygen to circulate, you need RBC to circulate the oxygen. So if you're having anemia, ischemia, and the rest, then it's going to be difficult for this patient to recover quickly. And also there could be multiple organ failures. And this work, the first work, was supported by other series of works where they now went further to show how these infected RBCs could actually lead to dysregulation of hemoglobin and also ion metabolism. And interestingly, this paper, glycophorins actually interact with other proteins, and one of their favorite partners is the BAN3 in the cell membrane. And this paper also showed that the SARS-CoV-2 actually interacts with some of these BAN3 proteins. So going forward, in our own, will I say, WACCBIP here, there was a paper that showed that immune tolerance to COVID could be due to malaria, exposure to malaria from P. falciparum. And again, literature is also showing that some of these glycophorins are actually expressed on leukocytes, or should I say platelets, and are involved in the movement of these platelets during immune responses. And this is the paper that was done by WACCBIP showing it. And also other human genetic studies have been done, trying to link the role that malaria is playing in COVID response. So from most of the genetic papers that were done with respect to COVID severity, they found out that it could be due to, some people could be having susceptibility in terms of the H2 receptor. It could also be cytokine regulations in terms of immune response, and also disease progression and severity. And there were a couple of papers, and also some reviews have been done where certain genes have been identified to be potential candidates. But more interestingly was this studies that was done that showed that chromosome 3, the short arm of chromosome 3 region 21 was strongly associated with respiratory distress, as well as our usual H2 receptor, and also polymorphisms in these other 2 genes. And then also we know that ABO has been already linked to blood grouping. Now in their paper, which was published in New England, what they did was, it was a GWAS where they picked participants from Italy and Spain, and these were the number of cases that were used, and these were the number of controls that were used. And the outcome of it was these genes that they actually found. However, most of the literature that is online linking most of these GWAS studies or the human genetics to COVID severity or infection, most of them did not focus on structural variant. They focus more on the SNPs. And so the question then is, glycophorins have serious structural variant, a lot of them, which I've already shown you. And also I've also shown you there are a lot of interaction with some of these viruses with these sialic acid residues on those receptors. So the research question was, would the distribution of this copy number variant actually have any influence on SARS-CoV-2 infection, the variants that are circulating, as well as the burden of COVID? And I think that explains the aim for the work. These were the main objectives. In summary, we were supposed to figure out if the glycophorin EBA gene cluster region, the diversity in it, could actually be linked to COVID infections, the different variants that were circulating, especially the variants of concern, as well as the COVID severity that was observed in other parts of the world. And so for the research design, it was an observational case study work that focused on, and also some cohort genomic association work. We pick most of our data sets from online and we group them according to different regions. And also, I was fortunate to have very good mentors. So WACCBIP and Professor Wandari and my PhD supervisors gave me the opportunity to genotype some of these glycophorins already. So I picked it up also for my postdoc, where I genotyped extra. So then we went online to look for other different data sets, which I will explain briefly. So for the first part, which is the genomic part, we took online data sets, uploaded them onto the HPC, and the data sets we got were what has already been sequenced from malaria gen, as well as the 1000 Genome Project. We also looked for other information regarding this copy number variant that were already existing online. And on the HPC, remember I said earlier on that Leffler has developed a pipeline to be able to pick this copy number variant within population using the 1000 Genome Project. So we adapted that in WACCBIP here, and the student that was really helping or supporting with it was Stephen Addo. He did a very good job. So we currently have a similar pipeline like what Leffler has. And then based on the data sets that we're having, we took it through bioinformatic sequencing and then got a copy number variants using the Hayden-Markovnikov model. And so having gotten this, we now added it to the original PCR genotype copy number variants that we're having, which is the DEL1 and then DEL2. And as you can see over here, I would like to thank Professor Wandari for being able to get me involved with this kind of work, because I think this is how you build young scientists to grow. So for the map, this is showing the different data sets that were used. And we decided to exclude North America because a lot of you all agree with me, America is very diverse. So it will be difficult.
3 minutes more.
I was told it's 20 minutes because it's a plenary talk. Yes.
Okay.
Yeah. Thank you. Yeah. So we filtered out North America because it's a little bit more diverse, and this is the distribution of the different data sets that we used. For the COVID data and then the SARS-CoV-2 infections, again, we went online. We took most of the variant strains from NextStrain and GSAID, and also information from CDC, WHO, and then our world in data. And putting it all together, we were able to come up with this approach where we try to link the different variants that were circulating with the different copy number variant as well as the COVID morbidity. So for the objective 1, this is the result. This is how the result looks like. Over here, you can see more of the, this is a combination of the various copy numbers. So on top are the deletions, and then we have the duplications stacked on top of each other. So you can see clearly East Africa and West Africa had a higher diversity, which is similar to what Leffler reported, putting all the data together. And these structural variants are very low in other regions across the world. And so when we compare these deletions, so now we stacked the deletions and the duplications together. On top here are the deletions stacked and the duplications stacked across the various regions. And then when you compare it with the COVID infections, you can see clearly we had lower infections in this region compared to Europe and the rest. And this kind of marries the original story that was proposed by Professor Wandari, Dr. Kwashi, and Dr. Yabiga Kushpepa. This is a spread view of how the deletions look like. And again, you can see this is where across the world, this is where the gene clusters are. And again, it's kind of linking it with the low COVID cases. So in summary, you can see clearly that these gene clusters have a certain effect. This is not to say that this is the only reason why there was low COVID cases in Africa, but this is just to show that clearly there is some form of contribution from this gene cluster towards the infections that were observed. So for the objective 2, this is how the variants came. The blue ones are the variants of interest, and the red ones are the variants of concern. And the goal was to see if there was any association with this copy number variant with the SARS-CoV-2 variant strains. And so this is the world map. This is where this is where we are having a high diversity in the gene cluster for the glycophorins. And this is where the first, should I say, the reference strain started from. And you can see clearly as the different variants came along, none of them originated from this hotspot for the copy number variant in the glycophorin gene region. And we tried to see if these variants, the peak of them coincided with Africa, which is over here. So this dotted line is for Africa. So we tried to link it with the results that we were getting from the copy number variant. And you can see clearly almost all of them were somehow identical except for the beta. Now the beta originated from South Africa, so it's likely this is what caused more spread within the population, the African population. Now this data also includes information from South Africa. What we are trying to do is try to decouple this information and see how, because from our result, there is none of these deletions within the South African region, which is Natal. I think this has been summarized already for lack of time, so I would like to move on. But it suggests that there's a strong selective factor that is ongoing since none of the variants were coming from any of these copy number, high copy number region. So for the objective 3, we are just similar to the objective 1, which was to marry these deletions with the COVID deaths. And as you can see over here, there was a similar pattern. And the same thing was kind of observed. So taking all this together, clearly some of the glycophorins, the gene cluster, the high diversity in this gene cluster could be leading to the number of lower cases that we were observing in the Western, Eastern, Central Africa. And it's probably due to the deletions, because the deletions were more higher. And as I said, when you delete this sialic acid reduce, maybe it prevents the virus from interacting very well with this erythrocyte. So in summary, we've shown that the high diversity of the glycophorin gene cluster in both chromosome 2 and 4 is actually probably having an effect on the COVID severity in terms of the cases, the variants, as well as the deaths that were observed. This is the first presentation on this association, and understanding this could actually lead to how we could probably treat some of the severe cases. And ongoing, we are still analyzing more data and then come up with more robust logistic regression models to be able to determine the degree to which this gene cluster is influencing the COVID situation that was observed. So for me, this is a joy because I have supervisors who have trained me for my PhD, and they have really done very well in mentoring me to the point that I've been able to work on glycophorins and also got the opportunity to work on the COVID. And gradually, I think I'm maturing by trying to link these two together and working on it. And I also like to thank my other two supervisors and also Leffler for being able to contribute to the development of the pipeline and getting this work done. And also for the two research assistants in WACCBIP who have done a tremendous job to get this done. And Collins, Dr. Kwashi for all the support, all the faculty, and all those who have been curating data either in terms of human genetics or COVID information online.
Okay, thank you, Dominic. Thank you. Yeah. Now we'll take only 2 questions because we are almost out of time.
So Dominic, this was quite interesting. I know you. I'm just curious if when you maybe using mortality data, you know, because the case data, it's we weren't detecting as much, so that might be hard to do it, and you'll probably even find stronger links when you use the mortality data.
Okay, thank you.
Because that's easier to track than cases.
Okay, thank you. So just to react to that, what we tried to do, initially we were using only WHO data, but we started seeing a few inconsistencies. So what we did was we put together data from the CDC, WHO, I think there was Johns Hopkins also, but a few, and then our data in the world, and we put it all together to sum the average, and that's what we use for the analysis. Yeah. Thank you.
Thank you very much, Dominic, for a very, very nice work. I think what you mentioned, you present is mainly association. Have you tried to design assay, taking sample from people with this deletion to do in the lab and see really if they attach or not? I think that will kind of validate, the excellent association that you are seeing.
Yeah.
Association is not causality.
Yeah, thank you. It's part of, I think I showed it, part of ongoing studies to get more data. And then secondly is to identify phenotypes who are having these deletions, take the RBCs and other cells and run some assays to see if it's actually affecting the growth or infectivity of the virus. But thank you for the, yeah.
Online question. Yeah, an online question. Yeah. Okay. Thank you.
Thank you.
All right, thank you. So we'll go for another quick session of vaccines and biotherapeutics, which will be followed up by another Turbo Talk session. I'd like to call the chairs Dr. Irene Owusu-Donko and Dr. Abdurrahman Kamara to take us through this.
So good afternoon. I know you've been going through a series of talks, but just know that this is the most interesting session of the conference because it's about vaccines and biotherapeutics. So I think we only have a plenary talk, and then we'll hand over to Dr. Abiola for the poster, the Turbo Talk. So our presenter is Dr. Fadio Bidmos, if I got it right. Yeah. So Fadio will be presenting. So from infection to innovation, cross-reactive human antibodies uncover next generation vaccines. It sounds very exciting. So you have floor for 15 minutes, followed by 5 minutes of question.
Okay, thank you very much. And I do agree, actually, that this will be the most exciting part of the whole conference. As infectious diseases researchers, I think it's very obvious now to the majority of the audience and all of us that these pathogens have a fantastic propensity to evade immune pressure, and so that, in a nutshell, is the rationale for continuous vaccine antigen discovery. Now, my lab at Imperial actually works across all of the different stages in the preclinical stage. So this is design, discovery, and of course delivery. But for today, in the next 15 minutes or so, what I will try to do is just show you what we do in terms of discovery. Now, as exemplar, the approach that we've used, which is known as reverse vaccinology 2.0, we've initially applied to these two pathogens, which cause pretty much the same disease, bacterial meningitis. They're the two chief causes. Neisseria meningitidis being a gram-negative bacterium, and strep pneumo being a gram-positive. And despite the availability of antibiotics, we have about 15% mortality. And this is mostly because the disease, the early symptoms of the disease are fever, tiredness, headache. And as the majority of us know, if you present to the GP surgery with these early symptoms, you're more likely to be given paracetamol and sent back home. But of course, as the disease progresses within about the next 16 to 18 hours post-infection, of course, the more specific symptoms begin to unravel. And these include, of course, the rash that we see, especially among Caucasians, but of course, tiredness and photophobia. So this is dislike of bright lights. Now, the key thing is, even if you have antibiotics, about 50% of survivors will still come down with some sort of long-term complications, deafness, limb amputations, and so on. And so, of course, this is one of those classic cases of prevention is better than cure. As I mentioned earlier, of course, we know that no matter the strength of the vaccines that we throw against this bacteria, they still manage to evade. The first protein-based meningococcal vaccine at the time of licensure in 2015 did have an efficacy of more than 90%. But of course, what we are seeing now from accruing data is that now this is reducing to about 70%. And there's nothing to say that this is not going to get worse over time. In terms of the pneumococcal vaccines, we have very effective conjugate vaccines, starting with PCV7 and 10 and 13. And now we're hearing that there are others that are going through the final stages of clinical trials that are looking at 20, 30 serotypes. When I first started working with this bacterium in 2014, there was just about 90 serotypes. Now we have more than 100. And there are some other papers that have said that, of course, this is going to continue to increase. So essentially, the nutshell is we do need to discover more antigens that will give us broader coverage. And of course, also look at things to make these vaccines cheaper, especially when we're thinking in the African context. Now, what's the approach we're using for antigen discovery? We go to patients that are convalescing from disease. We collect blood samples from them after about 7 days post-infection, and we clone antibodies from them. I'm not going to bore you about the antibody cloning process, but once we've cloned these antibodies, these are monoclonal antibodies, we screen them for reactivity against a surface epitope on the bacterium using flow cytometry. We also use ELISAs, but we found the flow cytometry is more sensitive. Next, we look at each one of these antibodies and assess its functional activity. In the field of meningococcal disease, we have a fantastic surrogate of protection, which is known as the serum bactericidal assay. And what happens in that assay, of course, is you take your antibody, whether it's plasma from mice, in our case, it's monoclonal antibodies. You add that to live bacteria, about 1,000 colony-forming units, and then you add exogenous complement. And what should happen if your antibody binds to a surface epitope is it will then recruit complement to mediate lysis. In the case of the pneumococcal bug, of course, we don't have a surrogate of protection, but the opsonophagocytic killing assay is generally accepted to be a good measure of functional activity of antibodies. So once you've identified those antibodies that are functional, the next question, of course, is then to find out exactly what antigen induced their production in the human host. And this, in a nutshell, is what we know as reverse vaccinology 2.0. Now, running through some of the data that we've generated over the years. As I said, this is more than a decade's worth of work. While this approach is very powerful, it's extremely laborious. And what we can see here, if I can find the button, okay, I can't find it. So on the top panel there, we have all of the antibodies that we've cloned that we know bind to a surface epitope. That's not exhaustive of the number of antibodies we cloned. In reality, we cloned more than 1,000 antibodies. And just about these ones were the ones that bound to a surface epitope. And on the y-axis, you can see all of the different meningococcal strains that we've screened against. So we had a 40-strain panel that are covering the most economically important strains and also representing geographic distribution. The take-home message from this is that we found antibodies that bind to multiple different strains, which means that the targets of these antibodies can actually compose a broadly effective vaccine. Next stage is to then see whether or not these antibodies can effect killing of the bacterium. And we've, in the case of the meningococcus, started with the serum bactericidal assay. And what we can see there, if you have a value less than 50%, of course, that means that's positive for SBA. And if you have anything beyond 50%, then it's negative for SBA. And we can see, of course, some of these antibodies mediating complement-dependent killing. For me, as the scientist working on this, it was particularly exciting for me to see that the very last antibody there, P2011D5, which seems to be binding to pretty much every strain, also has bactericidal killing. And we think we might have found that elusive universal epitope that's going to go into a vaccine that will give broad protection. The question that normally arises is, you've done this SBA, it's an in vitro assay. Do you still have expression of your antibodies cognate epitope in vivo? So what we did was we passively transferred 100 micrograms of some of these antibodies. We selected just 2 of them, the very first one and the last one. And we passively transferred 100 micrograms of these antibodies into mice and then challenged them with a lethal dose of the pathogen. And what we can see, which is self-explanatory, is that with P021A1, you do get some protection, which is better with P2011D5. And then when we use half of the amount, that's 50 micrograms of each antibody, and pull those together, you get full protection, which of course is established in literature. You get synergistic activity of antibodies. Similarly, with the work we've done with strep pneumo, we also cloned some antibodies, although fewer antibodies in this case because we had fewer patient samples to work with. For the purposes of this talk, I want us to focus on the second and the third antibody. That's P13-1D9 and HV1E12. We know what both antibodies bind to. I'm going to make it move because I'm not sure how this is working. You can see type 4 mutants there. And then the moment you delete that antigen, of course, you lose binding. And of course, we also can see that they mediate opsonophagocytic killing. Similarly to the in vivo assay with the meningococcus, we see protection for these two antibodies. With one of them, we found about 50% protection. With the second one, wasn't so great. But again, we did see synergistic protection when we pooled both antibodies. We also know exactly what part of the protein that those two antibodies bind to. What we've done here is we've fragmented the protein. We have a 300 amino acid at the end terminus of the protein. We spotted that onto a protein array, and then we also had other different fragments. And we can see here that the majority, if not all of the binding, actually is at the first 300 amino acids. One of the really interesting things here is we can even still narrow down further within that first 300 amino acids, and we can see that a lot more of the binding is within amino acids 151 to 250. Now we've used computational methods or AlphaFold to try to see if we could validate this data, and I can't show you that because the sequences are currently being protected, but we were able to exactly find the about 10 to 15 amino acid residue, which falls within the first 300 amino acids. Now, in summary, what this approach allows us to be able to do is to have this chimeric proteins. So we can have the epitope that each antibody binds to, and we can combine them together and graft them onto a protein backbone. And that will give us chimeric proteins, talking about next generation vaccine antigens. What that means is that we can have combination vaccines, so you can have an epitope from meningococcal antibody, combine that with an epitope from a pneumococcal antibody, and you have all of those strung along on one protein. This also means that we have enhanced immune targeting, because rather than having the full antigen, you can just really focus on the part that you know is immunogenic in humans. This will give us broader coverage, because as you could have seen from our data, if we looked at just those antibodies that give functional activity and you pull them together, pretty much we have full coverage. And finally, of course, we're going to be working with lesser proteins, and this will give cheaper vaccines. Science is very expensive. I would like to specifically acknowledge our funders, especially the UK Medical Research Council that have funded me for the past 10 years, and of course all of these other collaborators who have done fantastic work with us. I have been tasked to inform you that Imperial College now has this Global Connect Fund, which is a partnership between our Center for Bacterial Resistance Biology and WACCBIP, and specifically for the doctoral students. If you are interested in placement, especially in bacterial vaccines, please get in touch with us, and that can be under the auspices of the Global Connect Fund. Thank you very much for listening.
Thank you very much for that interesting talk. We'll now take questions from the audience. We'll take 2 questions for now.
Thank you for the presentation. I know you say you won't bore us with your antibody steps isolation, but that's actually what I'm interested in. Was that what method were you using to get your antibody? Is that probe or blind? What strategy?
Okay. I was actually very conscious of time, was the reason why I didn't want to bore you. So what we do is, once we've isolated the PBMCs from these patients, we sort them singly, one cell into each well of a 96-well plate, and then just a single free store cycle, release the RNA. We have the specific primers that will amplify the heavy, variable regions of the heavy and the light chain, those gene segments, and then we clone those into an IgG1 backbone, the vector. And then, of course, you combine the plasmid for the heavy chain and the light chain, co-transfect them into human embryonic kidney cells, HEC293 cells, and that will express your full-length IgG into the culture supernatant.
So are you sorting antigens with these cells?
Actually, yes. So we've done both. Initially, we started with sorting blind. Of course, that's the reason why the cloning precision was low, because from about 300 antibodies that we cloned, only about 8 of them were reactive. So what we've done since then is to use the whole bacterial cell, and we published this in 2023. So now we can use the whole bacterial cell to, as bait to identify and isolate the antigen-specific cells.
Any other questions from the audience? One more question there.
So my question has to do with, you spoke about splicing like the antigen of interest, let's say like instead of like the whole like organism or something like that, you splice the antigen with immunogenicity, like high immunogenicity. So like my question has to do with, let's say, if you extract the antibodies, the monoclonal antibodies, based on that antigen with high immunogenicity, what happens if like along the line, that particular antigen has been like mutated in an event or like in an effort by the organism trying to evade the immune system or trying to, you know, like change its course because of the pressure that the monoclonal antibody has on it? Like, what happened? Like, that's just my concern.
I'll just put this really simply. The vaccine antigen discovery process is going to be a continuous one.
Okay.
We're never going to get to that protein or that antigen that is going to give us lifelong protection. Like I mentioned with Bexero, the meningococcal vaccine, 10 years ago, 90% efficacy, now 70%. So it's going to be a continuous process. At least this will buy us maybe another 20 years of discovery of another antigen and vaccine. No.
All right. I'd like to thank all the chairs. You know, most people don't know this, but graduation has caused us to make a lot of changes to the program. And but the good part about that is that we have a number of graduates who we want to recognize. And these are so I'd like to, while we were here, the graduation was taking place. We weren't able to, we were represented, but all of us couldn't attend for obvious reasons. So we will recognize our graduates now from the PhD Molecular Cell Biology of Infectious Diseases program. So I hope, and then the MPhil program, I hope they're all here, so I'll call them as they appear in the record. Dr. Christopher Zabien Abana. Okay, maybe not yet here. He was supervised by Professor George Boateng Che, who spoke yesterday. He also spoke yesterday, Dr. Evelyn Yaria Boni and Professor Osborne Quay. Dr. Nana Egele Adade. Supervised by Professor Samuel Dodu, who just sat down, Professor Lydia Mosi, Dr. right at the back there, Dr. Stephen Della Ahato, and Professor Miguel Valvano. Dr. Cynthia Amisigo Malebna. Supervised by Professor Teresa Manfujira, current HOD of BCMB, Dr. Jack Santa, and Dr. Gloria Amegache. Dr. Angela Na Amele Ayiku. Supervised by Professor Samuel Dodu, Dr. Peter Kojokwashi, Dr. David Verna Jeffries, and Dr. Richard Paley. Dr. Alexandra Lindsay Zoom-Jokam. Supervised by Dr. Lily Pemka, Dr. Frank Liko, and Dr. Mehdi Buhadu. Dr. Franklin Yengdem Nukpem. He hasn't come here yet. Supervised by Professor Kwejo Asamoah-Kusi, Dr. Yao Aniwe, and Professor Gordon Awandari. Dr. Charles Ochieng Owal, supervised by Dr. Yao Bediako, Dr. Peter Kojokwashi, Dr. Mehdi Buhadu, and Professor George Boatinche. And then Dr. Sheila Santa. Supervised by Professor Osborne Quay, Dr. Charles Adokwe Brown, and Professor Emmanuel Aite Tego. I will also say that there's another PhD that I co-supervise in another department. I don't know if she's here, but Dr. Phillips Ekwia Amamu, supervised by Dr. Benjamin Oseibutre, Dr. Peter Kojokwashi, and Professor Gabriel Komla Ameka. I was just giving myself a shameless plug there. So, I think, and then we have the Master of Philosophy. Oh, that's, okay, they haven't graduated yet. So I can't call them up yet. But I think the PhDs can come up and take a picture with the boss. Okay, all the supervisors who are here.
I think it's okay. I mean, it's okay.
I think it's fine. Okay, ready?
All right, now I'll turn it over to Dr. Isaomi to take us through the Turbo Talks.
Yesterday we had Okay. Yeah, good afternoon once again. Yeah, so yesterday we had a good time here listening to our fellows presenting to us the interesting researches that they are doing. So this afternoon we are going to continue. Like I said yesterday, one slide, 2 minutes, no unnecessary animation, no video, and make it snappy as much as possible because of our time. So we are going to start with Becky.
Good afternoon, everyone. My project sought to look at how human genetics ties up to COVID-19 severity within the West African population. So we started off by looking at how SNPs and other genetic variations could affect the protein, which is ACE2, and how that can affect viral binding. I hope you will pass by my poster number 31 today. Thank you very much.
Thank you, Becky. Evans.
That was very snappy. Good afternoon. Yeah, so last Easter we heard some news. The Holy Bishop of Rome had gone to his father's house, and reports say he had multiple bouts of pneumonia before he went away with a heart attack. And I'm here to talk about the nasopharyngeal carriage, antimicrobial resistance patterns, and serotype distribution of Streptococcus pneumonia in diabetic patients in Ghana. But what's the point? So reports say he was a known diabetic and had multiple underlying conditions. Description that is fitting of about 1.4 million Ghanaians living with type 2 diabetes. And although these people are at risk of pneumococcal disease due to nasopharyngeal carriage, they have been excluded from the existing pneumococcal vaccination program. So the need for carriage studies. And I'm inviting you to poster number 2 or 32.
You don't know the number of your poster.
It says 2 in the email, but she said 31. Okay, so poster number 2 there to talk about pneumococcal.
Are you sure carriage. It's number 2? Anyway, poster number 2 or 32. Okay.
Yeah. So the name is Evans
you.
Thompson. I'm from the University of Cape Coast. Thank you.
All right. Yeah. Yeah.
Good afternoon. So, to study the development and the sequestration of Plasmodium falciparum gametocyte into the human bone marrow, we are using 2 different approaches. The first one exploits humanized mouse models. For creating these models, we put chondroid pellets from chondrogenic human precursors into under the skin of immunocompromised mice. After a few weeks, these pellets are able to form chimeric ossicles, and we demonstrated that by injecting immature Plasmodium falciparum gametocyte in the tail vein of these models, we are able to find them into the extravascular space of the chimeric ossicle after a few minutes. The second approach involves some discards from orthopedical surgery here in Ghana from asymptomatic individuals. So what we are going to do is to use these samples to find gametocytes into the intravascular and extravascular space of the bone marrow. Thank you.
Yeah, poster number 33. Poster number 33. Yeah, Stephen.
Good day to everyone. So it turns out that the malaria parasite is just like Gen Zs. I mean, the PFCSP gene keeps changing, and our vaccine is still stuck trying to keep up with this trend. So my study on the genetic diversity of the Plasmodium falciparum circumsporozoid protein, that's the PFCSP gene, across the 3 transmission zones in Ghana, aims to understand such diversity within the country, since the RTSS vaccine is being introduced at some point in Ghana. So if you're interested and you want to know more about the parasites implications on this vaccine, please meet me at poster number 34.
Thank you. Poster number 34. Coco.
Okay. Hello, everyone. So, away from infectious diseases, let's delve into food, which we all enjoy. How many of us worry about the impacts of our food on immediately and also on our future? I know some of our older people would do, however, not the deadly and toxigenic mycotoxins. These are secondary metabolites produced by some fungi that infect food commodities and animal products. And as the toxicologists will say, the dose makes the poison. So chronic mycotoxin exposure poses serious health threats to humans, such as mycotoxicosis, liver, and immune dysregulation. And whilst people have sought to use chemical means as a remedy, our research sought to use the antagonizing properties of naturally occurring microbes as a remedy for decontamination. And so meet me at poster number 35 if you want to know the amazing findings that we did. Thank you.
Thank you. So that's the poster stand for the foodies. Okay.
Hello everyone, my name is Juliet Nwekianan, and I'm excited to share with you a glimpse of my research. Imagine a cat hunting down a mouse. Fast, effective, and without collateral damage. Now, imagine using rodenticide instead. It might kill the mouse, but also could what? Poison the entire house. That is the dilemma we face with drug-resistant fungi. Our chemical solutions may work, but they pose serious side effects to humans and the environment. In my study, I isolated lactic acid bacteria from Meda, a Ghanaian traditional fermented maize drink. This lactic acid bacteria may act as natural microbial inhibitor, showing activity against Candida albicans and enterobacteria. This creates a room to exploring natural methods for microbial control and preservation. To know more or curious about finding more about this research, kindly visit poster number 38 for full details. Thank you.
Thank you, Juliet. Poster number 38. Darlington.
Klebsiella michiganensis from Michigan State is lurking in our Ghanaian hospitals. So this particular bacteria was first found in the state of Michigan a little over a decade ago, and it has been reported in other parts of the world, such as China, Switzerland, and in South Africa, but not yet in a part of the world. However, in our study, we found this right here in our hospitals. And if you want to know more about the public health risk it poses to us as individuals, as people, as health physicians, and in fact, everyone, please come to my poster number 39, right there. So it concerns us all.
Thank you, Darlington. Poster number 39, Klebsiella michiganensis from Michigan.
Please, I have a next slide. Okay. So, good day, everyone. My name is Belinda Kosio-Fosu, and my research focused on using in silico approaches to identify putative inhibitors of PFCDPK5, which is an essential parasite protein for merozoite egress. since it is not homologous to the human, it makes it a very good drug target. So in this study, we use homology modeling and molecular docking techniques to screen about 20,000 compounds, of which we found 5 compounds with favorable pharmacokinetics. Also, molecular dynamic simulation showed that they had stable interactions with key residues. So our future work would do in vitro testing to validate these proteins. We kindly come to poster number 44 to have a more interactive session. Thank you.
Thank you. Poster number 44. Benjamin.
Good day, everyone. My name is WACCBIP Benjamin Asenso, and the title of my poster is Serotyping and Molecular Characterization of Neonatal Sepsis, Associated Highly Resistant Streptococcus Pneumoniae. So globally, there are about 1.3 million cases of neonatal sepsis, and this can be attributed to Staph aureus, E. coli, and Streptococcus pneumoniae. But in Ghana, there's very limited information on the impact of Streptococcus pneumoniae on in neonatal sepsis cases. So our study seeks to provide insights into the virulence and pathogenicity of Streptococcus pneumoniae. Please do well to pass by poster number 45 today. Thank you.
Poster number 45. Samuel.
Next slide. Hi, everyone. My name is Samuel Intiamose. My work focuses on using machine learning to tackle one of today's health risks, which is antimicrobial resistance. Traditional methods delay in detecting resistance, which leads to fatal consequences. It also results in misuse of antibiotics. Now, I want to leverage on genomic and resistance data to train machine learning models to be able to predict resistant patterns and to be able to prescribe accurately. So visit my poster, number 46, for more information. Thank you.
Yeah, thank you, Samuel. Prince. Okay.
Hello, everyone. My name is Prince, and I'm working on HIV cure. So the story is pretty simple. So ARVs, or antiretrovirals, are what is currently being used to treat these patients, but they are not cure for HIV because they don't target the latent proviruses. And for that reason, studies have shown that they have to be on these ARVs for over 70 years before the reservoir will be cleared. So could natural products or herbal extracts be the next cure for HIV? Because, indigens are using these extracts to treat HIV and other infections, but scientific validations towards this are lacking. So in our lab, we sought to use 2 extracts, which are coded NL and ODO, to assess their antiviral properties as well as their effect on latency. So if you are interested, please pass by poster number 48 for us to talk more.
Thank you. Poster number 48. Martin.
Yeah. So my name is Martin Chamai. So I'll present about a new method of DNA extraction called a smart lead. So not all the steps that we go through while extracting nucleic acids are actually necessary. We can work a bit smarter by capitalizing on the 4 main steps of DNA extraction or RNA, engineer or design better reagents, and obtain results below 20 minutes. So this method has been successful for monkeypox, dengue, malaria, but it was a bit hard with archivable samples. So poster number 49 explains how exactly we made it a success for dry blood spots. See you then. Thank you.
Thank you. Poster 49. And Vanessa. Next person. Solomon. Okay.
Right. Good afternoon. My name is Solomon Amankwa-Osei, and my work is centered around plasminogen activator inhibitors 1 and 2 as diagnostic markers for pregnancy-related malaria-induced thrombosis. Studies suggest that 1 in 1,000 pregnancies are at risk of developing thrombosis. but 4 in every 1,000 pregnancies complicated with Plasmodium falciparum malaria is at risk of developing deep vein thrombosis. So we decided to evaluate the potential of plasminogen activator inhibitors 1 and 2 as markers for diagnosing malaria-induced thrombosis. Findings from my study suggest that they are good, but how good are they? I would like you to join me at poster 23.
Thank you. Poster 23, Solomon. Jessica.
Hello, everyone. I actually have a fun fact. I am standing here just 2 days after recovering from malaria. So the very parasite I studied tried very hard to knock me out this week. But thanks to an artemisinin-based drug, I was able to win the fight. So if that's where my, imagine first of all, what would have happened if the parasite was able to survive even in the presence of our very best anti-malarial drugs. I would have still been in the house and not enjoying the good WACCBIP food. But yes, so if you are interested in our in microbial gymnastics, please come to poster number 55, where I explain how the PF coronae may be giving the parasite some flexibility to evade and survive. So come to poster number 55. Poster number what? Number 55. Thank you very much.
Yeah. Thank you, Jessica. Before we go to the next person, I want to be sure you are not here for the food. So because of that, I want you to turn to someone sitting next to you and say, "This is who I am." If you are very familiar with Professor Awandari's story, it was one of these conferences that he attended that he met his PhD supervisor. You don't know who is sitting next to you. Maybe your next employer, your next supervisor, your next major collaborator, or prospective student. So can we quickly do an elevator pitch? 1 minute. Introduce yourself to who you are sitting next to. Some people, they are not talking on this side. All right, thank you. Oh, thank you. Okay, thank you so much. Yesterday, I wasn't able to introduce myself. My name is Abiola. I work on antimicrobial resistance. Okay, so let's go to the next person. I'm through.
Hello everyone. My work focused on the phenotypic analysis of Edwardiella tada isolates from cultured fish in Ghana. Edwardiella are bacterial pathogens of global concern in fish farming. Currently, there are about...
I'm to move closer to the mic.
Currently, there are 5 known species of this Edwardiella, with Edwardiella tada standing out as the only one with zoonotic potential. Although rare, it can cause bacteremia, which is very fatal, especially in immunocompromised individuals. While the MALDI-TOF MS, which is mostly used in diagnosing, is provides quick and reliable identification. It is expensive and not readily available to local farmers in low-income areas like Ghana. Hence, my work sought to establish a simplified phenobiochemical profile to accurately identify Edwardsella tada. Kindly pass by for more details.
Thank you. Poster number 56. Prisla.
Hi everyone, my name is Frederica and I'll be presenting on the topic genetic diversity of Plasmodium falciparum merozoite surface protein 1 in Ghana. So malaria vaccine research focuses on the different stages of the Plasmodium falciparum life cycle. Now the PFMSP1 is a blood stage antigen which elicits strong immune response. And then the N-terminal region of this PFMSP1 is highly polymorphic, especially at the block 2 region, which is characterized by 3 allelic families known as the K1, the MAT20, and then the RO33, which is driving this antigenic variation. Now in Ghana, since Ghana is a high burden malaria country, there's a need to determine the diversity of Plasmodium falciparum merozoite surface 1 across the 3 transmission zones in Ghana. To know more about this research, please pass by poster number 57 to know more. Thank you.
Thank you, Prisla. Esther.
Good afternoon, everyone. So, sulfadoxine pyrimethamine is an antimalarial drug that is used for the intermittent preventive treatment of malaria and seasonal malaria chemoprevention for children under the ages of 5 in high transmission areas. So, in Ghana, SMCs are administered in the northern regions of Ghana. The targets for these drugs are the 2 genes which are found in the malaria genome. That's the DHPS and the HFR genes. These genes are very crucial in parasite metabolism because they are needed for the production of folate, which the parasite needs for DNA replication and survival. Over the years, we've seen that the efficacy of this drug SP is being threatened by mutations found on the DHPS and the HFR genes, which are conferring resistance to the drug sulfadoxine and pyrimethamine respectively. Our study, this work seeks to determine the mutations that are being found on these genes and also to study their diversity and prevalence across transmission routes in Ghana. Kindly visit poster number 58 to satisfy your curiosity. Thank you.
Thank you. Poster number 58. Kukwao.
Good afternoon, everyone. My name is Patience Kukia, and my study assessed the phenotypic and genotypic characteristics of Staphylococcus aureus in clinical isolates from 3 hospitals in Ghana. Staphylococcus aureus is a major public health concern globally that accounts for hospital and community-acquired infections. According to the World Health Organization database, the proportion of bloodstream infection that is caused by methicillin-resistant Staphylococcus aureus is approximately 68% in Ghana. My study seems to assess these particular details. For more details, let's have a staff meeting at poster 29. Thank you.
All right, thank you. A meeting within meeting, so we'll visit the poster stand. Our last presenter for this section, Adam.
Good afternoon, all. My name is Awal Adamu, here to present to you part of our finding on, antimalarial drug surveillance in, northern Nigeria, to be specific in Kano.
Can you use the mic so that they can hear you?
Thank you. Now, the topic of the work is rarity of C-terminal Plasmodium falciparum multidrug resistance gene 1 polymorphism in Kano State, Nigeria. Well, it's not a good story to say that Nigeria remains the epicenter of malaria. And my state, Kano State, is unarguably the most populous in Nigeria. Unfortunately, we don't seem to have, much data on this really surveillance, of the anti-malaria drug resistant gene. So, we felt it's an opportunity for us to now go into search of really some of these mutants of Plasmodium parasite. So we began with this PFMDR1, which actually has been suggested to be a good molecular proof, maybe for unspecific molecular proof for this resistance. So I came here to share with you part of the finding. So if you're interested in the work, you can you're actually welcome to poster number 60 for more really.
Poster number 64. Thank you so much, Adamu. How many people have been to Nigeria before? Hey, you have to visit Nigeria before you die, okay? At least, as far as I know, we still have the best jollof in Africa. I know you will not agree with me, but I know Dr. Collins will agree with me because he has been to Nigeria and enjoyed the jollof. So I don't know if the jollof and lunch is ready this afternoon so that we... Please, can you get Dr. Okwashi? Is the lunch ready? Okay. So, and they said we are still awaiting the lunch. So, yes, a little stretch around. So we can go to the poster stand and have an interesting interaction with the presenters before we go for lunch. So, thank you.
What we do, we now look at each say fula. Fula and Ghana, they're everywhere, but that's where were those samples picked from? So now we checked in the data that fula from Ghana, fula from TB. Then, so, and luckily, even though they are going to merge their eventual ethnicities, we're able to get ethnicities that came from specific countries. And we use that now. So the first one that I showed you, that's the most interesting one. That one shows all the different ethnicities in West Africa and how which of them have the high glycophorins. Now, you can't, the only data that is out there, they don't tell you this person is Fula or that or the other challenge. This is why we came back, okay, we are going to do original analysis because I know this one, so we can put them in that region and then the child became clear that something serious. America, there was only Los Angeles, and you cannot use Los Angeles alone. It's even very diverse, so we have to take it out. Because at the end of the day, we will just say Africa ancestry, but where in Africa? Is East Africa? Is it West Africa? Interestingly, South Africa has zero, completely zero. Brazil, zero. Thousands samples, zero. And when you look at the death in South Africa, when you look at the death, very low. So, like I said, it's a story that is real there. Especially when I found a paper that was linking into RBCs, causing multi-organ damage, kidney and the rest. Because you know when RBC is the heme, so it realizes that the iron in the heme system causes all kinds of things. It's just an association to us. I don't want to look like I'm stretching and thanks to you again. We are growing. Was my second example. I'm happy to apply today. That's how I married. So we were on in Zambia. I kept hinting my daughter about this. No, he's my big guy. Leave this my office. This. So whenever it's time to cry, all my sis know that. And you sit down, lock the door. Oh, he did very well. Somebody The two of them. That's why I said for the first time I will show pictures of them. And I went back, and so I went to this system. When I came back, I see something. That's why I downloaded all the data sets. We created a whole pipeline and started.
It's just that, as we said, this is one of the, you can say that only this is one of the many factors why we,
So, I mean, started probably, then it hit me that I already have a the land really
inside the company.
Yeah, so that's how the whole thing
So what's next?
So next is to, for me, I actually made road for epidemic preparedness. Initially, I didn't want to do that because I felt with this conspiracy theories, if you begin to tell them this is why well, I was in New York. Yeah, I was in New York and I was explaining this to the desert, so I eventually made it the abstract submission. Then, oh, just last 2 weeks, I went for a meeting in Boston. I had friends in New York. Yeah, trying it, but because underground. Underground. Can you imagine what they would do now?
That was my daily invitation to work. That was my daily.
Oh, okay, sure. You enjoyed it?
Yes. After going to New York and look at that, maybe I'll make some.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meeting, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carrowmore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's Kaiser, South Africa, and our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center, and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa, and it's made me want to fight harder with the South African government to allow us to get visas for students so that we can have joint supervision of students, so that we can have more cross-collaboration. And yeah, I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here that being presented. I also think it's important to have people from around the world coming to this meal, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Now, going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you'll find here.
I'm David Conway at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carroll Moore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. Yeah. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, you know, the next 10 years can see WACCBIP go even further and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa, that's GAISA, South Africa, and our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa, and it's made me want to fight harder with the South African government to allow us to get visas for students so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meeting, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway, at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and with his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between the London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based research, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that it is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carrowmore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's GISA, South Africa, and our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa. And it's made me want to fight harder with the South African government to allow us to get visas for students so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meal, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to be working with Gordon and with his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between the London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based research, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that it is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carrowmore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. Yeah. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, you know, the next 10 years can see WACCBIP go even further and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa, that's GAISA, South Africa, and our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center, and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa, and it's made me want to fight harder with the South African government to allow us to get visas for students so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP ever since I can remember. So unfortunately, I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars, a lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meeting, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Now, going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you'll find here.
I'm David Conway of London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and with his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based research, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carrowmore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's GISA, South Africa, and our Our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa. And it's made me want to fight harder with the South African government to allow us to get visas for students so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here that being presented. I also think it's important to have people from around the world coming to this meeting, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway, at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between the London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carroll Moore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's GISA, South Africa. And our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center, and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa, and it's made me want to fight harder with the South African government to allow us to get visas for students, so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars, a lot of very significant work going on here that being presented. I also think it's important to have people from around the world coming to this meal, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Now, going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between the London School and WACCBIP and partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that it is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carroll Moore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the global immunology and immune sequencing for epidemic response in South Africa. That's GAISA, South Africa. And our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center and then even put together posters. That's always for me been a passion, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa. And it's made me want to fight harder with the South African government to allow us to get visas for students so that we can have joint supervision of students, so that we can have more cross-collaboration. I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meal, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Now, going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway, at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and his colleagues here at the University of Ghana since just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having a good relationship between London School and WACCBIP.
Hello everyone, my name is Romaïsta Djoutsa from Morocco. I'm a PhD student at the University of Ahmed of Fes. My research topic is combining artificial intelligence with lab-based experiments to tackle antimicrobial resistance. So as a PhD student, I recognize the global threats of antimicrobial resistance and the important role that bacterial genomics play in understanding resistance mechanisms. One of the powerful tools I have learned during this workshop is the whole workflow of next-generation sequencing, from sample preparation and library construction to sequencing using MySync.
My name is Roland Peter Komzuta from Ghana. I work as a biomedical laboratory scientist with the Ghana Health Service, specifically with the Eastern Regional Hospital, Koforidua. I joined this program because I wanted to gain deeper insight into antimicrobial resistance, especially using genomic tools like an NGS to decipher resistance patterns in bacteria. So when I found the form, I applied, and here I am on the program.
In 2023, we submitted a request to GSK to partner with us to organize 2 training workshops for African scientists. One workshop was to build capacity in microscopy and bioimaging, and the second workshop to build capacity in genome sequencing and bioinformatics. We received a lot of applications all across the African continent. This showed the high level of interest of participants to build their capacity in the subject areas. Due to the particular interest in the genome sequencing and bioinformatics workshop, we then submitted a renewal request to GSK to train more participants in these subject areas. We received approval from GSK for this subsequent renewal, and we are currently in the process of organizing these workshops. We received over 1,800 applicants for the genome sequencing and bioinformatics workshops from 47 different African countries, and we had to select 20 participants for each category of workshops, one for the intermediate level and one for the beginners level.
My name is Abiola, and I'm a research fellow at WACCBIP. Equally, I'm one of the facilitators of the AMR genomic workshop funded by GSK. Antimicrobial resistance is a global crisis with an annual mortality rate of 1 million, which might increase to 10 million by 2050 if we do nothing about it. The key challenge with antimicrobial resistance, especially within Africa, is lack of expertise. To bridge this gap, GSK has provided an opportunity to train the next generation of AMR fighters. And for the past 2 weeks, we have been guiding and training young African researchers from different parts of Africa. This is to say that we are building relevant capacity that can help us to deal with the challenge of antimicrobial resistance.
And then to act. It will act rapidly and then deal with it. So that's the implementation.
Because we do have the GSK funding, we are able to pull together young people, young scientists from east to west, south, and do this basic training. That is in 2 parts, a wet lab, NGS training that allows people to learn the workflows from DNA extraction or nucleic acid extraction to library preparation and next generation sequencing towards the identification of antimicrobial resistance. The other group looks at the downstream analysis, the bioinformatic tools that will enable them to do AMR detection. So you take raw sequence data, you clean it, you do that rigorous QC, and then you do genome assembly, and you are able to build draft genomes that we can now use other related tools and databases like the CARD database, the Pediatric database, to detect AMR resistance and specific pathogens that we have sequenced as part of this training.
The support that we receive from GSK has been extremely helpful. And this is because we have enough resources to be able to even broaden the scope of the training and provide enough time for the training. So we end up bringing a 2 weeks intensive course, and this allows us to give in-depth training to the participants, which is more helpful than having shorter courses because of limitation due to the limit of resources. The approach that we use, or the methodology that we use for training in our workshop, is more of a hands-on approach. And this is basically because of the nature of our work. So we want the participant to be able to have an experience and build confidence when they are working in the lab. So we allow the participants to practice on actual samples all the way from the beginning. You have the participants who work with actual samples and see the process, the entire process from sample QC, library preparation, library QC, all the way until they load the sample into the machine and they're able to generate the data. And this is a method or an approach that we've seen is more impactful as opposed to just talking to the participants and explaining to them what they are supposed to do.
My name is Mary Kameni. I'm a research student at the Molecular Diagnostic Research Group at the University of Yaoundé 1 in Cameroon. So actually, I'm a third-year PhD student. I have been struggling with my research, with some parts of my research. I really wanted to learn more on next-generation sequencing, and I was opportune to be selected among the 20 participants for hands-on training in WACCBIP on antimicrobial resistance, genomics of bacterial and bioinformatics. So coming to WACCBIP in these 2 weeks has really enhanced my skills because I wanted to learn about next generation sequencing for so long. I've tried several opportunities, but I couldn't get it. So this opportunity enabled me to gain hands-on on whole genome sequencing of bacteria using the Illumina preparation kit, from library preparation, going through the DNA extraction, DNA quantification, going through the documentation, and all the steps for sequencing. So I'm happy and I'm thankful to WACCBIP, and I'm equally thankful to DSK for granting me this opportunity, because the invaluable resources I've gotten here, the skills I've learned here are going to really help me to pursue my research activities back home in Cameroon and to be able to complete with my research objectives till I will defend my PhD. So I'm very grateful for this opportunity. Thank you very much.
Hi, my name is Victor Mosha. I am a laboratory scientist from the Kilimanjaro Clinical Research Institute in Tanzania. My motivation to attend this workshop is based on my upcoming project on the surveillance of Neisseria gonorrhea in Tanzania. In this project, we tend to do both phenotypic and genotypic characterization of gonorrhea. And my institute is well equipped with both molecular and microbiological equipment, but we are underutilizing it. So I want to use the knowledge and skills gained from this training to effectively utilize the equipment that we have in my institute to do the genomic work and phenotypic work for the Neisseria gonorrhea. I'm very grateful for WACCBIP and GSK for organizing this training. I know it is very expensive organizing such kind of training, and it is not even easy to get opportunity to do this kind of training. So for me, getting this opportunity, I am really grateful.
My name is Yemaachi. I come from Togo, and especially in Institut National d'Hygiène of Lomé. I thank the director of WACCBIP, Professor Gordon, and all the rest of the team of WACCBIP for doing a lot of work to help and in this project of training the young people in bioinformatics and AMR.
Hi, my name is Amina Saliu. I'm a veterinary epidemiologist. I'm from Nigeria. I have been opportunity to partake in the WACCBIP training on antimicrobial resistance, bacterial genomics, and bioinformatics. It's been very explicit, it's been very interesting. It's very wonderful because I've learned so much. As a researcher in antimicrobial resistance and food safety, I recognize the growing need of using bioinformatics in research in in discovering the resistance mechanism of the bacteria and also the evolution. I have been able to advance my computational skills in bioinformatics. I've learned so much. I've learned how to do bacteria assembly, annotations, use of BLAST, the antimicrobial resistance databases. I've learned so much. The facilitators have been wonderful. The colleagues, researchers, have been able to network with fellow like-minded researchers. I've also met experts. I've been taught by experts in the field, and it's been very impactful.
Teach others, even empower others. In fact, if you have the capacity, we are also organize a workshop for a few people. And transfer the skills. Amplify the effect. And tell them about WACCBIP. And invite them to also come here when they get the opportunity. So building a community of scientists for the future of Africa. There's a lot of talent on the continent. We believe that African scientists, we can compete with anybody in the world if we're given the right support and the right environment. And so we believe that once you come here and have this experience, you can go and try to also make a difference where you are coming from. And we'll continue to build a community and strengthen each other and empower African science. So we thank you. Stay in touch with us. Come back again. And wish you great success wherever you go. Tell us about your success, because these things help us to get support for what we're doing.
We'd like to thank GSK for this support, and we hope to continue partnering with them to build capacity for African scientists on the continent. Thank you, GSK.
Bonjour, my name is Christelle Loubeon. I'm a microbiologist and I'm from the University of Abomey-Calavi in Benin. First of all, I would like to express my sincere gratitude to WACCBIP and GSK for this insightful opportunity. One of the crucial and valuable lessons that I have returned from this workshop is the critical role of integrating genome sequencing and data analysis through bioinformatics tool to decode the complexities of AMR. For the first part of the training, I was part of the bioinformatics group, and I've built a strong foundation in data analysis, and this allowed me to analyze large data sets more effectively, and then making my research more robust and data-driven. And for this follow-up training, I'm focusing on enhancing my proficiency on genome sequencing, especially using Illumina technology.
In February, we did a hands-on training where we had both lab and downstream data processing training. And the current workshop is a follow-up on that workshop where we are selecting participants who have their own projects that are quite mature, where they have collected samples and have specific questions that they want to address. The aim is also to help some of them who are students and postdocs to achieve their postdoc objectives and their student research objectives. So the current work we are doing is a follow-up on the previous workshop, but it's more targeted at specific projects that we can take from sample collection through to quality control, sequencing, and data analysis.
Bonjour, my name is Hélène Kamasa. I'm a PhD student at WACCBIP. I had the opportunity to participate in AMR and sequencing workshop organized by WACCBIP. Through this workshop, I gained valuable skills and hands-on experience in sequencing methodology, bioinformatics, tools, and pipeline for AMR papers. I intended to use this knowledge gained in my ongoing research, focusing on cervical cancer and vaginal macroplasma.
I'm part of the GSK workshop. I facilitated some of the sections. For the past 2 weeks, it's obvious that they have improved in terms of their skills and expertise. They have been able to process data, understand data, analyze data, and also interpret it to help in terms of addressing public health related infections such as antimicrobial resistance, which is a global crisis. So I want to say a big thank you to GSK for the funding and also to WACCBIP and for the opportunity, especially to be a part of the success story of these young Africans. And my word will be that when you give people opportunities, they have a tendency to do well. And within Africa, we have young African talent who knows what they are doing. And with necessary support, they would do well and become global stars.
Hi, my name is Bigo Ayakubu. I'm a student of Komikuma University of Science and Technology and a participant of this NGS bioinformatics workshop. I think this is the follow-up of the initial workshop, which was basically on bioinformatics, and then we've come to continue with the wet lab aspect. And for me, this is an eye-opener because I'm learning how to culture bacteria for the first time. I'm also learning antibiotic sensitivity test for the first time, DNA extraction, and of course, sequencing. Going forward, I intend to put this kind of practice into my field of study, and then part of my PhD as well. I intend to go into antibiotic surveillance and of course characterization.
I would like to imbibe these techniques into my PhD work, which is on AMR surveillance in the Bono region. Thank you very much.
My name is Musa Shanga. I'm a scientist at the Radboud Institute for Molecular Life Sciences. I have been a friend and supporter of the WACCBIP for ever since I can remember. So unfortunately I haven't been here since the beginning, but the parts that I've seen, I've been very impressed. I've been very impressed with the presentations, seminars. A lot of very significant work going on here being presented. I also think it's important to have people from around the world coming to this meal, and so all of these have impressed me over this week. I've also had really great interactions with trainees and postdocs, and that's really exposed me to the very high level of training that is here. I mean, the training here is as good as it gets. Now, going to MIT or to Rockefeller or to Stanford or whatever, any of these elite institutions, you could find trainees of similar level that you find here.
I'm David Conway, at the London School of Hygiene and Tropical Medicine, and it's been my pleasure to have been working with Gordon and his colleagues here at the University of Ghana just before the foundation of the WACCBIP Centre, and to have enjoyed the last 10 years having
or is going online. Give me a PowerPoint.
And partners elsewhere in West Africa. I think our research, as UK-based researchers, has benefited from WACCBIP having joined together many collaborators who we were already working with. Yes, so I think that there's probably two things that stand out. One is the way that WACCBIP is engaging people beyond the pure scientific community. So you could say translating science into practice, whether it's technology and industrial applications or the public relevance of science. And at the same time, the other thing that it is very impressive is that it keeps the cell biology at the centre, and this cutting-edge cell biology, really new things that I have not been aware of before, that I'm learning about at this conference, which is really exciting. So I think having cell biology in the name of the WACCBIP brand has actually turned out to be really special, because many other centres don't retain that focus. So balancing the discovery, cell biology and the applications to society is something that I think has come out strongly in this conference.
A grant from Carroll Moore International. We're a supply and logistics company working in low middle income countries, and we've been working with WACCBIP doing their supply and logistics for equipment, consumables and reagents since inception, and with the players even before WACCBIP was created. I've already spoken to Gordon and to Lydia and just commended them on the opening ceremony. I think the most interesting opening ceremony I've been to for a long time, because they're usually somewhat tedious, and this one was very interesting, the variety of the speakers and the way that they broke it up with film excerpts that had been pre-arranged. So of the sessions that I attended, and I didn't attend all the sessions because it's always an opportunity to meet people and you end up being outside of the conference room, but the snake venom conversation, the snake venom talk that has just happened was interesting, because I've followed that over a number of years, and it is a very neglected element within the research world. But I did go to the pitch conference yesterday, the pitch session, where the groups were pitching their biotechnology ideas, and I thought that was really well done. WACCBIP has come a long way in 10 years. I don't think there's any centre that we visit and we work with elsewhere in Africa that has moved so far in such a short period of time. Usually it's taken twice as long or 3 times as long to get to the position that WACCBIP are in. So, you know, the next 10 years can see WACCBIP go even further, and will remain a solid mark on the research world in Africa, and hopefully make that one of those centres that keeps research south-south, not just north-south.
My name is Janelle Beeman. I'm the co-principal investigator for the Global Immunology and Immune Sequencing for Epidemic Response in South Africa. That's GISA, South Africa. And our laboratory is the Antibody Immunity Research Unit that's based at the National Institute for Communicable Diseases. I've been completely blown away by this conference. The opening ceremony was so inspiring. It was so wonderful to hear high-level people speak such wonderful accolades of WACCBIP. It really made me realize what an impact that the center has made over the past 10 years. The conference itself, the scientific presentations have just been of such a high caliber. I've enjoyed even the ones that I don't know anything about. I've enjoyed all of them. They've been clear, they've been concise and direct, and I think very, very good cheering has been a mark of this conference. I've loved the content, and I've loved the quality of the research that's been presented at the conference. A highlight for me was also seeing the high school students that were invited to come and experience the center, and then even put together posters. That's always for me been a passion, you know, reaching the young people from high school and school age. So I really like that initiative. And I was also very impressed with the entrepreneurship pitches that were presented. They had some really good ideas, and the pitches themselves were very well constructed. I think working with WACCBIP has really opened my eyes to the potential in Africa, the excellence that's present in Ghana and in broader West Africa, and it's made me want to fight harder with the South African government to allow us to get visas for students, so that we can have joint supervision of students, so that we can have more cross-collaboration, and yeah, I think it's just made me have a much greater appreciation for the potential and the excellence in Africa.
Bonjour, my name is Christelle Loubeon. I'm a microbiologist and I'm from the University of Abomey-Calavi in Benin. First of all, I would like to express my sincere gratitude to WACCBIP and GSK for this insightful opportunity. One of the crucial
Hello everyone, my name is Yemaachi from Morocco. I'm a PhD student at the University of Ahmed of Fes. My research topic is combining artificial intelligence with lab-based experiments to tackle antimicrobial resistance. So as a PhD student, I recognize the global threats of antimicrobial resistance and the important role that bacterial genomics play in understanding resistance mechanisms. One of the powerful tools I have learned during this workshop is the whole workflow of next-generation sequencing, from sample preparation and library construction to sequencing using MySync.
My name is Roland Peter Komzuta from Ghana. I work as a biomedical laboratory scientist with the Ghana Health Service, specifically with the Eastern Regional Hospital, Koforidua. I joined this program because I wanted to gain deeper insight into antimicrobial resistance, especially using genomic tools like an NGS to decipher resistance patterns in bacteria. So when I found the form, I applied, and here I am on the program.
In 2023, we submitted a request to GSK to partner with us to organize 2 training workshops for African scientists. One workshop was to build capacity in microscopy and bioimaging, and the second workshop to build capacity in genome sequencing and bioinformatics. We received a lot of applications all across the African continent. This showed the high level of interest of participants to build their capacity in the subject areas. Due to the particular interest in the genome sequencing and bioinformatics workshop, we then submitted a renewal request to GSK to train more participants in these subject areas. We received approval from GSK for this subsequent renewal, and we are currently in the process of organizing these workshops. We received over 1,800 applicants for the genome sequencing and bioinformatics workshops from 47 different African countries, and we had to select 20 participants for each category of workshops, one for the intermediate level and one for the beginners level.
My name is Abiola, and I'm a research fellow at WACCBIP. Equally, I'm one of the facilitators of the AMR genomic workshop funded by GSK. Antimicrobial resistance is a global crisis with an annual mortality rate of 1 million, which might increase to 10 million by 2050 if we do nothing about it. The key challenge with antimicrobial resistance, especially within Africa, is lack of expertise. To bridge this gap, GSK has provided an opportunity to train the next generation of AMR fighters. And for the past 2 weeks, we have been guiding and training young African researchers from different parts of Africa. This is to say that we are building relevant capacity that can help us to deal with the challenge of antimicrobial resistance.
And then to act. It will act rapidly and then deal with it. So that's the implementation.
Because we do have the GSK funding, we are able to pull together young people, young scientists from east to west, south, and do this basic training that is in 2 parts, a wet lab, NGS training that allows people to learn the workflows from DNA extraction or nucleic acid extraction to library preparation and next generation sequencing towards the identification of antimicrobial resistance. The other group looks at the downstream analysis, the bioinformatic tools that will enable them to do AMR detection. So you take raw sequence data, you clean it, you do that rigorous QC, and then you do genome assembly, and you are able to build draft genomes that we can now use other related tools and databases like the CARD database, the Pediatric database, to detect AMR resistance and specific pathogens that we have sequenced as part of this training.
The support that we receive from GSK has been extremely helpful. And this is because we have enough resources to be able to even broaden the scope of the training and provide enough time for the training. So we end up bringing a 2 weeks intensive course, and this allows us to give in-depth training to the participants, which is more helpful than having shorter courses because of limitation due to the limit of resources. The approach that we use, or the methodology that we use for training in our workshop, is more of a hands-on approach. And this is basically because of the nature of our work. So we want the participant to be able to have an experience and build confidence when they are working in the lab. So we allow the participants to practice on actual samples all the way from the beginning. You have the participants who work with actual samples and see the entire process from sample QC, library preparation, library QC, all the way until they load the sample into the machine and they're able to generate the data. And this is a method or an approach that we've seen is more impactful as opposed to just talking to the participants and explaining to them what they are supposed to do.
My name is Mary Kameni. I'm a research student at the Molecular Diagnostic Research Group at the University of Yaoundé 1 in Cameroon. So actually, I'm a third-year PhD student. I have been struggling with my research, with some parts of my research. I really wanted to learn more on next-generation sequencing, and I was opportune to be selected among the 20 participants for hands-on training in WACCBIP on antimicrobial resistance, genomics of bacterial and bioinformatics. So coming to WACCBIP in these 2 weeks has really enhanced my skills because I wanted to learn about next generation sequencing for so long. I've tried several opportunities, but I couldn't get it. So this opportunity enabled me to gain hands-on on whole genome sequencing of bacteria using the Illumina preparation kit, from library preparation, going through the DNA extraction, DNA quantification, going through the documentation, and all the steps for sequencing. So I'm happy and I'm thankful to WACCBIP, and I'm equally thankful to GSK for granting me this opportunity, because the invaluable resources I've gotten here, the skills I've learned here are going to really help me to pursue my research activities back home in Cameroon and to be able to complete with my research objectives till I will defend my PhD. So I'm very grateful for this opportunity. Thank you very much.
Hi, my name is Victor Mosha. I am a laboratory scientist from the Kilimanjaro Clinical Research Institute in Tanzania. My motivation to attend this workshop is based on my upcoming project on the surveillance of Neisseria gonorrhea in Tanzania. In this project, we tend to do both phenotypic and genotypic characterization of gonorrhea. And my institute is well equipped with both molecular and microbiological equipment, but we are underutilizing it. So I want to use the knowledge and skills gained from this training to effectively utilize the equipment that we have in my institute to do the genomic work and phenotypic work for the Neisseria gonorrhea. I'm very grateful for WACCBIP and GSK for organizing this training. I know it is very expensive organizing such kind of training, and it is not even easy to get opportunity to do this kind of training. So for me, getting this opportunity, I am really grateful.
My name is Yemaachi. I come from Togo, and especially in Institut National d'Hygiène of Lomé. I thank the director of WACCBIP, Professor London, and all the rest of the team of WACCBIP for doing a lot of work to help, and in this project of training the young people in bioinformatics and AMR.
Hi, my name is Amina Saliu. I'm a veterinary epidemiologist. I'm from Nigeria. I have been opportunity to partake in the WACCBIP training on antimicrobial resistance, bacterial genomics, and bioinformatics. It's been very explicit, it's been very interesting. It's very wonderful because I've learned so much. As a researcher in antimicrobial resistance and food safety, I recognize the growing need of using bioinformatics in research, in discovering the resistance mechanism of the bacteria and also the evolution. I have been able to advance my computational skills in bioinformatics. I've learned so much. I've learned how to do bacteria assembly, annotations, use of BLAST, the antimicrobial resistance databases. I've learned so much. The facilitators have been wonderful. The colleagues, researchers, have been able to network with fellow like-minded researchers. I've also met experts. I've been taught by experts in the field, and it's been very, very impactful.
Teach others, even empower others. In fact, if you have the capacity, we are also organize a workshop for a few people. And transfer the skills. Amplify the effect. And tell them about WACCBIP. And invite them to also come here when they get the opportunity. So building a community of scientists for the future of Africa. There's a lot of talent on the continent. We believe that African scientists, we can compete with anybody in the world if we're given the right support and the right environment. And so we believe that once you come here and have this experience, you can go and try to also make a difference where you are coming from. And we'll continue to build a community and strengthen each other and empower African science. So we thank you. Stay in touch with us. Come back again. And wish you great success wherever you go. Tell us about your success, because these things help us to get support for what we're doing.
We'd like to thank GSK for this support, and we hope to continue partnering with them to build capacity for African scientists on the continent. Thank you, GSK.
Bonjour, my name is Christelle Loubeon. I'm a microbiologist, and I'm from the University of Abomey-Calavi in Benin. First of all, I would like to express my sincere gratitude to WACCBIP and GSK for this insightful opportunity. One of the crucial and valuable lessons that I have returned from this workshop is the critical role of integrating genome sequencing and data analysis through bioinformatics tools to decode the complexities of AMR. For the first part of the training, I was part of the bioinformatics group, and I've built a strong foundation in data analysis, and this allowed me to analyze large data sets more effectively, and then making my research more robust and data-driven. And for this follow-up training, I'm focusing on enhancing my proficiency on genome sequencing, especially using Illumina technology.
In February, we did a hands-on training where we had both lab and downstream data processing training. And the current workshop is a follow-up on that workshop, where we are selecting participants who have their own projects that are quite mature, where they have collected samples and have specific questions that they want to address. The aim is also to help some of them who are students and postdocs to achieve their postdoc objectives and their student research objectives. So the current work we are doing is a follow-up on the previous workshop, but it's more targeted at specific projects that we can take from sample collection through to quality control, sequencing, and data analysis.
Bonjour, my name is Hélène Kamasa. I'm a PhD student at WACCBIP. I had the opportunity to participate in AMR and sequencing workshop organized by WACCBIP. Through this workshop, I gained valuable skills and hands-on experience in sequencing methodology, bioinformatics, tools, and pipeline for AMR detection. I intended to use this knowledge gained in my ongoing research, focusing on cervical cancer and vaginal microbiome.
I'm part of the JSK workshop. I facilitated some of the sections. For the past 2 weeks, it's obvious that they have improved in terms of their skills and expertise. They have been able to process data, understand data, analyze data, and also interpret it to help in terms of addressing public health related infections such as antimicrobial resistance, which is a global crisis. So I want to say a big thank you to GSK for the funding, and also to WACCBIP and for the opportunity, especially to be a part of the success story of these young Africans. And my word would be that when you give people opportunities, they have a tendency to do well. And within Africa, we have young African talent who knows what they are doing. And with necessary support, they would do well and become global stars.
Hi, my name is Yakubu. I'm a student of University of Science and Technology and a participant of this NGS bioinformatics workshop. I think this is the follow-up of the initial workshop, which was basically on bioinformatics, and then we've come to continue with the wet lab aspect. And for me, this is an eye-opener because I'm learning how to culture bacteria for the first time. I'm also learning antibiotic sensitivity test for the first time, DNA extraction, and of course, sequencing. Going forward, I intend to put this kind of practice into my field of study, and then part of my PhD as well. I intend to go into antibiotic surveillance and of course characterization.
I would like to imbibe these techniques into my PhD work, which is on AMR surveillance in the Buno region. Thank you very much.
Hello everyone, my name is Yemaachi from Morocco. I'm a PhD student at the University of Ahmed of Fes. My research topic is combining artificial intelligence with lab-based experiments to tackle antimicrobial resistance. So as a PhD student, I recognize the global threats of antimicrobial resistance and the important role that bacterial genomics play in understanding resistance mechanisms. One of the powerful tools I have learned during this workshop is the whole workflow of next-generation sequencing, from sample preparation and library construction to sequencing using MySync.
My name is Roland Peter Komzuta from Ghana. I work as a biomedical laboratory scientist with the Ghana Health Service, specifically with the Eastern Regional Hospital, Koforidua. I joined this program because I wanted to gain deeper insight into antimicrobial resistance, especially using genomic tools like an NGS to decipher resistance patterns in bacteria. So when I found the form, I applied, and here I am on the program.
In 2023, we submitted a request to GSK to partner with us to organize 2 training workshops for African scientists. One workshop was to build capacity in microscopy and bioimaging, and a second workshop to build capacity in genome sequencing and bioinformatics. We received a lot of applications all across the African continent. This showed the high level of interest of participants to build their capacity in the subject areas. Due to the particular interest in the genome sequencing and bioinformatics workshop, we then submitted a renewal request to GSK to train more participants in these subject areas. We received approval from GSK for this subsequent renewal, and we are currently in the process of organizing these workshops. We received over 1,800 applicants for the genome sequencing and bioinformatics workshops from 47 different African countries, and we had to select 20 participants for each category of workshops, one for the intermediate level and one for the beginners level.
My name is Abiola, and I'm a research fellow at WACCBIP. Equally, I'm one of the facilitators of the AMR genomic workshop funded by GSK. Antimicrobial resistance is a global crisis with an annual mortality rate of 1 million, which might increase to 10 million by 2050 if we do nothing about it. The key challenge with antimicrobial resistance, especially within Africa, is lack of expertise. To bridge this gap, GSK has provided an opportunity to train the next generation of AMR fighters. And for the past 2 weeks, we have been guiding and training young African researchers from different parts of Africa. This is to say that we are building relevant capacity that can help us to deal with the challenge of antimicrobial resistance.
That are being presented, and then act. It will act rapidly and then deal with it. So that's the implementation.
Because we do have the GSK funding, we are able to pull together young people, young scientists from east to west, south, and do this basic training that is in 2 parts, a wet lab, NGS training that allows people to learn the workflows from DNA extraction or nucleic acid extraction to library preparation and next generation sequencing towards the identification of antimicrobial resistance. The other group looks at the downstream analysis, the bioinformatic tools that will enable them to do AMR detection. So you take raw sequence data, you clean it, you do that rigorous QC, and then you do genome assembly, and you are able to build draft genomes that we can now use other related tools and databases like the CARD database, the Pediatric database, to detect AMR resistance and specific pathogens that we have sequenced as part of this training.
The support that we receive from GSK has been extremely helpful. And this is because we have enough resources to be able to even broaden the scope of the training and provide enough time for the training. So we end up doing a 2 weeks intensive course, and this allows us to give in-depth training to the participants, which is more helpful than having shorter courses because of limitation due to the limit of resources. The approach that we use, or the methodology that we use for training in our workshop, is more of a hands-on approach. And this is basically because of the nature of our work. So we want the participant to be able to have an experience and build confidence when they are working in the lab. So we allow the participants to practice on actual samples all the way from the beginning. You have the participants who work with
capture samples and see the process, the entire process from sample QC, library preparation, library QC, all the way until they load the sample into the machine and they're able to generate the data. And this is a method or an approach that we've seen is more impactful as opposed to just talking to the participants and explaining to them what they are supposed to do.
My name is Mary Kameni. I'm a research student at the Molecular Diagnostic Research Group at the University of Yaoundé 1 in Cameroon. So actually, I'm a third-year PhD student. I have been struggling with my research, with some parts of my research. I really wanted to learn more on next generation sequencing, and I was opportune to be selected among the 20 participants for hands-on training in WACCBIP on antimicrobial resistance, genomics of bacterial and bioinformatics. So coming to WACCBIP in these 2 weeks has really enhanced my skills because I wanted to learn about next generation sequencing for so long. I've tried several opportunities, but I couldn't get it. So this opportunity enabled me to gain hands-on on whole genome sequencing of bacteria using the Illumina preparation kit, from library preparation, going through the DNA extraction, DNA quantification, going through the documentation, and all the steps for sequencing. So I'm happy and I'm thankful to WACCBIP, and I'm equally thankful to DSK for granting me this opportunity, because the invaluable resources I've gotten here, the skills I've learned here are going to really help me to pursue my research activities back home in Cameroon and to be able to complete with my research objective till I will defend my PhD. So I'm very grateful for this opportunity. Thank you very much.
Hi, my name is Victor Mosha. I am a laboratory scientist from the Kilimanjaro Clinical Research Institute in Tanzania. My motivation to attend this workshop is based on my upcoming project on the surveillance of Neisseria gonorrhea in Tanzania. In this project, we tend to do both phenotypic and genotypic characterization of gonorrhea. And my institute is well equipped with both molecular and microbiological equipment, but we are underutilizing it. So I want to use the knowledge and skills gained from this training to effectively utilize the equipment that we have in my institute to do the genomic work and phenotypic work for the Neisseria gonorrhea. I'm very grateful for WACCBIP and GSK for organizing this training. I know it is very expensive organizing such kind of training, and it is not even easy to get opportunity to do this kind of training. So for me, getting this opportunity, I am really grateful.
My name is Yemaachi. I come from Togo, and especially in Institut National d'Hygiène of Lomé. I thank the director of WACCBIP, Professor Gordon, and all the rest of the team of WACCBIP for doing a lot of work to help, and in this project of training the young people in bioinformatics and AMR.
Hi, my name is Amina Saliu. I'm a veterinary epidemiologist. I'm from Nigeria. I have been opportune to partake in the WACCBIP training on antimicrobial resistance, bacterial genomics, and bioinformatics. It's been very explicit, it's been very interesting. It's very wonderful because I've learned so much. As a researcher in antimicrobial resistance and food safety, I recognize the growing need of using bioinformatics in research in discovering the resistance mechanism of the bacteria and also the evolution. I have been able to advance my computational skills in bioinformatics. I've learned so much. I've learned how to do bacteria assembly, annotations, use of BLAST, the antimicrobial resistance databases. I've learned so much. The facilitators have been wonderful. The colleagues, researchers have been able to network with fellow like-minded researchers. I've also met experts. I've been taught by experts in the field, and it's been very, very impactful.
Teach others, you want to empower others. In fact, if you have the capacity, we also organize a workshop for a few people. And transfer the skills. Amplify the effect. And tell them about WACCBIP. And invite them to also come here when they get the opportunity. So building the community of scientists for the future of Africa. There's a lot of talent on the continent. We believe that African scientists can compete with anybody in the world if we're given the right support and the right environment. And so we believe that once you come here and have this experience, you can go and try to also make a difference where you are coming from. And we'll continue to build a community and strengthen each other and empower African science. So we thank you. Stay in touch with us. Come back again. And wish you great success wherever you go. Tell us about your success, because these things help us to get support for what we're doing.
We'd like to thank GSK for this support, and we hope to continue partnering with them to build capacity for African scientists on the continent. Thank you, GSK.
Hello everyone, my name is Yemaachi from Morocco. I'm a PhD student at the University of Ahmed of Fes. My research topic is combining artificial intelligence with lab-based experiments to tackle antimicrobial resistance. So as a PhD student, I recognize the global threat of antimicrobial resistance and the important role that bacterial genomics play in understanding resistance mechanisms. One of the powerful tools I have learned during this workshop is the whole workflow of next-generation sequencing, from sample preparation and library construction to sequencing using MySync.
My name is Roland Peter Komzuta from Ghana. I work as a biomedical laboratory scientist with the Ghana Health Service, specifically with the Eastern Regional Hospital, Koforidua. I joined this program because I wanted to gain deeper insight into antimicrobial resistance, especially using genomic tools like an NGS to decipher resistance patterns in bacteria. So when I found the form, I applied, and here I am on the program.
In 2023, we submitted a request to GSK to partner with us to organize 2 training workshops for African scientists. One workshop was to build capacity in microscopy and bioimaging, and the second workshop to build capacity in genome sequencing and bioinformatics. We received a lot of applications all across the African continent. This showed the high level of interest of participants to build their capacity in the subject areas. Due to the particular interest in the genome sequencing and bioinformatics workshop, we then submitted a renewal request to GSK to train more participants in these subject areas. We received approval from GSK for this subsequent renewal, and we are currently in the process of organizing these workshops. We received over 1,800 applicants for the genome sequencing and bioinformatics workshops from 47 different African countries, and we had to select 20 participants for each category of workshops, one for the intermediate level and one for the beginners level.
My name is Abiola, and I'm a research fellow at WACCBIP. Equally, I'm one of the facilitators of the AMR genomic workshop funded by GSK. Antimicrobial resistance is a global crisis with an annual mortality rate of 1 million, which might increase to 10 million by 2050 if we do nothing about it. The key challenge with antimicrobial resistance, especially within Africa, is lack of expertise. To bridge this gap, GSK has provided an opportunity to train the next generation of AMR fighters. And for the past 2 weeks, we have been guiding and training young African researchers from different parts of Africa. This is to say that we are building relevant capacity that can help us to deal with the challenge of antimicrobial resistance.
And then it will act rapidly and then deal with it. So that's the technical mutation.
Because we do have the GSK funding, we are able to pull together young people, young scientists from east to west, south, and do this basic training that is in 2 parts, a wet lab, NGS training that allows people to learn the workflows from DNA extraction or nucleic acid extraction to library preparation and next generation sequencing towards the identification of antimicrobial resistance. The other group looks at the downstream analysis, the bioinformatic tools that will enable them to do AMR detection. So you take raw sequence data, you clean it, you do that rigorous QC, and then you do genome assembly, and you are able to build draft genomes that we can now use other related tools and databases like the CARD database, the Pediatric database, to detect AMR resistance and specific pathogens that we have sequenced as part of this training.
The support that we receive from GSK has been extremely helpful. And this is because we have enough resources to be able to even broaden the scope of the training and provide enough time for the training. So we end up bringing a 2 weeks intensive course, and this allows us to give in-depth training to the participants, which is more helpful than having shorter courses because of limitation due to the limit of resources. The approach that we use, or the methodology that we use for training in our workshop, is more of a hands-on approach, and this is basically because of the nature of our work. So we want the participant to be able to have an experience and build confidence when they are working in the lab. So we allow the participants to practice on actual samples all the way from the beginning. You have the participants who work with actual samples and see the entire process from sample QC, library preparation, library QC, all the way until they load the sample into the machine and they're able to generate the data. And this is a method or an approach that we've seen is more impactful as opposed to just talking to the participants and explaining to them what they are supposed to do.
My name is Mary Kameni. I'm a research student at the Molecular Diagnostic Research Group at the University of Yaoundé 1 in Cameroon. So actually, I'm a third-year PhD student. I have been struggling with my research, with some parts of my research. I really wanted to learn more on next-generation sequencing, and I was opportune to be selected among the 20 participants for hands-on training in WACCBIP on antimicrobial resistance, genomics of bacterial and bioinformatics. So coming to WACCBIP in these 2 weeks has really enhanced my skills because I wanted to learn about next generation sequencing for so long. I've tried several opportunities, but I couldn't get it. So this opportunity enabled me to gain hands-on on whole genome sequencing of bacteria using the Illumina preparation kit, from library preparation, going through the DNA extraction, DNA quantification, going through the documentation, and all the steps for sequencing. So I'm happy and I'm thankful to WACCBIP, and I'm equally thankful to GSK for granting me this opportunity, because the invaluable resources I've gotten here, the skills I've learned here are going to really help me to pursue my research activities back home in Cameroon and to be able to complete with my research objective till I will defend my PhD. So I'm very grateful for this opportunity. Thank you very much.
Hi, my name is Victor Mosha. I am a laboratory scientist from the Kilimanjaro Clinical Research Institute in Tanzania. My motivation to attend this workshop is based on my upcoming project on the surveillance of Neisseria gonorrhea in Tanzania. In this project, we tend to do both phenotypic and genotypic characterization of gonorrhea. And my institute is well equipped with both molecular and microbiological equipment, but we are underutilizing it. So I want to use the knowledge and skills gained from this training to effectively utilize the equipment that we have in my institute to do the genomic work and phenotypic work for the Neisseria gonorrhea. I'm very grateful for WACCBIP and GSK for organizing this training. I know it is very expensive organizing such kind of training, and it is not even easy to get opportunity to do this kind of training. So for me getting this opportunity, I am really grateful.
My name is Yemaachi. I come from Togo, and especially in Institut National d'Hygiène of Lomé. I thank the director of WACCBIP, Professor Gordon, and all the rest of the team of WACCBIP for doing a lot of work to help and in this project of training the young people in bioinformatics and AMR.
Hi, my name is Amina Saliu. I'm a veterinary epidemiologist. I'm from Nigeria. I have been opportunity to partake in the WACCBIP training on antimicrobial resistance, bacterial genomics, and bioinformatics. It's been very explicit, it's been very interesting. It's very wonderful because I've learned so much. As a researcher in antimicrobial resistance and food safety, I recognize the growing need of using bioinformatics in research in discovering the resistance mechanism of the bacteria and also the evolution. I have been able to advance my computational skills in bioinformatics. I've learned so much. I've learned how to do bacteria assembly, annotations, use of BLAST, the antimicrobial resistance databases. I've learned so much. The facilitators have been wonderful. The colleagues, researchers, have been able to network with fellow like-minded researchers. I've also met experts. I've been taught by experts in the field, and it's been very impactful.
Teach others, to empower others. In fact, if you have the capacity, we are also organize a workshop for a few people.
Yeah.
And transfer the skills. And they amplify the effect. And tell them about WACCBIP. And invite them to also come here when they get the opportunity. Yes, building a community of scientists for the future of Africa. There's a lot of talent on the continent. We believe that African scientists we can compete with anybody in the world if we're given the right support and the right environment. And so we believe that once you come here, you have this experience.
So we're almost there. We're just about to start the last session of the day. And as always, if you know somebody who is supposed to be here who's not here, just send them a message, tell them that the session has started. And slight change to the chairs. I'd like to first thank and then invite the advisory board chair, Professor Kurt Dich, for agreeing to chair this session, along with Dr. Collins Misita Muranga.
Good afternoon. You are all welcome to this session.
You're going to have the first plenary presentation from Isaiah Debra. Isaiah can come to the front. He's going to talk to you about whole transcriptome analysis reveals key metabolic genes involved in insecticide resistance in Anopheles gambiae from the coastal region. You can start. You have 15 minutes.
All right, thank you very much. Vector control is one of the major tools for malaria control, and we entomologists believe that once we kill all the mosquitoes, then there will not be malaria. And yes, vector control is the major tool that help in eliminating malaria in some countries. But the question is that easy to kill the mosquito? Because we know mosquito is the deadliest animal on earth. And once we kill them, there's no need for vaccine development, antigenic variation in the parasites, and all this stuff. Let's kill the mosquito first. Then once we kill the mosquito first, then we are free from malaria. But it's not easy to kill the mosquito. So for my talk, I titled it Whole Transcriptomic Analysis Reveals key metabolic genes involved in insecticide resistance in Anopheles gambiae sensu stricto, or in simple term SS, from coastal Ghana. Malaria is still a major public health challenge in endemic areas in sub-Saharan Africa. And in Ghana, malaria is endemic, and it varies across all the ecological zones in Ghana. And then in the southern, the coastal region and the forest belt, it peaks twice in the year. But in the northern savanna zones, we have one single wet season that we have increase in malaria transmission. Oh, okay. So vector control is critical and the most single effective malaria preventive tool. And it packs around the two major vector control tools: long-lasting insecticide-treated net and indoor residual spraying. But the challenge is that most of these vectors have developed resistance to the public health insecticides, and rapid emergence of resistance is a serious threat to malaria control. There are many mechanisms that mosquitoes adapt to resist or develop resistance to the public health insecticide. But for the purpose of my presentation, I will focus on metabolic resistance and then the cuticular resistance, that's sticking of the cuticles. Metabolic resistance poses a serious threat to vector control. This paper talks about the high intensity of resistance in the coastal region of Accra, and high resistance in vector control among malaria vectors has compelled the National Malaria Elimination Program to switch to dual active impregnated piperonal butyroside PBU nets, and then also switch from Atelic, the pyrimiphos meter, to Fedora fusion that are now being used for vector control. So we hypothesize that metabolic mechanisms are key mediators of insecticide resistance in Anopheles gambiae in the coastal zone of Ghana. Therefore, we aim to first investigate the phenotypic resistance to specific frontline public health insecticides and then also explore the underlying mechanism of resistance in the major malaria vector, Anopheles gambiae, in Greater Accra region. Specifically, we determined the intensity of resistance of Anopheles gambiae to the frontline insecticides in the coastal Ghana. And then also we went on to determine the mechanisms of resistance that may be at play. So we sample from various locations in the coastal region within Accra. So if you see us at the back of your house holding ladders, we are not there to collect your soup or to fetch your soup, but we are looking for mosquito larvae. So following successful sampling of the mosquito larvae, we do sorting in the lab, and then we move on to perform the WHO tube bioassays. We first perform the first diagnostic dose assay, and then we move on to perform the intensity assay, and then we perform the PBO, the synergies assay. Then we separate the legs of the mosquitoes, and then we extracted DNA from the legs, and then we went ahead to do speciation using PCR. So following RNA isolation and library preparation, we sequence the RNA using Illumina platform, NovaSeq Illumina platform. Then for the transcriptomic analysis, we have three main groups. We have the resistance group that we phenotype, that we expose them to deltametrin, permetrin, and pyrimidines. And then the susceptible Kisumu strain that we know they are very susceptible to the insecticides. And then we have the unexposed group that we collected from the field, all right, but we've not challenged them with any insecticide. Then we follow this pipeline, RNA-seq-POP computational pipeline developed by Nagy et al. 2003 for the analysis. Upon obtaining the FastQC, we went ahead to do quality control, reprocessing, removing the trimmed adapters, and then we aligned it to the Anopheles gambiae reference genome. And then following quantification, we use the seq for the differential gene expression analysis. From our phenotypic resistance, or from our data, using the WHO tube bioassay and using the WHO criteria for testing for insecticide resistance, when the mortality rate, which is the total number of dead mosquitoes exposed to the insecticide over the total exposed times 100, when it is greater than or less than greater than or equal to 90% is classified as the population is susceptible. Between 90 to 97 possible resistance, meaning that we have to confirm it by exposing it to subsequent population, the same population to the same insecticide. Then when it is less than 90%, then we say the population is resistant. So if you look at deltametrin at times 1 impregnated people, we have only 2 mortality rates, times 5 mortality rate, deltametrin, times 10 even, we are still getting 10 mortality rates. Compared to WHO criteria for testing for insecticide resistance, you realize that these mosquitoes were in the tubes and they were saluting us, they are not dying. And this is a worrying trend, because if the mosquitoes are not dying, then it means malaria is going to stay with us. Because the parasitologists, they couldn't find the solution, the vaccine people, they couldn't find solution. And the only people who will give us the solution is the vector control. But now, Gambi, the most efficient malaria vector in Ghana, is not dying. And even we went ahead to pre-expose them to the synergies. The synergy is the PBO. PBO action is that it inhibits the cytochrome P450, the major enzymes that metabolize the insecticide. So pre-exposure to PBU 4% before we expose them to the insecticide. But still, if you watch closely, 31% for this site in Accra. Then when you move on, the same story across board, different side. And once the mosquitoes are not dying, then it becomes a serious threat. We must find ways and means to see how we can work, because they must die for us to survive. Even as we are sitting here, the fish must die for us to eat. And nature is such a way that another organism must die for another organism to survive. You are challenging the parasite with artemisinin-based combination therapy for the parasite to die for you to survive. But the parasite came to seek refuge in you. So let's kill the mosquito, then we'll get the whole job done. And this is the same situation in other sites, high resistance across all the areas to the insecticide. We went ahead to look at our gene expression data, and then this is the total number of genes that we tested, and using the cutoff of p-value of 0.01 and then fold chain greater than 1.5, we found out that more genes are upregulated compared to genes that are downregulated. And then we move ahead to look at the major genes that are overexpressed in the resistance population, the etametrin and then the permethrin. And then we realize that cytochrome P450s, CYP6, M3, and all these genes are the major genes that were overexpressed in the permethrin and then the deltametrin. And then, unique to the deltametrin, we have the GSTs, glutathione S transferases. And then criticular protein gene families, we have this guy here that are overexpressed in the deltametrin. Then these same cytochrome 450s are the same genes that are overexpressed in the permethrin. Permethrin and deltametrin they belong to a group of public health insecticide called the pyrethroid. Then we move on to look at the permethrin, pyrimidine meter is an organophosphate. And then you realize that most of the cytochrome P450s, that genes that were expressed in the pyrethroids, most of them are expressed. And then carbonyl esterases. Then we move on to look at genes that are common genes that are expressed in all these insecticides, pyrimidine, methyl, deltametrin, and then permethrin. And these guys here, CYP6M3, CYP6M2, and these other guys, they are strong candidate genes that metabolize these insecticides. And these ones are unique to each insecticide. And then among them, CYP9K1, CYP6P3, these genes are important candidate genes that studies have reported they are role in pyrethroid resistance, metabolic resistance. From our heat map, we still have these genes that they are mostly expressed in all the insecticides, as I explained earlier. And then we have certain gene families, GSTs, that their expression is unique to the pyrimidines. And then the carbonyl esterases, they are unique to the pyrimidines. And the white shows genes that they are not expressed using the same threshold that I mentioned earlier. Salivary gland protein, SG3, is highly overexpressed in all the conditions. And then also this D7R1 is mostly overexpressed in the resistant population of all the insecticides. But then when you come down, cuticular protein genes, they are uniquely overexpressed in the control, but not in the test population, suggesting that either they are genes that switch off when they come into presence with the insecticide. This is subject to further investigation, because studies have reported the role of these genes in, they play a significant role in pyrethroid resistance. So we then look at the key genes associated with multiple resistance. And these cytogroup P450s are the major genes. If you look at their full chain, they are the major genes that we say they are associated with resistance in all the insecticides, the pyrimidines, the tametrin, and epinephrine. They could be profiled or prioritized for insecticide resistance monitoring in the coastal region of Ghana. And then we also have carbonzyl esterases, and then GSTs. That are overexpressed in all the populations. So in summary, Anopheles gambiae is highly resistant to pyrethroids at higher concentration, times 5 and 10, and then pre-exposure to the synergies couldn't restore susceptibility to the insecticide. PBO dual active bed nets may not fully mitigate pyrethroid resistance, highlighting the need to rapidly introduce other next generation active, dual active nets. Metabolic resistance mediated by cytogroup P450, GSTs, and carbonzyl esterases are important mechanisms of resistance to the insecticides. These are references, and I would like to thank our funding partners, Melinda and Gates Foundation, and then the Foreign Commonwealth and Development Office, NIHR, the World Bank, and Wellcome Trust, Ghana Health Service, and then to our boss, who is the PI, indefatigable Dr. Lucas, and then the co-PI, Dr. Niwe, Prof. Gordon, and the medical entomology team of WACCBIP, and the malaria, the bioinformatics lab. Sincere thanks to all of them for their support and providing funding for the project. Thank you.
Questions from the audience?
Thank you for this beautiful presentation. Sorry, my question may sound like jovial. Well, I have this crazy thought that it's not a good idea to eliminate mosquito. What? Yeah, really. What's your really? Yeah, you know, when you eliminate mosquito, do you really care about what organism will now replace them? I mean, their habitat. Don't you think we may have a substitute that is going to be more lethal than the mosquito? So, what's your thinking about this? Thank you.
Thank you too. Are you saying that the substitute you are talking about, are you saying that we need another substitute to come and transmit malaria? Or you are talking about the ecological balance or something like that? Well, we know that there are some animals that feed on mosquitoes. And you know, insects are very nutritious. And mosquito is highly nutritious, I mean, for them. But to us, we know that when they are no more, those animals or those organisms will feed on other organisms. It's like you want to eat wachi and the wachi salad doesn't have wachi, you take jollof, or equally take banku, and you are good to go. So I think they are doing more harm than good. Imagine all the animals on earth, mosquito is the deadliest. So let's do away with them and there will not be malaria. This is simple as that. No sympathy, okay? No sympathy.
A question over here? Towards the front?
Yeah. Prof, so with reference to the genes that are upregulated and the ones that are downregulated, does it matter how many genes are or what functions and roles the genes play? Would you say that because there are more genes upregulated as opposed to downregulated, it means they are not responding or responding to the drugs that they are challenged with, as opposed to what functions that those genes play, that is their role? What is your take as far as the numbers? Because I didn't get that.
I think the more the numbers too are overexpressed and their importance, we normally look at their importance. What role do they play in the metabolic pathway? Then that will tell us how, whether they are important or not. Now, your question is both. If they are more that are upregulated, then we look at which ones are they. Are they important? Are they metabolic genes? Are they genes that we know their notation, all those things? If once we know and well characterized, then we can move on. But then also we look at what the number, the role they also play. If they are few, what role do they play? So it's all important. The numbers, if they are many, upregulated, and they are very effective in metabolizing the insecticide, their role is characterized, is reported, or we move on to do functional validation, then that's good. If they are few and they are doing the job too, that's good.
Deborah, I have a naive question. So you are identifying genes in mosquitoes that are associated with resistance, right? So it's because the female Anopheles are the one that transmit the parasite, right? I'm just curious to know if there is a new way to discriminate male and female mosquitoes and see how then this resistance may be interfering.
Oh, okay, thank you very much. Now, in the mosquito world, or in the family of mosquitoes, unfortunately, the females are the troublesome ones. Male mosquitoes don't transmit malaria. They mate once after they emerge and they die.
You are killing, with insecticide, you kill all of them, right? So
yeah, and the males too, they are very weak. So they feed on nectars and sugar from plants and then they die. But the females are very resilient, very, very efficient transmitters. And once we do away with them, then there will be no problem. Studies are looking at how other studies, not within our study here, but they are looking at how they can genetically engineer these male mosquitoes so that when they go out and they mate with the females, they can reproduce. Or aim at reducing the population of the mosquitoes so that there will not be malaria. I hope it's clear.
Okay, thank you for your presentation. Mine is just an observation. From your data, it now shows that the PBO no longer serves as a synergist, right? So it now means, could it be that the enzymes that are involved in metabolic resistance have now, perhaps maybe in these insects, have been changed? Since they are still the markers for metabolic resistance, right? You still found the cytochrome P4, 50s, but the PBO no longer work. So I'm just thinking maybe if your group can look at these enzymes, maybe, perhaps maybe they have truly have been changed such that they no longer are affected by the PBO. Maybe your group can look into that. Thank you.
Yes, thank you very much. Resistance is very complex. And the mosquito use other mechanisms that we couldn't, study all. But we have another data that will look at other whole genome data that will look at other mechanisms of resistance in the vector population. Thank you.
Okay, let's give Isaiah a hand.
We are going to the next presenter on anti-malaria drug resistance, Emily S.
Good afternoon, everyone. I'm Emily Badoum from Groupe de Recherche Action en Santé, Burkina Faso. It's my pleasure to present our results on antimalarial drug resistance markers in P. falciparum, an analysis of genetic variants and haplotype. In 2024, we had around 263 million cases and around 600,000 deaths due to malaria in 83 endemic countries, with 95% cases and 96% of deaths reported in Africa. Around 76% of deaths were in children under 5 years of age. My country, Burkina Faso, is among the 10 most affected countries with 3.1% of cases and 2.7% of deaths. More than 11 million of cases and over 3 deaths were due to malaria in 2024. Despite progress in treatment and prevention, the effectiveness of control strategies remains compromised by the constant emergence of resistance to malaria drugs and the parasite genetic diversity. P. falciparum has the ability to adapt its genome under pressure from the widespread of use of drugs. Several genes have been identified as essential in the emergence of spread of resistance. We can cite like CRT, which confers resistance to chloroquine, MDR1, which is involved in resistance to several other antimalarial drugs, DHFR and DHPS, which are responsible for resistance to sulfadoxine pyrimethamine, and the key 13, which is associated with resistance to antimicinin. What's happening? Oh. The evolution of these genes through the acquisition of point mutations leads to a gradual loss of treatment efficacy, making malaria control complex. In addition, we have the CSP gene, which is a main surface protein of the sporozoid stage of P. falciparum, which appears to be polymorphic. This polymorphism could influence the vaccine effectiveness and allow the parasite to escape the human response. The combination of drug resistance and genetic diversity of antigens such as CSP could pose multiple challenges in our context. There is thus a need to closely monitor developments in order to adjust treatment policies and develop new generations of treatment and vaccines capable of overcoming P. falciparum rapid adaptation. Knowing the markers of resistance and the characteristic of key antigens such as CSP is not only a scientific challenge, but also the key to why not eradicating malaria one day. This study aims to identify mutations in antimalarial resistance and the CSP genes in P. falciparum population in Banfora. This study samples were collected in the southern part of Burkina Faso, where there is malaria is highly endemic, and where we have also an almost continuous transmission of malaria. The samples were collected from September 2020 to March 2021 in children aged 1.5 to 12 years living in Banfora. We did a 6-month longitudinal follow-up combined with case detection surveys of symptomatic and asymptomatic cases. We selected the samples by random. The samples were obtained from a study which aimed to primarily assess the incidence of clinical malaria and to identify the most effective anti-malarial drug to use for a pre-vaccination radical cure strategy. We did a follow-up for 24 weeks. For the study, we collect slides for where we use a microscopy to detect the parasite, and we also collect DBS to study the resistant gene. For the expression, we used QIAGEN, and after we did a multiplex PCR to amplify our gene of interest, and after we did Oxford nanopore technology sequencing. This study had been approved by the Health Research Healthy Committee. The analysis of individual bases were performed on the nanopore rapid analysis and variant explorer pipeline using MinMinon, and the results were analyzed and using air to calculate frequencies and to generate plots. Here we can see the samples characteristic. We had a median age of participants which were at 6.53 with a majority over 5 years of old, and the samples were mainly collected from asymptomatic patients. We had 4 synonymous mutations of the K13 genes. The low prevalence of the CRT could indicate, in our study, could indicate a gradual regression since the withdrawal of the chloroquine in Burkina Faso in 2005. The MDR1 mutation, a minor mutation at the position 86, low prevalence in our study, may suggest that AL used as first-line treatment still retain good therapeutic efficacy in our region. We try to know if there is a better coexistence of different mutations. For the DHFR mutation, the 51, the 59, and the 108 are close, which suggests a strong correlation. For the MDR1, the 2 mutations also coexist, but only moderately. And for the DHPS mutation, there are far apart, which suggests that there are by combining DHFR and DHPH haplotypes, we have only 1.16%, and the quadruple mutant which induce partial resistance to SP therapy is found at 19.65%, and we had no quintuple mutant, which indicates that the efficacy of SP could still be maintained in our study area. We obtained 25 SNPs of the CSP gene mutation in our study. Several studies highlighted a significant genetic diversity in different endemic regions, and the genetic diversity of this gene could compromise the long-term efficacy of the vaccine, namely ATSS and R21. Some studies in Africa and Asia showed that the mutations are more frequent and diverse in Africa than in Asia. Sorry. Oh, okay. We obtained a very little resistance to chloroquine, and we had no resistance detected to artemisinin, which reflects that that artemisinin is still high efficacy. The highest rate of resistance were observed for the sulfadoxine alone, which confirmed a major loss of efficacy of this molecule in monotherapy. Like a summary, we obtained moderate prevalences of the mutation associated with antimalarial drug resistance in P. falciparum in children after a radical cure, the absence of quintuple mutants and no validated K13 mutation seems that both SP and ACT may retain partial efficacy in our study area. There is a need to continue. The continued use of SP and ACT should be closely with the results obtained in some African countries. The diversity of the CSP gene suggests the need to monitor vaccine-targeted regions alongside drug resistance markers to better inform therapeutic and vaccine strategies. I would like to thank, at the end of this presentation, GRAC teams, WACCBIP teams for the training, children and their parents, and also all the founders. Thanks for your attention.
Questions from the audience? I can start. You mentioned the lack of CRT mutations associated with chloroquine resistance, that was quite low.
Yeah.
Was it much higher than that in the past?
Yes.
And then the second question I was going to ask is, has chloroquine been forcibly removed from the market, or can you still get chloroquine in private clinics?
No, I think maybe because for this study, we have a special population. It's not the normal population. To reuse maybe chloroquine, we have to use it with a normal population to see if you have the same prevalence before.
Thanks. Any other questions?
Thank you, Emily. So in Ghana, we see less than 0.5% of the CRT, the T allele for chloroquine resistance. But you are seeing around 5%. So, and I see your collection site is around the upper west region of Ghana, because your country overlaps ours all the way down. What do you think is happening? Would you say that, because people move across the border, right, from Hamele, right? They move up and down between our countries, and we have this difference.
Someone tells yesterday that mosquitoes don't need visa. So it's we have some studies where they had around 1.5 or less than, but as I said, this is special. So maybe it will be due to what you are saying, but I'm not sure.
What is the first line treatment in
AL
in Bonfara?
For
uncomplicated malaria.
Malaria, it's AL.
AL.
AL, yes.
Okay, that will explain it.
Any other questions? Okay, thank you.
Okay, thank you.
Thank you very much. So our next presenter is Philip Hilani, and he's going to give an online presentation.
Hello, can you hear me?
Yes. Philip, you can start. You have 12 minutes to present.
Okay. Good afternoon, everyone, and thank you for the opportunity. I understand one or two persons standing between you and the party, so I'll try to be as quick as I can. If you bear with me for a couple of minutes, I'll want to share with you what I've been doing as part of my PhD project, which seeks to functionally characterize mycin motos in the malaria parasite. From the two previous presentations, you would agree with me that the parasite has declared war for decades now, and all we are trying to do is to win. I'd like to start off with this quote that says from this book, The Art of War, that if you know your enemy and you know yourself, then you don't need to fear the result of hundreds of battles. And that simply means if you know your enemy, your chances of winning the war kind of increase. And so in the fight to win this war, I chose to study a family of proteins in the parasite called myosins. These myosins are motoproteins that walk along actin tracts. And they do this by hydrolyzing ATP to release inorganic phosphate. And the release of this inorganic phosphate and the tight binding of ADP, excuse me, results in a conformational change that allows the myosin molecules to undergo conformational change and then can bind and walk along actin tracks, as I'm showing in this video. These motor proteins play important roles in living cells. They are well characterized in humans and other animals. And this cycle of ATP release and hydrolysis of ATP is important for the functionality of these proteins. So myosins, the parasite myosins are quite different from the human myosins, and this gives us an advantage to design small molecules that can mediate, that can modulate this ATPase cycle. Canonically, this is what myosin looks like. They have the head domain that harbors the ATP binding site, the actin binding site, and then IQ motifs where light chains that regulate the activity of these proteins bind, like calmodulins and other proteins that I'll be talking about in a bit. And then some of them have a tail, and then these tails interact with cargos and other interacting partners to effect the function that they do. These proteins are quite interesting, and as a proof of concept, there's this publication that came out in 2023 where they have designed a small molecule that interacts with myosin A, modulates its activity, its ATPase activity, and an analog of this molecule has also been shown to kill parasites at multi-stage levels, and then even parasites that are already resistant to the frontline antimalarials that we use. So these proteins are quite druggable targets. So the problem we have seen so far is that the parasites are developing resistance, just like we've heard from the previous talk, and we need to, if we need to win this war, then we need to develop more arsenals to tackle the problem of malaria. At different points in the parasite life cycle, you would appreciate that the parasite need to generate force, for instance, when they want to get into red blood cells. Then they need to maintain shape, they need to move molecules around. And so to do these stage-specific events, the parasite requires this family of proteins called myosin motor proteins, which is why I was interested in studying them. For instance, in the invasion of erythrocytes by merozoites, we see myosin A, which is the far most characterized myosin. We see it interacting with actin and then provides the force that is necessary for the process of invasion. But that's not the only myosin we have in the malaria parasite. There are 6 other ones belonging to different families. A study in Plasmodium berghei, the rodent species of the parasite, tried to use a systematic mutagenesis approach to knock out these genes and see their functionality. And myosin A, myosin F, and myosin K were shown to block the progression of parasites, indicating that they are essential, but we do not know if this is the same thing in Plasmodium falciparum. In Toxoplasma gondii, myosin J, one of them, in conjunction with centrin 2, has been shown to affect to be involved in the contraction of the basal complex, which is the membrane that is underneath the and so what I sought to do then was to design a CRISPR-mediated riboswitch construct, which is a fancy name for tagging a gene with a sequence, this sequence called genomics. And what happens is we design guide RNA to the 3' end of the gene of interest. So I've done this for both myosin J and myosin F, but for the purpose of this talk, I'll be talking majorly about myosin J. And so we design guide RNAs, and then we co-transfect with the repair template. And what happens is when the gene is transcribed, it forms a chimeric mRNA that has the genomic sequence, which folds it into hairpin structures. And upon addition of glucosamine, this activates this tertiary structure mRNA that degrades the mRNA, and that way we are able to knock down the protein. And so once I got all these constructs in, I sequenced them to make sure that the constructs are good, and then I did transfection. Once transfectants are back, I PCR genotype them. So I have primers that are specific to the wild type, and I have primers that are specific to the GLIMI sequence. And so that way I'll be able to differentiate whether I got transgenic parasites back or wild type. And clearly you can see here in C and D for both of the guides I used here, I got transgenic parasites back. I then went on, because I had a triple HA tag on the protein, I went on to detect the protein by a HA antibody. And clearly here we see the protein for the 2 different clones I used here, and in 3D7 you see there's no detection of the signal, meaning that we got truly transgenic parasites. And in expansion microscopy, I also showed that we can detect the protein going in ring-like structures around dividing schizons. I tried to knock down the protein to see what effect it has on segmentation of schizons, because I noticed from my experiments that this protein was mostly expressed in schizons. And what I see is that there is no impact on the segmentation of schizons. And then I did similar things for mycin F. And this, while we were doing this in the lab, this paper came out which kind of agrees with what the results we were having. They were able to localize mycin J to ring-like structures that, like what I showed previously. And this also agrees with microarray data and even transcriptome data that the protein is expressed at the very tail end of parasite development. And so I started asking the question, what is the effect of knocking down these proteins on parasite growth and development? And so I treated parasites with different concentrations of glucosamine and then monitored them over 4 cycles. And here I'm showing for both 3D7 and mycin J tag lines, and I do not seem, there seem to be no defect in the growth and development of asexual stage parasites. I did statistical analysis to see if there will be any subtle difference, but clearly you can see the different concentrations of glucosamine. This is a Western blot showing that when we treat parasites with glucosamine for the tag line, this is untreated, we see the band, but when you treat parasites, the protein is degraded, therefore we do not detect it in Western blot. I also did an experiment where I took proteins that are localized to the inner membrane complex, like GAP45 and GAP45, and mycin A tail interacting protein, which are both localized to the inner membrane complex, and there do not seem to be any impact on protein expression for these proteins. While this was going on, the same lab that published the paper I talked about earlier then properly scooped me. They did very similar experiments which they published in PLOS Pathogens, and they showed that knocking out mycin J does not affect the growth and development of the asexual stage parasites, which was good for them, bad for me. But then it got me thinking of possible gaps that we could also exploit. And so what I identified is that at the moment we still don't know what the interacting partners of mycin J are, and we do not know the role that these proteins play in gametocytes, which are like the sexual stages of parasite. And so what I did then was to induce gametocytogenesis in 3D7 and my tag lines. And so what we do is to set up parasites at 1% rings and then allow them to go up to 3 days and then do limited media change. And then on day 4, we treat them with heparin to block subsequent asexual growth, because we now have high parasitemia, which would induce the formation of gametocytes. And then from day 5 down to day 14, we monitor gametocytes, making GIMSA stains, and then fixing parasites for immunofluorescent assays. And so I was able to generate these gametocytes. These are stage 5 gametocytes for both my 3D7 line and the J tag line. And so I started asking the question, what is the localization pattern of this protein in gametocytes? And here I'm showing 3D7 strains where I have labeled with my HA, GAP45, and tubulin antibody, which is a protein that is very abundant in gametocytes. And clearly, as you can see from the white arrow I'm pointing here, shows the similar staining patterns for GAP45 as what is published in literature. And when I labeled my taglines with anti-HA antibody, clearly you can see, even though there is some level of background for the HA antibody, clearly you can see specific staining of gametocytes from different stages, especially the later stages where you see enrichment of the proteins going around the periphery of the parasites, and especially enriched at the polar ends of the parasite. And we see something quite similar with tubulin antibody, as we see here. When you co-localize them, when you merge them, rather, you see this yellow staining, which indicates some level of co-localization. I took one of the images, zoomed them out a bit for you to see, and here I'm showing this yellow color, which indicates some level of co-localization. But the challenge is it's hard to say what is co-localized or not at this point using regular IFAs, which is why I then resorted to expansion microscopy, where you seed your parasites on cover slips and then make a gel on it, and then using cross-linking agents, you are able to expand the gel. And as you expand the gel, you expand the samples, and that way we're able to get expanded gametocytes, which we then image on the microscope. So here I stained with HA antibody, GAP45, and then tubulin, just like I did previously. And you can appreciate then the level of details you can get. And so we see GAP45 in the outer membrane of the parasite, and we see HA, the white arrow, showing that it's just underneath the GAP45. And I make this short movie just to show you what I'm saying. You see GAP45 going around the periphery of the parasites, and then you see the HA staining in green, which is just underneath the IMC staining. And then it's also very similar to what we see for tubulin, and you see the same similar staining pattern for tubulin. If you flip this around just a bit for you to see, you see GAP45 going around, and then tubulin and HA staining going just underneath the GAP45 staining. And this is important because the localization of the protein kind of determines the function it performs. I slice this up a bit for you to appreciate what I'm saying. We see GAP45 there. You see the HA and the tubulin just underneath the GAP45. And this is similar to what has been reported previously for sporozoids, where you see the parasite plasma membrane, the inner membrane complex, and you see tubulin staining. So it appears that myosin J kind of intercalates with tubulin structures, but further experiments is then required to decipher this. And then what I then sought to do was to determine if myosin J is important for gametocytogenesis. And so I induced gametocytes again, and then treated them with 2.5 millimolar glucosamine. And then here I'm showing the wild type treated and untreated, and you see there is no defect on gametocytes, whether we treat them or untreat them with glucosamine. And then I did the same thing for my tagline, and clearly you can see there seemed to be a defect in, especially from stage 3 gametocytes, when I then treat with glucosamine, indicating that myosin J could be playing a role in gametocytogenesis. I'm now quantifying this to put numbers to it, determining how much gametocytes come from these taglines, and then also do things like maturation and S-flagellation assays. But the question still remains, is myosin J truly knocked down in these parasites? And so I then induced gametocytes and then harvested lysate and did Western blot. And here I'm showing when you don't treat gametocytes, you see the expression of myosin J, but upon treatment with 2.5 millimolar glucosamine, there is a reduction, although not a complete knockdown, in the expression of these proteins. I got samples from a collaborator's lab from Plasmodium bergii, and clearly you can see that my antibodies are quite specific, myosin J antibody and even the H antibody is able to pick up a protein. And then I also use PCR to make sure I'm working with the right parasites, just the same genotyping assays I showed earlier, confirming that. But then what cellular processes do these proteins play in the parasite? Just have a few slides left now. So I did co-immunoprecipitation assays, and this is just my first run, and clearly I was able to pick up myosin J, and then I also picked up myosin-related proteins like the PF centrin-2, actin, and then calmodulin, which is known to interact with myosins. I then went on to do protein informatics to kind of verify some of these interacting partners. I used AlphaFold, which is an artificial intelligence enhanced protein structure prediction software program. And what I'm showing here is the structure of myosin J, the head domain, and then the neck region, and then PF centrin-2, and I'm putting in PF centrin-2 and myosin J to see if they could interact. What is interesting here is that centrin-2 could have bound to the motor neck, to any part of the protein, but then it binds to the neck domain, which harbors the IQ motifs I mentioned earlier, and that's interesting. I then tried different combinations of centrin-2 and myosin J, using 2 copies of centrin-2, for instance here, adding calcium, because centrin-2 is an EF-HAN containing protein, and calcium kind of affects how the binding of these proteins to their targets. And here I'm showing when you add 2 copies of centrin-2, it kind of distorts the neck domain of the protein. Adding 2 copies of centrin-2 and calcium ion kind of restores the distorted domain here, but then it is still not looking good. And then when I tried 3 copies of centrin-2, it restores completely the orientation of the protein architecture of myosin J, and then when you add calcium, centrin-2 kind of reorients in this direction, which is interesting. I then went into the lab, made constructs for these proteins expressing GST tagged myosin J motor neck, and then the different tail domains. Here I'm showing the expression gels in insect cells. We see myosin J expressed here, the tail 1, tail 2, and then the tail 2 separately. I used the antibodies against the tags, GST and his antibodies and my myosin J antibody. And clearly I'm showing here the detection of the different fragments of the protein, and the pre-immune did not detect the protein, showing that the antibodies are specific. I then did this final experiment where I co-transfected insect cells with GST, myosin J motor neck, and PF centrin-2. And here you see myosin J co-transfected with PF centrin-2, and then centrin-2 all by itself. We see a faint band here, which in Western blot using anti-MYC antibody, because centrin-2 is tagged with MYC antibody, with MYC tag. We see the protein coming down in the bead pellet, indicating that the protein could be stuck with myosin J, which could be which indicates that the protein could be interacting with myosin J.
Okay.
To summarize, I've shown you that we are able to generate transgenic parasite lines using the CRISPR-riboswitch system. We knocked down the protein, and we did not see any defect in the growth and development over 4 cycles. I've also been able to show you my first run with profiling the interactome of myosin J, and we have evidence that centrin-2 could be interacting with myosin J. I've also shown you that when we knock down myosin J in gametocytes, there seem to be some these funky looking gametocytes, which could be indicating that myosin J is playing a role in gametocytogenesis. And finally, I was also able to show you that we are able to localize centrin-2 to basal ring-like compartments in schizons, and even in gametocytes underneath the IMC, which indicates which indicates localization to basal complex. If there are people in the audience who...
Philip, I think we have plenty of scientists here, so we don't need the layman's summary. Thank you.
Okay.
Do we have any questions for Philip? I have one question for you, Philip. Are you still with us?
Yeah.
So when you showed the gametocytes seem to have an effect with your knockdown, male and female gametes have very different motilities. Did you notice any differences between males and females in your knockdowns?
I'm yet to quantify that. I've just got markers to detect male and female gametocytes. This is something I'm planning to do, but I'm yet to check that.
Any other questions from the audience? Okay, thank you, Philip.
Thank you very much.
All right, we'll take the last presentation for the day from Elkanah Evans, who's going to switch us from Plasmodium to Trypanosome. He's also presenting from online.
Good evening, my name is Evans Mauta Elkanah. I'm presenting on the discovery of the novel. So just a minute. So I'm presenting on the discovery of novel trypanothione reductase inhibitors through pharmacophore modeling, virtual screening, and molecular dynamic simulations. New insights for human African trypanosomiasis. So HAT or sleep sickness is a tropical neglected disease of the global health concern, especially in the sub-Saharan Africa. Over 92% of the cases are caused by T. brucei gabiense, and they are normally found in the central and the Western African regions. 7% of the cases are normally distributed in the Eastern and the Southern African region, and they are caused by T. brucei rhodesiense. All the forms are transmitted by the Glossina or the tsetse flies. The diagram you are seeing here is a simplified pathogenesis of the disease. Now, for the HAT treatment, during the early stage, pentamidine or suramine drugs can be used to clear the parasites since the parasites are already in the blood. However, during the late stages or the maligoencephalitic stage, the parasites have already crossed the blood-brain barrier, and therefore these two drugs cannot be used. In that case, melasoprol is a standard drug for the treatment of the HAT, but it is met with some limitations, including high toxicity because of the arsenic compounds, and also the drug has got a growing resistance. Also to mention is that the patients have got high risk of encephalopathy, which have got incidences of between 2 to 10% of the cases. And also sadly to report is that all the patients who have this encephalopathy, 50% of the patients, they are likely to die. And therefore, there needs some intervention to be done. Encephalopathy is nothing but a severe and potentially fatal neurological post-treatment reaction obtained by using of the melasoprol. Because of these challenges, there has been some advances whereby we have got several alternatives of the nifurtimox and the flunithiol combination. These ones have shown improved efficacy and reduced resistance, but this still requires multiple IV doses, which are logistically challenging to the patient. And also, since 2021, there was the introduction of the fexinitazole. That is a 10-day oral treatment for early and late stages of the HAT. But again, this one is showing to be having less effective in case whereby there is a severe late-stage cases. So therefore, we need aggressive options, which is still needed. So our research focused on development of a drug candidate that works as best as melasoprol, but with less side effects. The approach that we used, we started with the pharmacophore modeling, in which we used particular software called Protein Plus to map out the key pharmacophoric features between melasoprol and the target, that is the trypanosome brucei trypanothione reductase. And then we prepared a library of active and inactive compounds, and then we were able to screen this particular combination. We were able to validate that particular pharmacophore query using this particular enzyme, that is the trypanothione reductase, that is the major target. We carried out this particular structural-based virtual screening across 5 major databases, which are actually largest including maltod, coconut, and others. And then we were doing virtual screening in cycle-wise. Every round we were carrying out screening for one, and then we were able to rank all the hits. And then we were able to pick the first 20 of the hits. That is the first stage of the screening. And then we use that one as a multiple ligand molecular docking. And then after doing all that, we were able to come up with 2 molecules, which we assigned called the VS1 and VS2. And then we carried out to do structural interaction to find out whether these particular ligands were fitting into active site of the protein, that is the trypanadiol reductase. We proceeded to do pharmacokinetics evaluation, and then to validate this particular interaction, we were able to carry out the molecular dynamic simulations across the 100 nanoseconds. And then we also did a free binding energy calculation using the MMPBSA, that is the molecular mechanics Poisson-Boltzmann surface area method. And then we concluded by carrying out per residue energy decomposition. Now, as I mentioned, we were able to, we started by deriving the pharmacophore properties of the melasopro, that is a standard drug, and we were able to use 3 of the pharmacophore properties as predicted by the protein plus. And this one included the hydrogen bond donor disposition, hydrophobic as well as aromatic. And then from that, just to mention it, that we were able to carry out, we found 3 active compounds which were having very good IC50s of less than 10 micromolar, and then we generated 14 conformers from that. And then we were able to create decoys, 60 decoys we created from the gene database. That is a database of useful decoys. And then we were able to validate this particular mixture of decoys and active compound using the AUC. And then using that approach, we were able to screen 56,940,690 unique molecules from the 5 databases. That one includes the zinc, PubChem, melamine, coconut, and then maltod. Now, for the validation purposes, as I mentioned, we did receive operating characteristic curve, whereby upon the ranking of the prepared library, as you can see from this diagram here, our model was able to correctly identify an active compound, was able to distinguish between an active compound from decoys at 1.7% better than a random selection with around 75% confidence interval. And then we were able to, so our results showed that both VS1 and VS2, which was our top 2 hits, had better affinity to the trypanadiol reductase enzyme compared to melasopro, which is the standard drug, whereby VS1 had negative 11.3 kilocalories per mole, while VS2 had around negative 10.6. This one, they are better compared to negative 7.8.
You need to speak a little bit louder.
Okay. Can you hear me now?
Yes, it's better.
Yeah. So after we did superimposition of the ligand against the receptors, the ligands were able to be fitting to the receptor. And then we carried out also VS1 and VS2 showed a stable binding in the enzyme active site, whereby the VS1 was held in position by the hydrophobic bonds, while the VS2 was stabilized by hydrogen bond, which were dissociated by asparagine 130 and histidine 128. And then after that, we were able to absorption, distribution, metabolism, excretion, toxicity prediction, and most importantly, we found that the VS1 and VS2, which is molecule 1 and molecule 2, they were able to be predicted to be able to be absorbed in the GIT. And then we were able to carry out the entire screening that I have not been able to show here, but most importantly, most of the drug violation rules were not violated in our compound, including Lembitsky's, Goose, Vaba, and Igen, and others. Now, VS1 and VS2 also, the complexes maintain very low and stable RMSD as compared to the upper forms, having a fluctuation of less than 0.4 nanometers. And that one was because it shows that the ligand was best fitting in the enzyme active site. Now, we were able to carry out this particular root mean square deviation, especially because of some amino acids in the active sites. They are normally flexible, and it also showed very little changes of less than 0.5. This one implies that there was not so much changes in the active site. Finally, on the same note, we were able to check the structural conformity or the structural compactness of the enzyme with the ligand complex. This is because we believe that the enzyme are dynamics in nature and therefore they can fold and unfold. Therefore, to do that, we carried out radius of gyration that is specifically total and around the axis for both VS1 and VS2. And we could see that the upper form, okay, here you can see the black one, the TBTR. This one is the trypanosome brucei trypanodione reductase when it is not in bound form. And then the red one it shows is complex with VS1, and then green one is complex with VS2. We can really appreciate that the one that is bound with the ligands they were more compact than the one that is the one in the upper form. We finally continued to carry out the hydrogen bond interactions, and therefore we could see that our complexes were forming stable hydrogen bond interaction across the 100 nanoseconds, whereby the VS1 was having between 1 to 3 hydrogen bond interaction with the enzyme, and then the other one was having between 2 to 3. We carried out this one called solvent accessible surface area, and we could appreciate that the holoform was having smaller SASA values, and that is the smaller solvent accessible surface area values, meaning that the binding of the ligand to the enzyme was occupying some considerable amount of surface of the solvent accessible surface area, meaning that the ligand were fitting in the active site. Maybe to sum up, we finally did this particular high free binding, free binding energy, and we could find that the VS1 was having very high free binding energy. This one we employed the generalized bond equation, and we could see that the VS1 free binding energy was negative 34.93. And upon going further, we were able to carry out the per residue energy decomposition, and majority of this energy was coming from VS1 313, which we had assigned around negative 7.57. Same apply to the VS2, negative 32.93, and this one coming still negative 11.84. This meant that the binding was not by random, but it was basically strong binding. Maybe to summarize, we screened around 56,940,690 unique molecules across the 5 chemical databases. Following the structural based virtual screening, we obtained 2 ligands, VS1 and VS2, with better binding affinity against trypanosome trypanodione reductase enzyme than melasoprol. Both VS1 and VS2 had better pharmacokinetic properties than melasoprol. And then we propose that the future work should include in vitro screening as well as in vivo studies. Allow me to acknowledge this team who really contributed to the work. Prof. Jira, who is the chair of department of biochemistry, and then Dr. Coddings, and then others. Thank you.
Questions from the audience? I have a question. Do these compounds exist yet, and how hard will they be to make?
Yeah, the compounds, they exist. The databases that I screened against, they are there, but they carry different forms. So they exist. It's only a part of ordering from the vendors. Thank you.
Any other questions? I think we have an exhausted audience. Okay, thank you.
Thank you very much.
Thank you, chairs, and thank you, audience. Just a few housekeeping before I turn it over to the boss. First off, thanks to everybody who scored the posters and the orals over the last 2 days. Philip was the last person to be scored today, so we'd like to give everybody a chance to score the orals today. And then we will announce the scores during the gala. But I think now I would just like to call Professor Andari to begin the closing ceremony. And it's noteworthy that we're still late, but it's not by that much.
All right. So, 3 days later, I think it has been another very, very interesting gathering. When we started this conference, you have an announcement?
Yeah, sorry. So I forgot one thing. Those who are here will receive a link for the monitoring and evaluation to do the conference evaluation, and then those online will also receive a separate link to talk about the sound and all those things. So just be aware, and please try and fill it out for us so that we can make any adjustments for next year if needed.
So I was saying that when we started this about 10 years ago, the main objective was to provide a platform for our students and fellows to present their work. And we had also hoped that by doing so, we provide an opportunity to elevate the discussion of science in this, our ecosystem. And I think you would agree with me that from year to year, we have elevated the discussion. So I want us to appreciate all the presenters for their contributions. I also want to thank all of you for participating. It's always a very packed schedule, and it's almost like having a job for 3 days to to sit through and listen to such a diverse range of presentations. You know, in WACCBIP we do everything that is important. We are not focused on one disease or one organism. We do everything that's important. We just provide a platform for good science, so the individuals determine what they are interested in. We're using the platforms that we provide. So because of that, there's a lot of diversity. You had work from bacteria to cancer cells, right? And everything in between. So, you know, I don't take your commitment lightly for being here and sticking through to listen to all our presentations, especially the presentations from our fellows who, you know, really treasure the opportunity to have the audience to talk to you, and some of them presenting to an audience for the first time. You know, so this is something that means a lot to them. So I thank you, the audience, for being here and for supporting all our young people. So thank you very much. I especially want to thank our collaborators who have traveled to be here. This is summertime. Many people would like to go and have a vacation with their family or just take a leave and relax. But our collaborators always make the time to come here and stay for several days, some of them more than a week. Not because they don't have anything to do, but because of the commitment and the belief they have in what we're doing. And so we really appreciate your support and continued collaboration. So let's acknowledge all our collaborators. Now, I'll thank my team last, but before I do that, I always ask for a little bit of feedback. Obviously, you are going to have the opportunity to do the evaluation anonymously. So that has been shared with you. So you can say you can curse and do whatever you want there. But we also give you an opportunity to give feedback to everybody here. So especially those who have been here for the first time. Who has been here for the first time? Who is here for the first time? Okay. So I want those who have been here for the first time to give us their thoughts. What you liked, what you didn't like. And we are not sensitive. We take criticism. So please feel free to tell us what you didn't like so that we can do better. So can we get a microphone?
Ladies first.
Oh.
Okay.
You put me on the spot here.
Yeah, so for the benefit of everybody, just mention your name and the institution you're coming from.
So I'm Francesca Florini. I'm a research associate at Weill Cornell Medicine in New York. I really enjoyed the conference. I think the level of the talks was very good and very interesting, diverse topics. As you said, I like that it's not all malaria all the time or all tuberculosis all the time.
Although there's a lot of malaria.
Yeah.
So.
But that I like.
I'm
so.
guilty for that, but yeah. Okay, good.
Shall I?
And what can we do better?
We need to get a better pointer.
Okay, ICT guys, your pointer is not good. Is it the changing of the slides or the pointing? Which one is the issue?
I don't know. I think it's the slide changes.
The slide change is the issue, yeah. So I think in some years we just put a laptop here to make it easy for people to change slides. But anyway, so we'll work on that. We need a pointer that the buttons are much clearer. I think one of the issues is that these buttons you can't really see. So you can easily press exit or escape instead of next. And I saw that a lot of people would sort of escape and then they have to come back in.
Another
so we
thing
need something that is very clear with arrows on it. Raised arrows so you can see forward, backward, and so on and so forth. Yeah. Yes.
I don't know if this might be me, maybe I missed it, but I think it would have been useful to have a list of the posters. Like if I miss the number, maybe it is somewhere. It's
not in the brochure.
Okay.
So did you download the brochure?
I told you it was my fault. Okay, good.
Yeah.
No,
there's a brochure which you can download. It's much voluminous.
And
we have the program which is sort of light for you to be able to access easily to follow. Okay. But then we have a very elaborate brochure which you should download and keep because it has a lot of information and nice pictures.
And maybe a bit more time for the poster session.
Yeah. We always get that. So yeah, we have to work on that. Last year we had 5 days, so we had more time. Last year we did a whole week because it was our 10th anniversary. But when it's 3 days here, it's a lot. And now we have so many students and all of them want to have a chance to present their poster. So we end up with over 100 posters and then it's very tough. So Peter, we have to see how we can have more time for the posters or have less posters, one or the other. Yeah. Anyway, so thank you.
Thank you.
Are you done? Yes. Thank you very much. Yeah. But please download the, the brochure. Our comms team, they usually spend a lot of time designing the brochure. It takes a lot of their time, and they do a really good job. So please download it and keep it in honor of their effort. We used to print them, nice copies that you can take, but as you know, we have to be more, more sensitive to the environment. So we stopped printing them. We just do the soft copies. So, but you can download it and keep it on your laptop and enjoy when you're on the plane or whenever you're bored. Yeah. And then normally it's on the website. It's on our website. under documents, or what's the tab? Annual updates. It's under annual updates on the website. So all the previous years are there as well. You can always refer to it. Yeah. Next year you may have your picture in next year's brochure, because sometimes the pictures from this year will come in next year's brochure. So, so look out for next year's brochure. You may be in there. Yeah. Anyway, yes. Any other?
Yes.
Yes.
Okay.
Introduce
First of all,
yourself, and then I know you, but others don't. So.
Okay. My name is Tochukwu Dubem Ayaduba, originally Nigerian. I'm from Imperial College London. I'm a postdoc, at Imperial College London. I'm really amazed by the kind of work that's going on here, the kind of research that the students are engaged in, the kind of training that they go through. So I had the opportunity of going to the labs, visiting the labs, and I'm really amazed about what you guys are doing, and it's very, very impressive. So in terms, and I must say that I was humbled by the depth of the knowledge of cell biology. And obviously, before I come next time, I have to brush up on my immunology game so that I can flow with every presentation. In terms of what could be improved on, obviously by next year, this is going to be much bigger, so maybe get a bigger hall.
Yes. Yes, we are also working on that. So maybe you take us to Dangote to give us about $5 million. We have space right here. We can put up a much bigger hall. So yeah, we are working on that. We hope we can get money to put up a new. But you can see that even the labs are already full.
Yes.
You know, at any particular time, we have about 100 graduate students in the building, and then numerous interns and research assistants and all that. So there's a lot of people. There's a lot of density of people. Yes. So yeah, we need another building of this size. You know. So yeah, we're working on that.
Okay, sir.
Trump permitting. Yeah. So thank you. Yeah.
Okay. So finally, I want to ask just a question. So, what would be your advice to someone that would like to, replicate what you've done here, maybe somewhere else? What would be your advice to such a person? Thank you.
I thought Yao gave you the advice. Yeah. Anyway, so I usually tell people that before I started this, I didn't go to any leadership training, formal training for leadership. I didn't sit down and plot some grand idea or anything like that. So it's not as if, there's a master plan somewhere. What it is having the passion for what you want to do and just having the drive and believing in people, believing in young people especially. So we started is just like an experiment. And what really made it work is that the young people really bought into the idea and got excited about it. And so all the energy we have came from the energy of the young people. They gave us the oxygen to keep going and to feed on itself, right? Those of you who have been here over the years, you see that I have lesser and lesser of a role here, right? Because we have stood up a lot of new superstars here. And so I think that really what it is to identify smart people and empower them. That's all that we've done, really. I'm not some special person. It is the people that I've given the opportunity to be here and who have embraced the opportunity and stepped up. That's why we have been as successful as we've been. So I would say that I don't have a magic formula. The only formula I have is that You have the idea, believe in it, believe in Africa, believe in the talent that we have on this continent, find the talent, and give them room to express their talent. And that's really what we've done.
Okay. Thank you. I'm Dr. Oko Olayinka from Anchor University, Lagos, Nigeria. I must commend, I and the professor that came from Nigeria, we are really saying we are from biochemistry. Like me, my first degree, second degree, and PhD is in biochemistry. So the way you've been able to merge cell biology and biochemistry together here as a single department, and the students are able to, merge them together, and it shows in the research, is really commendable. And I give you kudos for that. And that really reflects in the, most of the work that was that were presented. And also, like he said, what you have in the labs and the things you are doing, it shows that Africa can be great if empowered. Many things that people are rushing out there to get, we can replicate them here if the conducive environments are provided. And we hope that such will be done. Like you are clamoring for the Dangotes to come and do things here. We hope that they will listen to us and they will do that. Now, like for the case of the posters, what I would advise is that, let's say we have 100 posters. For the first food day or the second food day, you can concentrate on poster 1 to 50. Then the second day, you can concentrate on posters 51 to 100. You observe that the way the posters were, where they were mounted, most of the people that were at the second floor, there was no, there was not too much traffic there, like the ones at the first floor. So I think we should look into that. I think that's all. God bless you, sir.
Thank you. Yeah, so we've sort of waffled on that. There were some years where we had the posters done, certain particular posters for a certain day, and then take them down, and then put another set up. And then we had some feedback saying that, some people they won't get the time to see all the posters, so keep all of them up. So all the 3 days, people will have time to see those that they want at their own pace, So yeah, we go back and forth, but it's well noted. I think generally the posters, we have to find a way to, now that we have so many now, we have to find innovative ways to make sure that people will see the posters and have time to review them. Yes, please. Please, Leopold's hand was up first. I'll come to you, sir.
Thank you very much. I want to commend your exceptional leadership. I had the opportunity to say this to you in private, but I'm taking the opportunity to say it. And again, you standing up there and requesting this feedback live is still a testimony of that, because things that we say here, I think that you are willing to take forward. And I think you really serve by example. I'm humble and I'm learning, because what you say is something that we can see. I learn a lot. It's an exceptional conference, from basic science to translational science to biotech. I think through the program, we can really see that, when there is a will, there is a way. And I think Yao really portrayed that a lot. We need to really be bold in what we are doing. And I think, your leadership is really paving the way. A lot of points has already been raised about how to improve this. I think what I would just want to add is to continue to commit to engage with a wider community, because I think the more they are diverse group of people coming together, that's really what spices up the idea. Sometimes this type of initiative tend to dwindle when we get comfortable in the same energy with which you wanted to really uplift. Everything here should continue because science is always evolving, and it evolves by engaging new partner rather than getting in a space where you say, okay, we're comfortable, we can do everything by ourselves. Maybe we don't need people from outside again. People from outside always come with fresh eyes and with fresh ideas. So really well done.
Thank you. Yeah, so, yes, thank you for the feedback. We are always open to new ideas and new people. That in fact, that's really what we believe in. So if you, anybody who is here will tell you that we are always building new partnerships and new friendships. In fact, the way we built this network of people is that when we started this, basically, we those who started it together, everybody brought their collaborators on board, and their collaborators brought their collaborators on board, and that's how we grew the network. So we basically started like this and grew that way. And we welcome everybody to come and be part of us. And many people, when they come, they stay and they bring somebody else. So the network just keeps growing. So yeah, we are really, really welcoming to new people, new ideas, new concepts, new everything. Thank you. Sorry.
So I just wanted to add that we had a wonderful experience. This is like the first time in Ghana, first time in Africa.
Oh, wow.
So we had a wonderful experience, and I was really, really impressed with the student presentations, both on stage as well as for the posters. One thing I might just like to add, I don't know how logistically possible that would be, if you can have parallel sessions for viruses, bacteria, protozoa. I mean, I work with viruses, I understand very little of viruses, and much less of that of bacteria and protozoa. Yeah. So it'll be easier for people like me to follow the presentation, and students will get more chance to present. Yeah. I mean, poster is good, but definitely a talk is better. Yeah. So that is one thing, if you can add, I guess it will like add to the conference. Thank you.
Thank you. That's another common feedback we get about doing parallel sessions. We always debate it and go back and forth. We say, okay, let's split up into 3 groups. And then the counter argument is that the students really want to present in front of a big audience and they want everybody to see them and all that. So anyway, so we'll take it on board. It's something we've been talking about, so we'll do that. Okay. Anybody else? Otherwise, we can wrap up. Good. Now, let me ask, there's one at the back, one last one.
Yeah, hello. My name is Flavia, a PhD fellow at WACCBIP. I really enjoyed the conference. Is this your first time? No, it's actually my first time coming in person.
Last year you're not here, okay.
Yes, the previous years I've joined online. Okay. And I don't remember what happened in the previous years for the Fridays I came, but I had a couple of people I wanted to talk to who were big people. And I know usually the lunchtime is the time for networking. Unfortunately, they were at a different place from the rest of us. And so I couldn't really interact with them. So I was hoping that probably next time, maybe after taking their food, they could join the rest of us and then we can have more time to interact with them. Thank you.
Good. So that's a very interesting one, because this is actually the first time we have separated the faculty from the students, and I think it was just due to the numbers, trying to organize better. So maybe we just need more 7 points, but allow the mixture to continue. But yeah, that's good feedback. Yeah, it's the first time we've done it, but you've made a very good point that that's the networking time, which we really treasure, is for us to all be at the same level and chat. So that's very good feedback. We take that. Thank you. All right. I wanted to ask one question before we go. Would you pay a registration fee to come to this conference if you had to? Since we started this, we don't charge anything. Normally we use grants that we have. We've been lucky that we have capacity building grants, so we always budget for conference in our grants. And then we also solicit for sponsorship. You saw some sponsors from businesses that benefit from our operations. So that's how we always fund it. So you don't have to pay anything. Maybe you have to pay your way here, but you don't have to pay any registration fee. You get everything free, food, everything. Now, we've been talking about how is this sustainable? Can we keep finding money to do it without any contribution from participants? And you know, on the other hand, we want people to come, whether you have travel funds or not. You should be able to come. You know, so would you pay to come? Oh, be honest. I want to hear feedback. Would you pay to come? Will you pay a registration fee to come?
Just to comment, I've been to an online conference where there was no logistical arrangements like this, but I had to pay. So I
know, right?
Yeah, I think paying to come wouldn't be a big deal.
Okay. All right. Good. Any other thoughts? Yeah.
I mean, I think the conference is already, you know, making traction, and I think the community, I mean, people will rather benefit to be part of the conference because of the insightful research that are being shared. Personally, I will pay to come, but I think that it's a model for sustainability that will be good to implement, and also to try. The other thing is that, I mean, if you have some resources, you can rather make it on a competitive base, which actually even increase the interest. I mean, say a researcher or a student from outside trying to, you know, apply for compete for a grant to have like a travel award to come. I mean, that's something that can put on your CV. It's still good. So rather than just make it open and free, you know, if you have some funds, you put it on that competitive basis. I mean, you just continue to raise the bar for the conference. But I think it's a model to explore. Thank you.
Okay. Thank you. Okay. Good. So the beers are getting warm, so I think we've talked enough. We'll continue the discussion when we go out there. I want to conclude by thanking my team, right? Like I said, I have been removing myself from this, machine gradually. We, over a period of time, assembled young, talented people from security men to deputy director. You know, these are some of the smartest people we have in Ghana, in Africa. The most committed and most passionate people that we have. And that's why they are here. Because over here, if you don't meet the standard, you wouldn't last here. So those who are here have gone through selection, right? Positive selection. They've gone through positive selection. So we have people here who have been here for 10 years. That means they've withstood my craziness and my high intensity. So I want to appreciate all the members of my team that we call the WACCBIP machine. We've built a machine. So it has different parts. Some people pick you from the airport. Some people make sure you have lunch. Some people make sure you have a name tag. Some people are on Twitter. I mean, it's a whole machine. I don't even know how to describe it. That's why I call it a machine, you know, because there's a lot of people doing different things in different parts of the building for everything to work well. And this is what we do every time when we have workshops, we have scientific meetings. Every time these people do their part. So I want to thank my people for all the hard work. Thank you.
So, it's time for beer. Thank you once again for being here. You are all ambassadors for us. I always say that once you've come here, you are an ambassador for us. You never know where you are, when you are in a meeting, that there's an opportunity to say something about what we do here and how we can get more support. Maybe on a grant panel, and a WACCBIP fellow is applying for a grant, and some of the people on the panel say, who is this, what WACCBIP, what is that? I've never heard that name before. I don't think they could do PCR there. You know, and this happens. People tell me these stories because, you know, believe me or not, there are people who have never heard of WACCBIP and they are not interested in hearing. So they read a grant and there's a student from WACCBIP. And straight away they've made their mind that it can't be. You cannot do, you know, CRISPR in this place. I've never heard it before, you know. So already our fellows are at a disadvantage because they don't buy it. But you have been here, and you are our ambassadors. So tell people what we believe in and the platform we are providing. And together we continue. Thank you.
Thank you. All right. So I have a small public service announcement before we leave. Please, we'll want to ask you to leave your name tags. So if you want, you can take away your name. We'll be collecting we want to recycle. Okay, so please, they would be collecting it.
If you really need it as a souvenir, you can take it back.
You go take my own. I beg take. I'm doing my own.